system service. Hi, so we're now live on YouTube. Wait, what?
Okay, perfect.
NMC room 1, traditional talk. Right?
Yeah, that's correct.
The development of autophagy induced peptides is put in the fine. That's fine. Yeah, cool.
Okay, hello, everyone is already connected. So we will start, so welcome, first of all. I'm Alberto from Blue Brain Project at Geneva, Switzerland. And we will start at 9 o'clock, well, 9 o'clock at here in Switzerland. So at 00 on your clock whenever you are. So today it's the second day of Neuromatch Conference 3.0. I'm very glad that we have seen a lot of submissions and a lot of participation, and also there is kind of a big interest in the conference. And thank you. I will like to thank all the speakers that are here today and that will share with us their work from well, maybe from all over the world. Today we have Germany represented here, but also China and Australia, so spanning a large size of the globe. Okay, so it's 9 o'clock, so I will start. So good morning or good evening all, and welcome to this session in room 1. So today we will have speakers from 3 different continents, and we will hear 3 amazing talks from 3 different topics. That we will start with the macroscopic oscillator dynamics, and then we will go deeper into neurons, neuromodulators, and finally on autophagies. I would like to invite to the stage our first speaker. He's Daniel Kluger, and he's a postdoc at the Institute for Biomagnetism and Biosignal Analysis at the University of Münster in Germany. I'm here with my backend here. It's that is helping with the chat and so on is Aditya. So I invite you to send all your questions on the question and answer panel, or use the chat, and we will use the chat to share information about the participants. And I will be here keeping the time. And so please, Daniel, you can share your screen, and we can wait to hear your talk. And you are muted.
All right, so you can hear me now?
Yeah.
Perfect. Thank you so much for the introduction, Alberto, and thank you for being here and giving me the opportunity to present some findings that some work that I did with Joachim Groß at the University of Münster regarding the interplay of respiration, so the act of breathing, neural oscillations, and eventually behavior. And let's start with what we know. What we know about respiration is that it originates largely in the medulla. There's a microcircuit called the pre-Bötzinger complex that through rhythmic firing initiates rhythmic breathing, and that's basically automatic most of the time. What we also know is that there are pathways to cortical areas. So if you think of meditation or other brain states where respiration plays a central role, you'll see that there's cortical involvement in the act of breathing. So when breathing is not automatic. We also have cortical oscillations, which are markers of brain state. So perception, cognition, action, and these interact with respiration. That's what we know. Like if you think of the meditation example, for example, you actively use your breathing to alter your brain state. Or if you have a panic attack, so a different brain state, then your breathing adapts to that changed state. What we don't know, however, is this direct link of breathing and neuroprocessing per se. So basically we know parts of the puzzle, but we don't know the full picture yet. And there are recent studies showing that respiration phase makes a difference in, for example, perceptual tasks. So you have a mental rotation task, and when the stimulus onset is during inspiration, so you breathe in, then you have a higher accuracy than when the stimulus is presented at another phase. The same is true for physiological markers like the acoustic startle response. So you have an elevated response during ongoing expiration. And there was one intracranial EEG study showing that neural oscillations in a wide variety of brain regions vary with the respiration cycle. So respiration phase modulates these invasively measured brain activity patterns. So that was what we started with. And one potential interpretation is that neuroprocessing and the efficiency of neuroprocessing are modulated by a respiration phase. So we started out with a very simple task. So participants just sat in the MEG and had their forearm raised by 30 degrees and kept it there. So you have this isometric contraction, and they were either breathing normally or deeply. So two conditions in separate blocks. And then we measured MEG, we measured EMG signals, so muscle signals from the forearm, and we measured the respiration. And then you can take that continuous respiration signal, collapse it, and then have like an average respiration. So you have this deep condition with deeper breaths than the normal respiration. And then you can apply a moving window analysis to see how the coherence, so the synchrony between brain oscillations and the rhythmic oscillations in the muscle activity vary over this respiration cycle. So for each time point, you get a coherence value for each frequency. And what you expect to see in these isometric tasks. So if you have a steady muscle contraction, you raise the right arm, so you should see contralaterally over the sensory motor cortices, you should see high coherence within the beta band. And that's what we found in the continuous data. That was just a sanity check. But then if you use the respiration signal and have a phase lock brain signal and muscle signal to that respiration signal, you can see whether that changes, whether there is significant coherence within these two breathing conditions. So you have the deep breathing condition and the normal breathing condition. And we find this beta coherence in phase lock data as well, which makes sense. The interesting thing was first, this reached way into alpha, so that was quite interesting. But what we also saw was that there was a difference between those breathing conditions. So you have deep minus normal here, And you see that the coherence in the beta band, so from 22 to 27 Hertz, is higher for the normal breathing condition than the deep breathing condition. And you can look at this over time here. So this is deep breathing, this is normal breathing, and these are the subject averages of respiration. So along the respiration cycle, you can clearly see that the coherence value in this beta band changes within the motor cortex. So to put it very simply, you have a ramp here. So you have like a decreased coherence here, and then it goes up during expiration. And that's more evident for the deep breathing versus the normal breathing. So that was quite interesting to see that whether you deep breathly or normally has an effect on the synchrony between your muscle and your brain activation on the contralateral motor cortex. And then we went into source space and looked at where this coherence came from, because you see that you can simplify it to this is low and this is high, but there's obviously something more complex going on. So we tested two separate models. So one was the single cycle model that basically follows the time course of respiration. And then we tested a double cycle model, which is double the frequency of the respiration, so a higher order change in coherence. And we localized these two, and you see that the significant sensor patterns, so this is sensor space, of course, this source space, they include the contralateral motor cortices, but they extend it as well. So we found significant coherence for both the single and the higher frequency cycle. So there seem to be separate layers of modulation, so ways in which respiration modulates coherence. And this is just the same in source space. So as expected, you see these activation patterns in the contralateral motor cortex, but also extending forward into frontal areas and parietal as well. So a more extended network than expected, maybe. And one way to interpret this is that the efficiency between, so in the communication between muscle and brain is more efficient during some of these respiration phases, which would be in line with perceptual advantages, during some respiration phases, but not others. So the second study we did was a resting state study. So if you find that breathing modulates neural processing, then maybe you don't need a task to see effects of that. So we did another MEG study, and participants just sat with eyes open for 2 runs of 5 minutes each and were just breathing normally. And again, we recorded MEG and the respiration signal, and after some transformations, you can have amplitude envelopes of different wavelets ranging from delta to high gamma oscillation. So basically from 2 to 150 Hertz. And then we used the modulation index as a way to quantify phase amplitude coupling to see first whether there's any modulation at all. So across the respiration cycle, is there some change in the oscillatory activity across this frequency range from 2 to 150 Hertz. And those are t-values here. So you see there's a basically significant modulation of oscillatory activity across the whole frequency band. So then the second question was, where does this take place? And we used non-negative matrix factorization, so NMF, to basically extract the main components of these modulation effects. And panel A shows you the superficial or the more cortical areas in which we found those modulation effects, comprising frontal eye field, SMA, so you have those midline activities here, ACC, PCC, SMA, which you would expect. And then you have a second array of of components in deeper, so basically cerebellar and brainstem sites. So each of those has a different frequency pattern. So those red shadowed areas are significant modulation. So basically you see that the SMA, for example, has a significant modulation of beta and low gamma oscillations across the respiration cycle. And one step further would be to try to further reduce dimensionality in these data and to group them with regard to their spectral fingerprints, so how frequencies are modulated. And then you can see that you have, for example, this yellow cluster is one midline cluster. So you have the SMA here, and you have the PCC here, and they share a similar characteristic of which oscillations are modulated by respiration. And this gives you an idea how that looks over time. So this is peak inspiration. So this is breathing in and breathing out. And you see the clear peak of both beta and low gamma oscillations around the peak inspiration. And other clusters, such as this deep cerebellar cluster here, has a whole different characteristic. And what was interesting, given that whole brain pattern, was to compare that, as we are in resting state, to compare that to resting state networks. So we just took some of the, let's say, most famous ones. So the default mode network, the dorsal tension network, and the salience network. And then you can look at the mapping. So you have components making up these canonical resting state networks, and we have this this network of components where oscillations are modulated by respiration, and then you can see how they map onto one another. So you have these resting state networks here, and you have this respiration control network, which has been established to be in charge of controlling respiratory activity. And you can see that those deep cerebellar brainstem areas all map onto this this respiration control network, and SMA as a motor area, of course, as well. And what you also see is that that basically all the components.
Sorry, Daniel, could you please conclude? We are out of time.
Sure. Thank you very much. What you also see is that these that basically all the nodes we find in the respiration modulation network are part of those default mode or any resting state network. So what's going to be interesting to see is how that translates in other task spaces. So whether we can actually find perceptual advantages during some respiratory phases, and what the mechanisms are behind that. So in the papers, if you like, there's some speculation about what could be the underlying mechanism. And let me just thank my supervisor, MPI Joachim Groß, for this very, very fun project. And if you like, we can go into some questions.
Thank you very much, Daniel. Very, very great talk. And let me see if we have any question from attendees. Not yet, so maybe I can ask you one briefly. So you have used MEG recording. So why did you choose MEG instead of EEG or other? Is that more suitable technique for your study?
Yeah, for the localization, yes, I would say so, because even if you have a 128 EEG channel, I think the anatomical location of those modulatory effects would be better in MEG, especially for the deeper sources. And you have the advantage on the temporal spectrum, so that's why MEG I would prefer MEG over fMRI. But we're thinking about combining them because EEG has other advantages, over MEG with regard to the directionality of sources. So maybe the next step would be to do a combined EG-MEG study for a final localization.
Perfect, great. Thank you very much. I think we should move on. Now I would like to give the stage to Mengyao Li. So please, you can share your screen.
Okay.
And she's a graduate student in the Peking University in China, and she her lab is developing some new cutting-edge research tools, and especially some advanced imaging probes to untangle the complexity of the nervous system in space and time. So we will see something of that today. So please, the floor is yours.
Okay. Thank you for introducing me, and very happy to join this new match meeting. And today I'm here to introduce my newly developed histamine synthesis and also trying to find some collaborators. So today my talk's topic is inspiring on histamine dynamics by development of genetic encoded graph synthesis. So we know that histamine is very important small molecule, which is known to be involved in many physiological functions. And it is, for example, it's involved in inflammatory responses and has a central role as a mediator of aging. And it could also control blood pressure, gastric acid secretion, and other physiological processes. And in CNS, we can see here, the histamine is act as a neurotransmitter for the brain, and those histaminergic neurons are mainly located in this TMN region, in the posterior hypothalamus, and send their axons to the whole brain. And those histaminergic neurons active only during the waking, and they could form a function as a maintain wakefulness and attention. And their interaction with other neurotransmitter systems could link basic homeostasis and other higher brain functions, including the sleep-wake regulation. And here are some current methods for detecting histamine. And we know a good sensor should at least fulfill this 5 aspects. It should have good sensitivity, specificity, the small molecule, and also good temporal and spatial resolution. And it would be very good for the method to be non-invasive. And people previously have used this microdialysis method to detect histamine. But this microdialysis method could be very invasive and loses spatial and temporal resolution. And people also use FSCV method to detect histamine, but it could also be very invasive. And also people develop this assay as an imaging method to detect histamine release. But since this temporal assay is based on the downstream gene expression, so it would lose most of the temporal resolution. So to fulfill those 5 aspects, we developed this graph sensor. And the graph means the GPCR activation-based sensors. So the basic principle is we inserted this DPJP to the nature design this molecule sensor, the GPCR. And this DPJP is conformational sensitive. So when the light binds to the GPCR, it will induce the GPCR has conformational change, and this conformational change could be transduced to the DPJP, so induce the fluorescence increase. So based on this strategy, we've already developed several neurotransmitter sensors, for example, acetylcholine sensor, dopamine sensor, epinephrine sensor, and peptide sensor. So also based on this strategy, we want to develop this histamine sensor. So as I previously have introduced, that our graph sensor is based on the nature design, the GPCRs for the small molecule. So for histamine, the nature is designed for at least 4 histamine receptors, H1 to H4. So firstly, we want to select that's the backbone for developing the sensor. So we inserted the CPGP to different HRH receptors, H1, 1, 2, 3, 4, and we found that H4 shows the best membrane trafficking. And then based on HRH4, we've done several rounds of optimization, and finally we got this AJ1H sensor. and it shows good membrane trafficking and also good response to histamine. The pattern affinity of this H1H is around 10 nanomole. And we also use local pulse method to detect the kinetics of the histamine. So we puff 100 micromole histamine to the cell and use lens scanning method to detect the signal change. We found that the on kinetics is around 0.5 seconds, and the off kinetics by puffing the antagonist is around 4 seconds. And also we want to characterize the specificity and specificity of the sensor. So we add different concentration of different monoamines or other neurotransmitters such as ACH, glutamate, and alpha to the histamine sensor. And we found that this histamine sensor, H1H, only responds to histamine and show good response, but do not show any response to other neurotransmitters or monoamines. And since this is a H4-based sensor, so it can be blocked by this J&J, the H4 specific antagonist, but not other three antagonists. And as neuroscientists also want to apply our sensor into the nervous system. So firstly, we tested the H1H in culture neurons and found it also have very big membrane localization. And it could respond to histamine dose independently. Also, the apparent affinity is around 10 nanomole. And also in culture neurons, it's also very specific and can be blocked by only by H4 antagonists. And then we want to apply our sensor to the nervous system to see if it can really reveal the endogenous histamine release. So firstly, since I have introduced that histamine is very important in the sleep-wake cycles. So we use our sensor to detect the sleep-wake cycle histamine dynamics. So we injected this H1H sensor or mutant sensor, which do not have response to histamine, to the MPF3 region of the mice, and use a photometry method to detect the histamine dynamics. And at the same time, we would record the EEG and EMG to reflect the sleep-wake condition of the mice. So this is one process of the recording. We can see here this blue region shows a wake condition. We can see the histamine signal is largely increased during wake, and it will be decreased during the REM sleep and also the non-REM state. And this is the quantification data of our sensor signal. We can see here during the wake condition, the sensor signal is the largest. Also, we use this mutant sensor, which do not have the response to histamine as an actual control, and we can see here in different conditions there's no significant histamine signal change. And also, we give the mice a pharmacological treatment. We give the mice ciprofloxacin, which is an H3 receptor antagonist. It is reported to could induce wakefulness and increase the histamine level in the mice. So also we can see here after injecting the ciprofloxacin to the mice, the wakefulness and also the histamine level is largely increased. And here is the quantification result. And we also treat the mice with another drug, which is caffeine. Caffeine is reported to promote wakefulness, but do not enhance histamine release. So as previously we have done, we can inject the histamine to the mice, and we can see the mice can wake for a longer time, but histamine level do not have significant increase. And this is the quantification result. Also, as I previously mentioned, that H4 antagonist would largely block our sensor response. So we also injected this J&J to the mice, and we can see the sensor signal is largely blocked. And so this is the all the data for the H1H. And as a tool development, we also want to have sensors with different dynamics and also with large response. So based on this H4 structure model, we have tuned the HA synthesis affinity. As shown here, we got this HA1M, which have 10-fold affinity decrease. And also we want by developing the histamine synthesis, we could inject to other histamine synthesis of nature from other species. For example, those different animals, they have different histamine synthesis, and especially this water bear-based HRH1, it seems to have the best performance yet. As shown here, the y-axis is the response, and the x-axis is different receptors from different animals. And so based on this water HRH1, we developed this HA2.0. We can also say it has good membrane trafficking, and it is brighter than the HA1H and also has larger responses. And the apparent affinity is shown here. It's around 500 nano. And we also, as previously have shown, we quantified the kinetics of this HA2.0 in the Hux cells, and the toll on is about 0.3 seconds, and the toll off is around 1 second. It could be much faster than HA1H. And also, we want to test the HA2.0 in ultra neurons, but it seems that the maximum response is not as good as HA1H, but since this is brighter than HA1H, so we think that the HA2.0 have a better signal-to-noise ratio. Also, we use the HA2.0 in living mice to see the spontaneous sleep-wake cycle of the histamine dynamics. It shows the same kind of result. And at the end, I want to thank Dr. Li for guidance, and to Dr. Wei and Chunling for collaboration. And this is our lab. And at advertisement, we have other 3 good sensors to show 3 days later. Down here, this ECB sensors, endocannabinoid sensor, the fiber-T sensor, also a set of neuropeptide sensors. So that's all for my today's talk. Questions are welcome.
Thank you very much, Mengyao. Very interesting talk and very interesting technology. And we have a question from the audience from Elena Jacobs.
Have you tried characterizing this sensor with a passive calcium indicator? Yes, well, actually, we are trying to do this, but the result is quite preliminary. So, so that's a good, indeed, that's a good suggestion, and we're doing that.
Okay, thank you again, and thank you for answering the questions. So I will move on, and we are right on time. Thank you again, Mengyao. And I would like to welcome our next speaker. She is Azin Amin. If you want to start sharing, perfect. She is a third-year PhD student at the Florey Institute of Neuroscience and Mental Health in the University of Melbourne, Australia. And she now we are going really deeper into the neuron, and especially into motor neurons. So please, Azin, the floor is yours. And if you have questions for the speakers, you can write them on the question and answer box, and you can also upvote other questions if you like them. So please.
All right, great. Thank you so much for the introduction, and thank you for giving me the opportunity to present my research. So basically, my project focuses on developing peptides to treat the neurodegenerative disease ALS, which is also known as MND. So I don't know whether anyone remembers, but this is from the Ice Bucket Challenge, which was where people had to dump cold ice water over their head. And this challenge was designed to raise awareness for this disease ALS. So amyotrophic lateral sclerosis, or ALS, is similar to other neurodegenerative diseases in the sense that there is an accumulation of misfolded proteins into these clumps or protein aggregates. And these accumulate in the motor neurons of patients, which then contributes to the degeneration of motor neurons. As a result of this, all these signalings to muscles get cut off. And so muscles start to waste away. This muscle wasting spreads around the body, and once it reaches the respiratory muscles, it causes death in the patients. And this usually happens 2 to 4 years after the onset of symptoms. So what's more is that there is no cure for this disease, and the only 2 treatments available, which are riluzole and edaravone, they can only extend survival by a few months. So there is a great urgency in trying to find a treatment for this disease. Normally in cells, there is a process called autophagy, which keeps the cells healthy by degrading down these misfolded proteins, protein aggregates, dysfunctional organelles, and any other toxic agents. So autophagy has 4 important steps. In the first step, a pre-autophagosomal structure forms in the cytoplasm. Then a complex which has this Beclin-1 protein as its core component. This LC3 protein gets lipidated to LC3-2 and gets incorporated into the membrane. And then this complex fuses the membranes together to form the double membrane autophagosomes. This same Beclin complex is then involved with the fusion of autophagosomes with lysosomes and the formation of autolysosomes. It's then in these autolysosomes where the enzymes degrade down all of this cargo, and any generated macromolecules gets recycled back into the cell. So we are thinking, because in ALS there is an accumulation of these protein aggregates, we are thinking that this autophagy pathway might be downregulated or dysfunctional. So if we could find a way to upregulate this pathway, we might be able to degrade down these protein aggregates and prevent motor neuron degeneration in ALS. So to upregulate this pathway, we focused on this Beclin-1 protein. So Beclin-1 protein is the master regulator of autophagy because it's involved in both the early and late stages of autophagy. And a lot of proteins interact with these different domains to regulate autophagy. So for example, the 3 identified domains of Beclin-1 protein are BH3, CCD, and ECD. From previous research, we know that this ECD region of Beclin-1 on its own is sufficient to induce autophagy. So we wanted to make peptides derived from this ECD region. So this is the whole ECD region. So for the first peptide, we decided to focus only on this yellow bit, and we named it Beclin-1. And then we decided to extend it from the N-terminus and named it Beclin-2. For Beclin-3, we decided to include the whole antiparallel beta strand. And for Beclin-4, we excluded this random code. So it's just that beta, random beta code. Oh, sorry, the beta strand. So these peptides were designed and synthesized. But as many of you might know, so peptides are great for drug candidates because they're small and they're very selective towards their target. But a major limitation of peptides is that they cannot penetrate the cell membrane, and so they can't get into the cell. To help my peptides get into the cell, I used a fragment derived from this apolipoprotein E. So why did I choose ApoE to help my peptides get into the cell? Because ApoE, compared to the other cell-penetrating peptides, is safe because it's an endogenous protein, and on its own, it has shown protective effects in injury and neurodegenerative disease animal models. ApoE is also potent because not only can it interact with the anionic proteoglycans, but it also has its own receptors on the cell membrane. And because most of these receptors are in the brain capillary endothelial cells, they are more selective towards the brain. So that's what we want. So it makes it selective towards the brain. And also our lab has shown previously that ApoE can cross the blood-brain barrier, which is great. But another problem we encounter is that once the peptides get into the cell, they will likely get entrapped within endosomes, and get degraded or recycled out of the cell. So to address this problem, we used the fragment derived from this hemagglutinin protein, and we named it HA2. So basically what will happen is that once the peptides get into the cells, they will get entrapped within endosomes. But then this HA2 peptide can disrupt this membrane and help our peptides escape into the cytoplasm and go and activate their target. So in the end, 13 different peptide analogs of Beclin-1 were designed and synthesized. Next, our question was whether they can upregulate or induce autophagy. To measure autophagy activity in cells, we use this reporter, which is the mCherry EGFB LC3 reporter. So I mentioned earlier that this LC3 protein gets incorporated and inserted into the membrane of autophagosomes. And because now in here it's tagged with EGFB and mCherry, autophagosomes will appear as yellow puncta. But as autophagosomes go on to fuse with lysosomes, the pH drops, and so EGFB gets quenched and only mCherry would be visible. So our autolysosomes will appear as red puncta. So by measuring the number of yellow puncta, we will know the level of autophagosomes, and by measuring the number of red puncta, we will know the number of autolysosomes. So if something is increasing autophagy, we should see more red puncta compared to yellow puncta. So we did that, and this is my result. So on the x-axis, we have the different peptides that were derived from this Beclin protein. And I have the two controls here. So rapamycin is my positive control. So this is the gold standard autophagy inducer, and is currently in phase 2 clinical trials for treating ALS. And bifidobacterium A1 is our negative control. So it inhibits autophagosome fusion with lysosome. So it prevents autolysosome formation. So as you can see, two of my peptides were able to increase autophagy significantly and even higher than our positive control rapamycin. And these are our controls. So basically, out of all these 13 different analogs that we synthesized, two of my peptides, HA2-ApoE-Beclin-3 and HA2-ApoE-Beclin-4, were able to induce autophagy. So our next question was whether they can reduce the protein aggregates that are in ALS. So ALS is mostly sporadic, but 10% is familial. And within those 10%, these are the genes that have been identified. These are some of the genes. But the most studied gene is the SOD1 gene, which encodes the superoxide dismutase protein, which is an enzyme in the body that converts harmful superoxide radicals into oxygen and hydrogen peroxide. But what happens in ALS is that these SOD1 proteins become misfolded and they aggregate. So I wanted to see whether my peptides can reduce these SOD1 aggregates. So I did Western blotting, and I looked at the insoluble fraction because that's where the misfolded proteins will go. So I looked at the wild type SOD1 and also the mutant SOD1. In this case, I'm looking at A4V. And as you can see, my peptides didn't have any effect on wild type SOD1, which is great, but they were able to significantly reduce the number of insoluble mutant SOD1 or the misfolded SOD1. To confirm these results, I also did confocal microscopy and I counted the number of cells containing these EGFB aggregates. And again, as you can see, my peptides were able to reduce the number of SOD1 aggregates. So these peptides can upregulate autophagy, they can reduce SOD1 aggregates. Our next question was whether that would translate to a beneficial effect to the health of the cells. So from previous studies, we know that mutant SOD1 activates a cascade of events, which leads to caspase 3 activation and eventually apoptosis. So I wanted to see whether the peptides can reduce caspase 3 activation. So in here, again, as expected, expression of this mutant SOD1 or A4V increased caspase 3 activation, but this was decreased back down after treatment with our peptide. And our controls here are HA2-ApoE scrambled Beclin 4, which is our negative control, and astarosporine, which is a drug that induces apoptosis, as our positive control. So in conclusion, HA2-ApoE Beclin 3 and HA2-ApoE Beclin 4 were able to increase autophagy in cells. And these were 2 to 10-fold more potent than the previously developed peptides. From this, we can conclude that the HA2-ApoE is required for the activity of peptide because Beclin 3 on its own wasn't able to induce autophagy, but it had to be conjugated to this HA2-ApoE, and the same with Beclin 4. And because Beclin 3 and Beclin 4 were most effective compared to Beclin 1 and Beclin 2, we can conclude that the inclusion of the antiparallel beta strands gives the highest activity. We also showed that the Beclin peptides can prevent mutant SOD1 aggregates, and as a result, they can reduce cleaved caspase 3 activation, which is mediated by mutant SOD1. And finally, I'd like to thank my supervisors and everyone at the MND lab, and thank you for listening.
Thank you very much, Azin, for your great talk. And for staying in the time. So thank you very much. Let's see if we have any question from the attendees. None right now, but I have one myself. So I'm kind of far away from your specific research area. What I wanted to ask is how. So, as far as understood, those are in vitro studies. Are you expecting to translate this approach also in vivo to see the same but on an in vivo study?
Yes, so currently I'm actually working with LC3 mice, which works exactly as that reporter, but it's just in mice. And yeah, I'm injecting my peptides into mice to see an effect. And if I do see an effect of autophagy activity, if I see it in LC3 mice, then I'm gonna go into SOD1 mice and see whether that would have any beneficial effects in an ALS mouse model.
Okay, perfect. That's I think that's the direction where you want to go to approach in the end and kind of find a solution for this disease. Okay, so if we don't have any more question, we are exactly ending at the time we are supposed to. So I would like to thank all the attendees who were with us today, and of course to Azin, Mengiao, and Daniel for their wonderful talks. So I encourage you, if you have additional questions or if you want to have some more details, approach them on Twitter or write them. I'm sure they are very happy to share their knowledge and their experience. So I will conclude the session here, and thank you again to my super backend Aditya for helping us making this session great. So enjoy the day or the evening according to your time zone, and thank you again, everybody. Bye bye. Enjoy the conference.
Thank you.
So hello everyone who is connected. And right now our first speaker is missing, so we will start with the second talk, but we will start it at the time it is supposed to start, so at 15 past the hour. I will put a screen on saying that. Okay. Hello, everyone. Hello, all attendees. I'm Alberto Antonietti from Blue Brain Project, Geneva, Switzerland. And sorry for for starting later than expected. So the first talk of today, we had to skip it since the speaker was missing. But we will start on time for our second talk today. So first of all, I would like to thank all of you being here, and also I want to thank the speakers for the talk they are about to give, and the back end of this room, Ali, and she is helping me in making this work, and thank you very much. So we will have only two talk and not three talk in this session, and the two talk that we have today have in common the topic, in the sense that they are both related to adaptation and learning, and we will start right on time with the second talk of today. So with the talk of Emma Ward. She did her PhD at the Donders Institute in the Netherlands, and she recently started a postdoc in Berlin at the Freie Universität. And her main interest is how expectation influence perception, and how that develops in infancy, and how that changes in between autistic and non-autistic people. And we will are going to hear more about her interesting research. So please, Emma, the floor is yours.
Thanks, Alberto. Hi, everyone. I'm just making sure that I'm sharing the correct screen. Okay, can you see the slides now?
Yeah.
Okay, perfect. All right, so as Alberto said, I did my PhD at the Donders Institute, and this is work that I did there along with Jan Buitelaar and Sabine Hunjes. And essentially, my PhD was about evaluating predictive processing accounts of autism. And these are theoretical accounts that claim to unify all symptoms of autism and explain the foundations, the cognitive mechanisms behind autism, by saying that autistic individuals assign higher precision to their prediction errors than non-autistic individuals. So essentially that would mean anytime somebody encounters an observation that does not match their model of the world, an autistic person would be more likely to update their model than a non-autistic person would. And this has a few consequences for perception. So incoming sensory input should therefore be seen as more novel by autistic participants, and experiences that bias perception in non-autistic individuals should bias autistic individuals less. So therefore we use adaptation because we know that we can bias perception in non-autistic individuals. And adaptation is essentially the phenomenon that exposure to stimuli on one end of a continuum biases later perception away from that end. And it's been shown to be reduced in autism in some studies, but also equivalent in autism in some studies. So we were curious. And as far as we know, there's so far nothing about the neural mechanisms underlying this in autism. So we used a similar task. And here's an example. So what you see is a continuum here from Ben Affleck on the left to Heath Ledger on the right, with evenly spaced face morphs, and the central face is exactly half Ben Affleck and half Heath Ledger. And what we're going to do now is expose you to one end of a continuum. So we're going to look at the face all the way on the left, which is Ben Affleck, for 5 seconds. So if you look at that one with me, 1, 2, 3, 4, 5, and then look at the center, it should look to you more like Heath Ledger now. Whereas if we look at the right one, so we're going to look at Heath Ledger now for 5 seconds, 1, 2, 3, 4, 5, and then look at the central face, it should now look more like Ben Affleck. So nothing changed about the physical stimulus, but your recent experience biased your perception. So if prediction errors are weighted more highly in the autistic group, they should experience this shift in their perception less. So they should show less behavioral adaptation in their responses and less differentiation of ERPs to adapted and unadapted stimuli. And we tested this not with face identity, like I just showed you, but with gaze direction, because this is also a nice continuum. And we have very clear neuromarkers of gaze direction and face perception. So, and these stimuli have also been used before in behavioral studies with autistic kids. So essentially what they saw was a familiarization in block 1 of gazes of 0 degrees, which is directly looking at you, or 5 degrees to the left or 5 degrees to the right. And then they had this long exposure like we just did in the second block with one very extreme gaze direction. And then in block 3, they were reminded of that very extreme gaze direction and again saw gaze of 0 or 5 degrees and asked to respond which way the person looked. So it looked something like this for them. So in block 1, they would see a trial like this. First, just the face, and then after the offset of the face, the question, which way did they look? And they responded with a button press. And then another one, which is 5 degrees. And after the offset of the face, respond with a button press, which way did they look? And then in block 2, they got this very extreme gaze direction. And then they were exposed to this for a few minutes. And then in block 3, they again saw the original faces with smaller gaze direction and were asked which way they looked. And because they were exposed in block 2, remember, to this extreme gaze direction to the left, they should perceive all of these faces after that as looking less left because they're biased away from that end of the continuum. So we did this with 55 teenagers, 26 autistic kids and 29 non-autistic kids, and they were all aged between 12 and 18, and the mean age of both groups was just a bit over 15. The groups didn't differ in their short form IQ scores, but they did differ in their social communication questionnaire scores, which is nice because that's a measure of autism symptoms. So on this continuous dimension of autism symptoms, our autistic kids were higher. And for the behavioral analyses, we used ordinal logistic multi-level modeling. And for the ERP analyses, we used cluster-based permutation testing. So I will first just talk you through the results of the non-autistic kids and then add the autistic kids after. So if you see here just the accuracy in block 1, so that's before they were exposed to the adaptation effect, you see accuracy was between like 60 and 100%, and that's actually quite good. I don't know if you noticed that these 5-degree gaze directions are quite subtle, so the kids did very well. And in block 3, post-adaptation, they were less accurate, but that makes sense because we wanted to bias their perception. We wanted them to perceive it not as it really was. And then if we add the autistic kids in blue, you see there are almost no differences at all. So the autistic kids did just as well as the non-autistic kids. And then if we look at the actual adaptation effect, here you see the response that they gave. So did they say that this person was looking in the direction they were adapted to, the direction they were not adapted to, or direct? So we've now normalized over left and right because some kids were adapted to left and some kids were adapted to right. And you see before they're adapted, they don't differentiate between which they've been adapted to and which not, which is good because it hasn't happened yet. They're about the same, and they're more often saying that the people look direct, which makes sense because the angles are quite low. And then after the adaptation, you see exactly what we expected, that the direction they're adapted to, they respond less because they're biased away from that direction. And then they respond much more to that those gazes are direct because they're quite subtle, but they're just shifted. And if we add in the autistic group in red again, in blue again, we again see almost identical results from the autistic and non-autistic kids. So there's no difference here between groups in how they respond to the adaptation. Okay, and then if we look at the ERPs, essentially what we were expecting was that there would be specific repetition suppression to the direction that people were adapted to after the adaptation. So here in green, we expected smaller responses for both directions because as the experiment went on, we get habituation anyway, but more repetition suppression to the one they were adapted to. And this was just an example, but if I show you now, the autistic group at the top and the non-autistic group at the bottom, you see neither of them show that pattern. So there's no differences for either group between adapted and unadapted, and no difference between the groups. And this is a mean of all 10 parietal and occipital electrodes, and that's based on a previous paper that we wrote, but sorry, but it's also the same pattern of results if you just look at P7 and P8 based on some other adult studies. So it really just doesn't seem to be there. And then we were also interested in general habituation. So if all incoming sensory input is perceived as more novel to autistic participants, you would expect they would habituate less in general. So we just took all responses from block 1, subtracted all responses from block 3 and plotted the difference wave. And again, you see autistic kids in blue and non-autistic kids in red. And the bold part is significant clusters. And you see that both groups habituated, but the groups did not differ. So at the bottom, we just plot them next to each other, and there's no significant clusters there. So we didn't see any group differences, which is inconsistent with predictive processing accounts of autism. And other findings tell the same story. So other papers that I have written and some colleagues with teenagers and younger kids do not find any evidence of predictive processing accounts of autism. And then, of course, this is really important for multiple reasons. So any account of autism should also address development, because autism is a developmental disorder. But on the other hand, I think any kind of general cognitive or neural mechanistic claim about how the brain works should also apply to younger kids. So a developmental perspective is important for predictive processing in general and for predictive processing accounts of autism. So it seems like the theory needs to be adjusted. And like one promising avenue that I can see at the moment is maybe taking into account different precision weighting at different levels of the cognitive hierarchy, which is stuff that people in the schizophrenia field are really working on a lot at the moment. And it sounds really interesting, but hasn't really made it over to autism yet. But I think in general, if we want a theory to be able to explain all of autism, we need to really make sure that it can address different domains, different ages, and that's not happening yet. So that was what I have for you. Does anybody have questions?
So thank you very much, Emma, for the very, very nice talk. So let's see if we have questions. It appears that we have one. Yeah, from David. Could it be that the lack of differences between group is related not enough statistical power? In the last peak, there seemed to be a trend. Perhaps Bayesian approach could help.
I'm assuming in the last peak, he means something about the ERPs. So yeah, in a previous paper, I did this exact paradigm with 2-year-olds who have autism in their family or not. And then we obviously didn't have button presses because there were only 2, but we did use Bayesian statistics on the ERPs. And essentially, I was not really happy with how we implemented the Bayesian statistics on the ERPs and therefore didn't do it here. I would be happy to do it, but how we did it then was I used JASP and then I had to take just one value from the ERP. So I took a peak and then did a t-test between the groups. I would be really happy to do it if you know anyway, because now we use this mass univariate testing on the ERPs, comparing every time point. If you would know how to do Bayesian statistics that way, I would be happy to do it.
And David is referring to the last image, he says.
The last image, oh, the habituation. Yeah, so indeed, this is the only place in the 2-year-olds that we did see a difference between the groups. So if anything, I think we see more of the habituation thing, but it's in the opposite direction than we would expect. So there's more habituation in the autistic kids than the non-autistic kids, and that does not fit with predictive processing, which says there should be less habituation in the autistic kids. But that's also the direction that we saw in the 2-year-olds. So if that was also here, then that would be really interesting. What I would say about the power is that at least with the ERPs, at least we do have very nice small error bars. So I think we have relatively clean data.
Thank you very much. Then we have other two questions from Benji and from Bianca. They are kind of related. So first of all, they say it's very cool work, and they are asking if the choice of so I direction, gaze direction is kind of implicated, or since, well, we are talking about autism disorder where social impact is kind of related. So maybe the choice of eye gaze as a stimulus could have an impact. And so do you want to say something about it?
Yeah, thanks. So actually, I really wanted to use a non-social stimuli because I think if it's more domain general, then it's more interesting to me. But my supervisor convinced me very well by pointing out that we have really good known ERPs for face and gaze discrimination. So I think there was a really strong reason to use these stimuli specifically, also because they had already been used in a behavioral study with autistic kids. But we did eye tracking because we knew that it might be a problem that the autistic kids might not look at the eyes and then they would not adapt. But in fact, our lack of results, our lack of difference between the groups shows that the autistic kids were looking at the eyes, right? Because they were influenced by the gaze direction. But we did compare the groups, and the autistic kids made fewer fixations to the area of interest around the eyes, and less of their fixations were to the area around the eyes, but they did spend about the same amount of time looking at the eyes, or they didn't differ in the mean duration of the fixation to the eyes. And apparently, even though they made less fixations there, it was enough to get the information because they were indeed biased in their perception by information that was only in the eyes.
Perfect. Thank you very much. So we have other questions, but unfortunately we are kind of out of time. So thank you very much, Emma. I encourage everyone who has a question left to reach out to Emma on Twitter or at her email address. So thanks again.
Thank you.
And then we can pass to our next speaker and last speaker of this session. So we will still see something about learning, not properly adaptation, but more spatial learning. So she is Charlene Tessereux. She is a PhD student at the University of Nottingham, and we will post the details in the chat. And I just wanted to tell you that she's looking for a postdoc. So if you are looking for a postdoc, please listen to her talk. So thank you, Charlene, and the floor is yours.
Can you see my slide okay?
Yeah, they are.
Okay, cool. Okay.
Perfect.
Hi, everyone. So my name is Charlene, and I'm going to present today some reinforcement learning approaches for rapid place learning in the Moyes Water maze. So being able to navigate our environment is a crucial ability, as it enables us to find food and shelter, for example. And in order to face everyday spatial navigation task problems, the brains have evolved amazing capabilities to adjust to various new situations. And one of them, for example, is when you go shopping, you park your car and you are able to find your car back after your shopping. So this means that you have formed a representation of where your car is, that you are able to retrieve this information when you need to get back there, but also that you are able to select a successful trajectory that will lead you there. And the underlying mechanism that enable this type of flexibility are the focus of ongoing research. And one of the ways to access this mechanism is to use rats in, for example, the water maze task, in which rats are put in a pool of cloudy water where they have to find a hidden platform here. And what happens in this apparatus is that if we represent the time that rats take to reach the platform across repeated exposure, so this is one trial, which is one swim to the goal location, what happens is the time decreases with repeated trials, showing that the rats are able to learn how to navigate to the platform efficiently across time. And we can also assess flexibility in this apparatus with changing the goal location every, for example, in that case, every 4 trials, the goal location will change. And what we can see here is that the second, third and fourth trial to the same goal location are very much shorter than the first trial, showing that one exposure to the new platform location is enough for the rats to take almost a direct path on the subsequent trials towards it. This is the equivalent to when we find our car back after our shopping, and this phenomenon is not a one-shot learning. And during my PhD, I've been trying to focus on building biologically plausible models of this one-shot learning behavior. And we know that the hippocampus is involved in spatial navigation, and especially in the case of the water maze. So hippocampal lesions, animal can still learn to navigate to a fixed goal location, but they are much slower to reach good performances. However, when the goal changes every 4 trial, for example, if hippocampal lesions animal are fully impaired, so one-shot learning is completely abolished by hippocampal lesion, as can be seen here, as we don't observe this drastic reduction in the time taken to reach the platform between the first and second and subsequent trials. So I have investigated how could hippocampal place representation be linked to behavior using reinforcement learning models. And in our case, we focused on an actor-critic architecture in which the agent here that models the rats in the water maze is the agent position is encoded within a place cell, a layer of place cell units mimicking hippocampal place cells. So that means that when the agent is close to their favorite location here, shown in gray, the place cell will be mostly active, and when the agent is further away, then the place cell will be much less active. And this position encoded within the place cell layer is fed as an input to a critic network. The critic network is used to compute how much reward can the agent expect to receive from every location in the maze, knowing that rewards are only given at what mimics the platform location. So basically the value function here computed by the critic is an estimation of reward expectancy in the future. And the same place cell input is used, the same place cell information is used as an input by an actor network whose role is to estimate what is the best direction to choose in every location. So from the actor network activities, we can then select a direction to follow so the agent can move to the subsequent state, where then it can compare the value of the next state to the value of the previous state. And this information is called a temporal difference error. And this temporal difference error can be used to update the connections between the place cell and the critic and the actor network, so that the critic can improve the estimation of the value function over the space, and also that successful moves, so moves that led to states that are higher in value, can be are more likely to occur again in the future. So this is how it looks like with repeated exposure to the fixed goal location. What you can see in the left here is that the agent can learn to navigate to a fixed goal location. And this implementation is partly biologically realistic, as we know that the TD error as neural correlates as in dopaminergic signals, but also that dopaminergic signals are involved in action selection, but also there are neural correlates of actor and critic, especially in the striatum. However, it is unclear whether the particular actor and critic roles are held within different or same neural population. So the dichotomy between actor and critic that is proposed in the model needs to be investigated further in the brain. And also the plasticity between of hippocampus striatal connections does not seem to depend on dopamine, which is what's being proposed as a learning mechanism in the model. So, but the main limitation of this agent is that it's not flexible to changes in goal location. As you can see here, when we change goal location, the agent takes many trials to reach good performance. However, rats are very fast to adjust. So in order to I've been trying to investigate ways to bring up flexibility in the water maze, and I'm going to present 2 ways to do this today. One of them is has been proposed in 2000 by Foster, Morris, and Diane, and consists in learning the coordinates throughout the maze using the place cell inputs. And these estimations of the coordinates can be used to compare the estimated goal location to the estimated position of the agent. And this comparison leads to a goal-directed vector that if our estimations of the coordinates are correct, then will directly lead the agent to the goal. And what we can see here, if we compute this, if we implement this model to a task where the goal location changes for trial, it reproduces the adaptation to new goal location that is also seen in animals in the Morris-Water maze task. And this implementation contains the important idea that a map-like representation of location within the maze enables efficient readout, which is the underlying key idea behind the idea of a cognitive map for spatial navigation. Moreover, there are neural correlates of goal vector informations in the hippocampal formation. However, there are a certain number of limitations currently. One of them is there are extensive experimental results showing that new goals locations induce changes within the place cell representations, and currently this approach uses place cell information to actually encode the goal location. So this doesn't seem to fully fit with experimental results, but also, and more importantly, the initialization of the model is unrealistic, as, for example, coordinate action is only available from the second trial. So we have proposed a new way to perform flexibility in the Morris-Water maze task, and in our case, the agent needs to learn how to reach different goal locations over the maze. So the actor and the critique network that leads to a particular goal location, which is what we call a strategy. And at every time step, the agent has information to whether the current strategy is good or not to lead to the current goal location through a goal prediction error. The goal prediction error computes the difference between the predicted error from the strategy to the observed error. So this goal prediction error will mainly be informative at goal locations, so whether they have been predicted but are not encountered, or they are encountered but have not been predicted before. And we use this goal prediction error as an input to a confidence parameter, which determines the level of confidence that the agent has in its strategy. So that if the agent is very low in confidence, then it will explore more its environment, so therefore not stick to its strategy really much. But however, when the confidence level is very high, then it will exploit its strategy more because it trusts that the strategy is correct for the current goal. So I'm going to show you a short video of how this looks like. You can see here the blue star is the agent that is currently following the strategy that leads to the goal J. And when it reaches the goal J, as for this trial, the real goal is in the yellow circle here. It has received a negative goal prediction error that then decreases its level of confidence and leads the agent to explore its environment. And after a bit, maybe the agent will find the goal, which is what happens here, and then can select a strategy that will maximize the goal prediction error, and then on the subsequent trials, go to the goal using this strategy. So the key here is that the confidence parameter evolves on a slow time scale. So if we implement, if we see the behavior of the rat over different repeated trials, when we change the goal location every 4 trials, what we can see is that it mimics the adaptation of the rats in the Morris-Water maze task. So, and moreover, this implementation is partly biologically realistic, as we know that prefrontal activities correlate with goal prediction errors and are involved in flexibility in spatial navigation. However, the main limitation now is that it requires pre-learning to goal locations, but we are currently working in improving this approach using a statistical sum of the strategies. So just to summarize, we've seen that a simple actor-critic architecture can learn to navigate to a goal, but is not flexible to changes in goal location. And we've seen two ways to bring up flexibility in this task. One of them uses coordinate-based representation of location in the maze that enables comparison between the goals and the agent location for goal-directed navigation. And the second one uses hierarchical computations that enable to separate different levels of control at different time scale. So to this, you can check my new preprint that has just been accepted for publication. And I want to thank the organizer and my supervisors and the audience also.
Thank you very much, Charlene, and congratulations for the acceptance of your work. And so, very nice talk. Let's see if we have questions from the audience. So I really love these kind of models to understand how the brain works. And if we don't have questions, I have one from my own. So in the water maze paradigm, okay. Okay, we have a question from Emma. I will read it. Emma wanted to ask Charlene if she can say something about the small increase in confidence during a random walk when the agent is not finding the goal. Is there a reason for that?
Yeah, it's the equation that governs the dynamic of the confidence level. It's got a very slow time scale, but it still will come back to a zero value by default. So that's the way currently it's encoded, which, I mean, it's not perfect, but that's equations. Yeah.
Okay, perfect. Thank you for answering that. And so my question was in the water maze protocol, the mouse, the animal doesn't have any landmark, right, to identify where he is.
Oh, it does. I mean, it uses, so not closed landmarks, but it uses landmarks like cues around the maze. So there are far away landmarks. Yeah.
Okay, perfect.
Yeah, which
so
are not present in the model actually.
Okay, yeah. I just wanted to understand if it could rely on some external landmarks to orientate. Otherwise, I couldn't see how the place cells could fire related to where he is.
Yeah.
Okay, perfect. So well, we have just a comment, so that the idea behind small increase of confidence could be rewarding the agent to at least keep going. Okay.
Well,
So thank you very much again to Emma and to Charlene, and also to Ali, our backend host for this talk, for this session. So I will close the session right on time, and thank you all the attendees for being here, and please reach out to the speakers for other information. Thank you again, and enjoy the conference.
Thanks, everybody.
Thanks.
Bye-bye.
Bye.
So welcome to everybody who is joining. We will start pretty soon. So we will start at the hour. So it's 11, almost 11 here in Switzerland. Okay, Monica is back. Okay, we are almost ready to start. So I would like to welcome you all to this session at Neuromatch 3.0. So today we are going to start a great session, all live, all interactive talks. So for the interactive talks, we'll have 5 minutes for the speaker to present a few slides and to introduce the topic, and then there is more room for discussions and question and answers. So to the speaker, please in the Q&A box type on screen so I can put you on screen to discuss with the speaker. And otherwise, you can just type your question, and I will write them for the speakers. And so I really encourage you to be very interactive. This is an interactive session, so we need your active participation to make it So the session has a kind of a common theme, which is the development and neurodevelopment. And we will start with Dr. Monica Tambalo. She will talk about cortical organoids. And she's a postdoctoral fellow. She's sponsored by the Fondazione Umberto Veronesi. And she's working at the Humanitas Research Institute in Italy, in Milan, which is my hometown. So I'm Alberto Antonietti from the Blue Brain Project at EPFL in Geneva, Switzerland, and I will leave the floor to Dr. Monica Tambalo. She recently moved from London back to Milan, and she will tell us about the organoids, brain organoids. So please, Monica, the floor is yours. And you are muted. Okay.
Hey, can you hear me? Yes.
Yeah.
Hi, everyone. Thank you for the nice introduction. And yes, so today I will talk about this project that I recently started in Simona Slodato laboratory in Milan. And just to move to the topic, I will just give you a brief introduction. And so we're interested in cortical development and to study brain development in general. And what is becoming apparent in recent years is that electrical activity within developing neurons is actually playing an important role in shaping brain development itself. And what has been shown is that electrical activity and calcium transient within the cells are able to influence gene expression and also the epigenetic of the developing neurons. And this altogether has a strong impact on brain development and circuit establishment, where it affects cell proliferation, cell death, as well as neuronal migration, morphogenesis, and also cell fate. So we want to study the impact of electrical activity on early cortical development. And this is particularly important because in some situation in the clinic, we have patients and children affected with very severe form of epilepsy that affect the babies even in the first months of their age, and in some cases also the fetus. So in these cases, the children have really a strong burst of epilepsy and of electrical activity, and we are wondering how such a strong epilepsy and strong effect can have an impact on the formation of a brain circuit. And so to study this problem, we are now using brain organoids as a model system. And in particular, we have set on a protocol developed by Paula Arlotta Lab in the US. And this was published last year by Velasco et al. And this is a protocol that allows us to drive cell differentiation towards cortical fate, and in particular in a very robust manner. So we are able to obtain cortical organoids that are really similar among each other and also similar across batches. So in this way, we can address questions about cell fate decision and also the molecular fate of these cells and how epilepsy, and I will show you our protocol to study epilepsy in a 3D context in a dish. So since I just recently started, I have just a preliminary result to show you. And I can show you that we are able to generate cortical organoids where they recapitulate some aspect of brain development and of cortical development. In particular, we can see that there are some rosettes of progenitors developing in the early phases of maturation of the organoids. We also see neurogenesis going on in the tissue. And with time, we have increased complexity of the organoid itself with presence of outer radial glia and also astrocytes. And importantly, we can detect a variety of neuronal subtypes, and among them, the cortical projection neurons are developing in a dynamic that resembles the one of the human developing cortex, with deep layer neuron forming first, followed by upper layer neuron. So we are confident that we can recapitulate key aspects of cortical development in vitro in a 3D context. And now we are moving on and trying to study this and to use this model really to have a human model system for infantile epilepsy. So what I'm currently doing is to use the cortical organoids to trigger epileptic-like currents by using a proconvalescent agent. And so we hope to have these established in the next months. And in parallel, we also obtain iPS cell lines from patients carrying mutation in two loci. One is the EIEE24, that is a mutation in an ion channel. And then another one, GLUT1, is a mutation in the glucose transporter. And these patients are affected with very severe forms of infantile epilepsy. So we're going to soon derive cortical organoids from these iPS cell lines. And then we're going to address cortical development in these different organoids carrying different backgrounds. And we're going to again trigger on these organoids epileptic-like currents and study downstream what are the effects in the establishment of a cortical tissue. So we can address at the cellular level how the circuit assemble after such strong epileptic currents. And then we also want to understand what is the molecular footprints left by epileptic currents into the tissue. And for that, we're going to use single-cell multi-omics approaches where we can profile the cell at the transcriptomic level as well as the epigenetic level to see how different cell populations are responding to such strong electrical activity. And we're finally going to use this information to infer gene regulatory network involved in epilepsy. And with these, we hope to identify some modules important for epilepsy that hopefully will help drive the identification of the novel treatments for infantile epilepsy. And this is basically the approach we're going to take, but it's just ongoing because I just recently started. So with that, I will finish by thanking all the Lodato Lab for the help and support, our collaborators, and in particular, Davide Pozzi for the help with the electrophysiology of the organoids, and also our collaborator that kindly share the iPS cell lines from different clinics in Italy. And as Alberto mentioned, my project is funded by Fondazione Veronesi in Italy. And thank you.
Thank you very much, Monica, for your presentation, and it has been great and very interesting project, the one that you started. and I really hope that you will find very interesting results. So let's see if we have any question from our attendees. If you want to ask a question directly, you can either write on stage in the Q&A box, and I can promote you to talk and ask your question directly, or you can just write it in the Q&A box, and I can read it to the speaker. While questions are coming, I have one question myself. I'm not very familiar with organoids. I wanted to ask you which is the, let's say, the hardware, the platform that you use to stimulate or to record from the organoid. So is it a multi-electrode array or something similar?
So we are starting with that. So we just started with that, but there are evidence in the literature that by just recording in acute on this MEA chip, we can actually record from the whole organoid. So that's something we are just starting to set up in the lab. But also we can use calcium imaging to image calcium transient within the organoid. And so we are mainly working on these two, and eventually some patch clamp as well for the cells.
Mm-hmm.
But the MEA chips are the ones that are preferentially used in the organoids community.
Maybe I could ask something.
At what age? So it was a great talk and a great idea. At what age are you planning to study the electrophysiology? Because I've been working a lot with organoids, and this is
so we are now tweaking a bit the protocol to try to enhance electrical activity. So I'm doing that currently. So we're gonna start by monitoring at different months. What it has been reported is after already 2 months of organoid culture, you can see some activity, but we just need to push it a bit to see it that early. So I was thinking about 3 months of age onwards to start analyzing them.
Mm-hmm. Okay.
So I have a follow-up question to my previous one. So using a multi-electrode array, for example, since you have a 3D organoid, so I assume you could record, let's say, only from the back surface. Am I correct?
Yes. So that's exactly one problem, that you record just the superficially the organoid. Some people, and we're also trying to slice the organoids to have a more interface of the organoid with the chip. And then I believe there are also some new chips coming out with some pillars. So this pillar will kind of be more central in the organoid, so you can eventually record from those. So there are alternatives that we can use to be more, to have more elaborate recording.
Okay, I see. Thanks for the clarification. And any other question for the panelists or from the attendees? Oh, we have one maybe. We have a question from Chloe. What is your hypothesis on what will be most affected, neurogenesis or connectivity or something else?
Yeah, well, I think it will be a combination of things that will be affected. So we are interested to look very immediate downstream to see the transcriptional changes that are known to happen quite quickly downstream of electrical activity. But on the long term, we are not too sure what will be mainly affected, but probably cell-fate decision, some migration, and maybe some level of cell death or balanced cell death and apoptosis. So probably a different type of neuronal cell type will be affected probably a bit differently, because now is emerging that different cell types respond differently. So that's why we want to do at the single cell level to see how the different populations respond. But yeah, I have to say early in early development, there is not too much known. It's mainly known later on in mouse adult brain development. So it will be interesting to see.
Okay, thank you again, Monica, for your presentation and for answering our questions. So we are perfectly on time, and we can move on to our second speaker, which is indeed a colleague of Monica. She's Rosalba Proce. She's working at the. Sorry, Rosalba. There is a bit of an echo, I think.
Yes.
Okay. Yeah. Okay. Maybe when I don't talk, then. Okay. No, I think it's gone now. So she's a research assistant at the Humanitas Research Hospital at the Laboratory of Developmental Neurobiology. Again, with the PI is Dr. Simona Lodato. And in her presentation, she will show us her master thesis project and where that has been done at the lab where she is finishing the project as a research assistant. So, well, first of all, congratulations for the graduation, for the master graduation, and we are going to again to look at the brain development. And so, please, Rosalba, the floor is yours.
Thank you so much, Alberto, and thank you all for being here. So today I will talk to you about this project that is mainly focused on understanding the role of the axis 6CR4, 6CL12 in cerebral cortex and cerebellum development in a particular mouse model. This axis is really important in the cerebral cortex because it participates in the regulation of the interneuron migration. In fact, the interneurons, which are the inhibitory neuronal population, born in the ventral part of the telencephalon, and they, once they are born, they migrate tangentially in order to reach the cortex. Once they reach the marginal zone and the ventricular zone, they change the migration into radial in order to enter the cortical plate. And this changing in the migration happens thanks to different signals and also thanks to 6CR4 and 6CL12. In fact, if we don't have 6CR4 in the mutant one, the positioning of these interneurons in the cortical plate is misplaced. On the other side, we have the cerebellum. Here, this axis is important because it regulates the proliferation and positioning of the Purkinje cells in the cerebellum. In fact, if we don't have the receptor 6CR4, we have an abnormal architecture of the cerebellum and also an abnormal positioning of the Purkinje cells. This axis is also important because some mutations that occur in 6CR4 are responsible for a rare immunodeficiency disorder called WHIM syndrome. These patients have is an immunodeficiency, so they have different immunological problems related to retention of the neutrophils in the bone marrow, decrease the number of T and B cells, but also they have some neurological defects related to global coordination, some affective problems related to anxiety and withdrawal behavior, and also recently they found with the MRI an abnormal orientation of the cerebellar foliar. Recently, a research group generated the WHIM syndrome animal model, which recapitulates all the immune phenotypes. But what is not known if these mice recapitulate also the neurological defects. So the purpose of this project is to analyze the impact of this WHIM mutation in this animal model in the cerebellum and in cerebral cortex development, and the impact also on the behavior. So looking at the results that we obtained so far, we performed a morphological analysis at P7 and P30 sagittal section of the cerebellum, and we found a morphological defect in the architecture of the lobule 3 and lobule 4-5 in the cerebellum, meaning that it's shorter. In fact, we measured the Purkinje cell layer, and we found that it's reduced at both P7 and P30 mutant mice. And also we measured the area of these cerebellum, both P7 and P30, and we found a reduction at P30 mice. Then we looked at the cellular level in the cerebellum, and we measured the number of the Purkinje cells, and we found a global reduction in the total amount of these Purkinje cells. On the other side, we were interested in looking at the migration of the interneurons in the cerebral cortex. So what we did was to counted the numbers of somatostatin positive interneuron in the somatosensory area. And what we found is a mild defect in the distribution of the interneurons in the somatosensory cortex. Then we looked at the behavioral level with some tests like the open field and the self-grooming test. And what we found was that these WIM mice stay longer in the corner of the arena, not exploring the rest of the arena in the open field. And this behavior seems to be related with a sort of freezing behavior, suggesting an anxiety-like behavior, confirmed also by the increased grooming activity. So in conclusion, these WIM mice seems to have a neurological phenotype. But of course, further studies needs to be done in order to understand a possible correlation with the human disease. And at the end, I would like to thank all the people in my lab for their support, the Calicordis lab because they gave us the mice, the Matteoli's lab because they helped us with the behavioral test, and Professor Badolato for the information of the patients. And I would like to thank all of you for your attention.
Thank you very much, Rosalba, for your great talk. And let's see if we have questions from the audience. In the meanwhile, I have a few questions myself. So I have in the last years I've been working a lot with the cerebellum from a computational point of view, but let's say that I know the structure pretty well. So it was interesting to see how the WIM syndrome physically changed the shape of the cerebellum and the numbers of neurons. What I wanted to ask you is this. So you say that there are some affective misbehaviors in the WIM syndrome. Since the cerebellum is mostly involved in motor task, well, not only motor task as far as we know, but mainly on motor learning and adaptation. Have you the WIM syndrome? Does it have also motor impairments? Does it create also motor impairments?
Well, in the patients, they found a mild defect in global motor coordination. In fact, we planned to do some motor tests also on our mice in order to see if they recapitulate also these motor defects with some behavioral tests like the Rotarod test. But yes, they have some, they call it a global motor coordination defect.
Okay, that's clear. Thank you. And could it be that? So I'm not an expert in behavioral studies, but could it be that the difference exploration of the cage could be, let's see, related to not only to an let's say affective or cognitive dysfunction, but also to a motor problem. So maybe the mouse or the animal is less prefers to stay in a place instead of exploring the cage or the field. Could it be a...
Yes. Well, when we looked at the open field can give us a lot of information. In fact, what we found was that the time that the mice spent in the corner was increased. But in order to discriminate if it was a problem of motor coordination, we looked at all the areas in the arena, the center, the walls, and the corners. And we found that the motor activity that in the open field is measured with the vertical counts and the vertical time is reduced in all the arena, meaning that probably they are not moving more as the control mice, but since they are doing this in all the arena, is not doing probably to a motor defect. Also because the ambulatory rate, that is the sort of how they move inside the arena, is reduced only in the corner, meaning that their behavior is showed mostly in the corner, meaning that they have some, how can we say, maybe fear or anxiety. They just stay in the corner and decide not to move, probably related to some affective problems. And in order to confirm this, we need to do more studies, more tests, like the self-grooming, which only measure the time of the mice that are grooming, and also the dark light chamber, which is an anxiety-related test, in order to confirm this.
Okay, thank you for the explanation. And then we have a question from the audience, from Chloe. So thank you, Chloe, for your question. She's asking if you observed any deficit in the brain immune cell microglia. Could they play a role in the defects that you are observing?
Well, this is really interesting because 6CL4 and 6CL12 axis is also present in the microglia. So actually it's something that we want to look at. We would like to look if the microglia is affected, meaning that looking at the cell number of the population, and also how they are expressed maybe in the brain, in the different areas, also in the cortex or in the cerebellum, maybe with some qPCR experiments. So yes, this is really interesting, and maybe it could be affected since we don't know, because of course it's immunodeficiency. So we have to think about that. Is still the immune system also in the brain could be affected.
Okay, perfect. Thank you for answering that. So if we don't have any more questions from Rosalba, maybe we can thank again Rosalba for our talk, and we can move on to our next and last speaker for today. So we stay in the area of neurodevelopment, and our next speaker is Antti Krontira. She's a PhD student at the Binder Lab at the Department of Translational Research in Psychiatry at the Max Planck Institute of Psychiatry at Munich in Germany. She will present about the molecular pathways of glucocorticoids effect on neurodevelopment. Please, Antti, the floor is yours. And please ask questions to our next speaker.
Thank you very much. Yeah, so I'm Anthea. I work with Elizabeth Binder at the Max Planck Institute of Psychiatry in Munich. And I'm going to talk to you a bit about how disturbances of the environment can affect brain development. So a step after what we've heard. Okay. So we work in the lab on the developmental origin of health and disease hypothesis, which states that disturbances during critical periods of development can have long-term effects in health even many years after. And we see that in the context of mental health. So if the mother during pregnancy suffers from stress-related psychiatric disorders, such as depression or PTSD or ADHD, or she receives synthetic glucocorticoid treatment, which is something that actually happens a lot in the Western societies during late last trimester because it helps with fetal lung development. All of this increase the glucocorticoid signaling from the mother to the fetus. And this has actually been associated extensively in the literature with different behavioral and psychiatric problems for the offspring after birth, such as attention deficits or cognitive impairments. So we wanted to summarize how glucocorticoids can mediate all of these behavioral effects, and we saw that excess of prenatal glucocorticoid signaling reduces the ability of the placenta during pregnancy, of course, to act as a protective barrier. It increases the basal functioning of the HPA axis of the offspring after birth, and it has different effects on neurodevelopment on the brain level with reduced cortical thickness, on the brain region level, for example, in the amygdala with increased volume and connectivity in girls, and at the cell level with disturbances on the proliferation of progenitor cells, which in two-dimensional cultures leads to decreased neurogenesis and increased gleogenesis. One of the ways that glucocorticoids mediate all of these effects is molecularly, is through the epigenetic landscape. And this is something that was studied in our lab and was published last year. So here we use the human hippocampal two-dimensional line, and we gave dexamethasone. We treated with dexamethasone. Dexamethasone is a glucocorticoid that is synthetic and is very specific to the glucocorticoid receptor. So when we treated these cells with dexamethasone, and then we stopped treating them for 20 days, even after that, we saw that there were some CPGs that stay hypomethylated, even after the washout of the stressor. And these CPGs, these genomic loci, epigenomic loci, we call them poised states. This is very interesting because the genes that are associated with these poised states have increased transcriptional responses after a second stressor, or they are genes that are developmental, so their expression profile is very well regulated through development. And because of this glucocorticoids and treatment and the differences in the epigenetic landscape, this expression profile of these genes changes. So this is what I'm going to focus today. I'm going to focus on how the developmental transcription factor mediates the effects of glucocorticoids on neurodevelopment. And this transcription factor is called ZBDB16. And we were very interested in ZBDB16 because we saw that from the UCSC genome browser that it has active enhancer elements that you can see here with yellow on the brain and specifically on fetal human brain samples. There is a glucocorticoid receptor binding site exactly on these active enhancers during human neurodevelopment. And more interestingly, there are hypomethylated CPG sites that you can see here highlighted in blue on these human neurodevelopmental enhancer elements. And for example, this one exactly where the glucocorticoid receptor binds. So the first thing I want to understand was when and where during human neurodevelopment this transcription factor is expressed, and for that I used cortical organoids. Here you can see a ventricle, and I used them very early on in their development and very late. So here you can see at the transcriptional level, but also at the protein level, that the expression of ZBDB16 is very dynamic, with high expression early on during development, and when the neurons start to arise, that you can see here with green, the expression falls practically to zero. The next question was if glucocorticoids can really change the expression pattern of this transcription factor. Here you can see a ventricle, ZBDB16 is with red, and you can see that it's expressed in all the progenitor, PAC6 positive cells that are with green. At day 50 is not expressed anymore, and when we give dexamethasone for 7 days, we can bring back the expression of the progenitors, and actually we'll bring it back exactly at the levels that it was before it physiologically went down. So what we wanted to understand what that means for brain development, and for that reason I use the mouse. Here you can see a mouse cortex at embryonic day 16, so after the majority of neurogenesis has happened and ZBDB16 is not expressed. When I give dexamethasone, I get overexpression of this protein, so I can use the mouse model to study brain development for ZBDB16. So what I used is in utero electroporations, where you give a plasmid at the ventricle at embryonic day 13. I started, it's when neurogenesis starts in mice, and this is a control plasmid, and analyzed at embryonic day 16. So most of the green cells have become neurons and have migrated to the cortical plate. When I give the overexpressing plasmid for ZBDB16, there is a different distribution of cells, with most of them staying in the subventricular zone. When I try to understand what these cells are, and here you can see a staining for apical radial glia with PAC6 and intermediate progenitors TBR2 positive cells gray. You can see that when I give the ZBDB16, there are a lot of pink cells, so overexpressing cells for both markers, TBR2 PAC6 cells. The majority of TBR2 cells are double positive. And now the scheme of the developing brain looks like that. So the apical radial glia, instead of becoming intermediate progenitors, they become this cell state, the PAC6 TBR2 positive cells that proliferate a lot. And actually these cells are proliferating, they create more progenitors and they create more neurons, deep layer neurons, which I'm not showing you. So if I want to summarize, we started from the glucocorticoids effects on the epigenetic landscape that change the expression profile of this developmental transcription factor, which causes different brain development in the mice. And now we're trying to understand how this happens, what is the molecular pathway. And we think actually this is one of the ways that glucocorticoids mediate their effects on behavioral and psychiatric phenotypes through neurodevelopment. So I want to thank my lab, my supervisor, Elizabeth Binder, also Silvia Capello and Cristina Chirusi from the developmental lab in the MPI of psychiatry, and all of you for listening.
Thank you very much, Antti, for your presentation. So let's see if we have questions coming from the audience, but also from other panelists. That is good too. And in the meanwhile, I just want to tell you and highlight that in the chat you can I posted two recent papers, one paper and one preprint from Antti. So if you want to go deeper into the details, you can have a look at them, and also I would like to highlight the opportunity to write papers from young audience, so from elementary school to high school. There is a special journal which is Frontiers for Young Minds, and I've also posted a recent paper that Antti has published there, and I encourage you to look at it. So we have a question from Elina Jacobs. I will write it to you. I will read it to you, Antti. Is there any evidence from human adult studies that you may have some similar cortical changes in the conditions you mentioned in the beginning of your talk, either from post-mortem studies or potentially MRI, though probably not enough resolution in the latter?
That's a very good question. That's always a very good question when it comes to psychiatry. So there are the effects of prenatal stress or prenatal glucocorticoids on behavioral outcomes are very well studied, with MRI, without MRI, with like questionnaires, behavioral questionnaires. The effects on the actual development of the cells have been studied in humans with MRI. Claudia Bush is a lab that does a lot of these things, and she has shown that actually there is, for example, enlarged amygdala due to prenatal associated with excess of prenatal glucocorticoids, and it's an enlarged brain region, and we think one of the ways, or the way that it is enlarged, is because during development there were more progenitors, so more neurons were created. It has been shown that there are effects of prenatal excess of glucocorticoids specifically on reduced cortical thickness, on reduced folding, which is also goes in line with effects during neurogenesis, so when I'm studying. In mice, I'm not sure if there is a study on prenatal excess of glucocorticoids on the cell identity. There are many studies on behavior, many studies on the brain region level, but not on the cell. There are a lot of studies though with, there are 5-6 studies with glucocorticoids treatment in 2-dimensional human cultures, which show actually exactly what I showed you for ZBTB16 overexpression, so increased proliferation of the progenitors. These studies actually cannot, of course, because they're 2-dimensional, cannot differentiate between progenitors, and they've also never shown what is the molecular pathway. We think that ZBTB16 is the molecular pathway, and we're trying actually to decipher, like I told you, which type of progenitors, which type of neurons are affected, and which type of glia is affected.
Thank you very much for your answer. So I think we don't have any open question. So
sorry, maybe I can ask.
Oh yeah, sure.
Yeah, Antti was really nice. I was wondering if you looked also at the epigenetic related to ZBTB16, since I believe it can act with HDAC to modify the acetylation.
You're right, it can. We haven't yet, but we're planning to do that. We have single cell attack data from our organoids, and they are being analyzed, so we need to see. We have FAC-sorted single cell methylation data from our organoids before and after glucocorticoids. They're also being analyzed now. So we want to study that. And the other thing I am doing now is we have a technique in the lab that is bisulfite sequencing, but targeted on specific genes. So now I'm trying to do targeted bisulfite sequencing on the locus of ZBTB16 before and after glucocorticoids. What I can tell you is that up until now, ZBTB16, through its interaction with HDACs, was considered a repressor. We think that in the brain it acts as an activator. That's what I can tell you. It has been published that it acts also as an activator. We think in our neural cells that it acts like an activator. So that's where we're focusing on.
Okay. So I think we can wrap up the session. So I would like to thank you, Antti, Monica, and Rosalba for their wonderful insights in their work, and I hope that all the attendees have enjoyed as much as I did. And thank you also for the question that that you asked. So to thank you, Chloe and Elina, for your questions. And a final thank you to Jelana, who was the back host for this session. So thank you again, everyone who has attended, and please reach out to the speakers to have more information or to connect with them to discuss more during these days of NeuroMatch conference. And enjoy the rest of the conference. So see you. Thank you all.
Thank you.
Thank you.
Bye.
Bye.
Hi, Irene.
Hello.
So welcome. First of all, so I was explaining how this works to Natasha before. So we will start at 15 minutes past the hour, since our first session talk has been cancelled. So at 15, Natasha will start, and then at 30 minutes, you will start. And so this is an interactive session. So you have 5 minutes to present your slides, more or less, and then there is more room for the Q&As from the audience or between the panelists. So until we, well, for this first 15 minutes, I will put a slide telling the audience that we are starting at 15 with Natasha's talk. Do you have any questions before we start? Oh, maybe maybe before I do that, Irene, would you like to try to share your screen to see if everything is working fine?
Yeah, sure.
Perfect.
Let's try that. Okay.
Okay.
Yeah.
Okay, we see the Acrobat Reader editor.
Yeah, I just have to do like full screen.
Okay,
and then
perfect.
That's fine. Yeah.
Okay, yeah, now it's full screen. Perfect. Okay,
cool.
so everything is set for later. Okay, see you in just a few minutes.
See you then.
So hello everyone. We are almost ready to start. Just a couple more minutes until we reach 15 minutes past the hour. Okay, I will start this this session. So the first, as you have seen in the message posted before, we had to start this this session 15 minutes later since our first talk has been cancelled. But we will start right now. So this session will have two interactive talks. So we will have 5 minutes for the presenter to show some slides, and then more room for Q&A, question and answers. So please type your questions in the Q&A box. And if you want to ask the question directly in person to the speaker, then just type on screen so I can promote you and let you ask the question directly with your own voice. And maybe in this way we can also have a more live discussion between speakers and attendees. So this session will be about emotions and different stage of our life. So we will start with childhood, and then we will pass to motherhood and related emotions. So I would like to ask Natasha Lopez Fischer. She is our first speaker today to start sharing her screen. And Natasha, if you are there, okay, perfect. So if you want to start sharing your screen, she is a postdoctoral researcher. Sorry, at the Center for Family and Population Research at the National University of Singapore. And here she is going to present a work with EG correlates of prosocial behaviors in young children. And when she's not working, she also likes to explore different cuisines and recipes and meet friends and watch Netflix series, not exactly and necessarily in this order. So please welcome Natasha, and the floor is yours. You're still muted.
Okay. Let me just, okay. So everybody is hearing me now? Okay, great.
Yes.
So thank you, Alberto, for the introduction. And hello, everyone. So again, my name is Natasha Fischer, and today I'll talk about my work named Advantageous Inequity Awareness in Young Children, the whole of late positive potential and emotional reactivity. So to begin with the presentation, I want to give some background information and introduce some basic concepts such as morality, fairness, and inequity aversion. So as definition, morality can be understood as the code of shared values, informing about proper conduct within a social group. One of these values is the pursuit of fairness and inequity aversion during social interactions. Fairness may be conceptualized as a way to access whether resources are distributed equally or close to equal among peers, whereas inequity aversion is the natural tendency to avoid unequal outcomes. These behaviors are present since early childhood, but there is little to none evidence about how they are computed by the brain during early ages. Therefore, our goal was to identify how young children perceive moral values. By doing so, we intend to contribute to the development of educational interventions tailored according to a child's developmental stage. To achieve this goal, we adopted a child-friendly version of the dictator game task, and in our task, each child played the receiver role, while the dictator role was played by the computer. This later fact was unknown for the child. The dictator was the one in charge to decide how many candies the child should get. 3 different offers could be made to the child. An advantageous unequal offer where the child would get more candies than the dictator. A disadvantageous unequal offer where the child would get less candies than the dictator. And a fair offer where both child and dictator received equal or close to equal amount of candies. I'm sorry, again here. So after each offer, child was then asked about how he or she was feeling, and answers could be very sad, sad, just okay, happy, or very happy. We recorded the EEG signal during the entire task. We analyzed behavioral EEG data from 17 children whose ages ranged from approximately 3 to 6 years old. Behavioral data was analyzed using a non-parametric analysis of variance, and EEG waveforms were analyzed in time domain using Monte Carlo approach with cluster-based correction for multiple comparisons. Regarding behavioral responses, we could find that preschoolers felt sadder when they received less candies than their peers compared to when they received more or equal amount of candies as their colleagues. In the other hand, they did not report feeling differently when receiving more candies than their peers, comparing to when they received equal or close to equal amount of candies. Regarding the EEG data, we could first observe that for early components, children had a statistically significant high EEG amplitude in time intervals corresponding to P100, N100, and N170 ERP components after receiving different types of candies allocations. These ERP components were previously linked to fast visual perception of different kinds of stimuli, such as facial expressions and emotionally charged stimuli. Furthermore, we could observe that for later ERP components, young children may not recognize having more candies than their peers as something bad due to statistically significant low levels of emotional regulation at early stages. This could be demonstrated by the higher LPP amplitudes after advantageous offers compared to when they had fair offers. Previous studies already demonstrated high LPP amplitudes for emotionally charged stimuli. We could also observe that during the LPP time interval, higher the EEG amplitudes were, higher emotional ratings given by the participants were, as well as it's seen by the regression analysis we did by the right side. So altogether, the results suggest that young children may not recognize a situation where they potentially have an advantage over others as something negative. We also found that Their increased emotional reaction to rewards may potentially influence their ability to properly identify unfairness, especially when it's directed towards others. These investigations shed light on how children construct and process moral values and may help to design efficient educational interventions to improve social skills since the very beginning of their development. So if you have further questions about the work I presented here, I will be happy to provide further clarifications and discuss my results. You can also send me an email, which is shown here in this slide, and follow me on Twitter. Thanks very much for the attention. I'm looking forward to hearing your questions.
Thank you very much, Natasha, for your presentation. And I shared in the chat your Twitter handle, so everyone can follow you on Twitter to ask follow-up questions or in general to follow your work. So we have one question. Okay, perfect. So we have a question from Ilya. What do you think the N170 reflects about the differences in allocation? Is that a common component in this literature?
Yeah, so actually N170 it's more commonly related to facial recognition and actually emotional facial recognition. In this literature about resource allocation, it's not that common, but there is some authors arguing about the function of early ERPs in social relevant stimuli, such as in children. It seems like children, as far as I could observe, it seems children, they have this early ERP is more proeminent than adults, especially when it comes to social stimuli. But it is often overlooked because most of the, not most, but some of the literature, they assume that children, they have the very same time intervals, the very same ERP time intervals as adults, which may be not the case. So there is some discussion about what is the function or what is the role of early versus late ERPs in social stimuli processing in pediatric populations. So there is also like a very good review paper about the dual processing of fairness computation, and this paper, it's just about like how the early and late components can address these different moments of appraisal of moral situations. Yeah.
And
yeah, it
as
can,
a follow-up,
it can be. Yeah, I see the question. Yeah, it can be different. Actually, there is like some literature. A paper from the 1980s, around 1980s, showing, for example, that the alpha band from infancy to 6 years old, the ranges are below the common ranges as adults, mostly because children, they are still having their brain networks building up, and also the scope and the school, it's not so well matured yet. So it seems to have some differences across adults and children. So it's worthy to investigate this type of influence when you're dealing with pediatric populations.
Thank you for the answer, and thank you, Ilya, for the questions. And I have I don't see any further question from the audience, so I have a question myself. So I was curious about the fact that in your study, the child involved has an advantage in one case. So the inequality, let's say, could be advantages or disadvantages for him or for her. Did you also observe, or someone else observed, if children reacts to inequality toward someone else? So if he is not directly involved in the deal.
Yeah, actually not in my study, but there are some literature saying that children can punish the agents of inequality, like they can ostracize or they can punish somehow the agents of inequality when it's directed towards others. It's possible, but this happens mostly with older children, like after 6 years old, I would say, because there is like a time-dependent development of morality. So, for example, when it comes to inequity aversion, there are some literature already showing that children, they mostly exhibit disadvantage and equity aversion. So they are since early ages, like since 15 months old, for example, they are already avoiding situations where they are having a disadvantage, but they do not avoid situations when they are having advantage, which is very interesting. This type, because the advantage is inequity, it's somewhat it's not so intuitive to have like this aversion to advantageous situations. So this is something learned, and this is something that is just developed after 6, 7, 8 years old. And it also depends on cultural norms. So to have this type of moral value, it seems that they have to downregulate their own emotion reactivity. So that's why I'm showing that it's probably it's the case for adults and also probably the case for children.
That's really super interesting.
Yeah,
really cool.
It's really nice. Yeah.
Okay, so since we don't have any further question, I think we can. Well, thank you again, Natasha, for your talk. And if the attendees have further questions, just reach out to her on Twitter or by email. So I will then pass to our next and last speaker for this session. She will tell us about motherhood, and she's Irene Sophia Plank, and she's a PhD student at the Einstein Center for Neuroscience and Berlin School of Mind and Brain in Germany. Okay, she's starting sharing her screen, and her main research interest is how we can understand other people and how context and life experiences influence this understanding. And her PhD project focuses on the factor parenthood and how it affects specifically empathy and theory of mind. So Irene, please, the floor is yours, and I welcome everyone to ask questions during the talk and afterward.
Thank you. Okay, so yeah, my talk is on the question whether motherhood is a moderator of empathy for pain, and these are the results of an fMRI study. So. Okay, so the background here is that we know from a few studies now that empathy can be modulated. So depending on who is the target of our empathizing, we have more or less empathy with this person. And it also depends on who is the one who is doing the empathizer. So both the one who empathizes and the one who is empathized upon influence how much empathy we experience. And we also know that empathy is quite important for parenting. So parents, empathy actually facilitates a positive development of their children. And there is also quite substantial overlap between the parental brain and affective processing areas, as well as specific empathy regions. So this led to our hypothesis that mothers have a higher activation in empathy-related areas than non-mothers in response to empathy for pain, and that this effect might be stronger for children than adults, so that it is specific to that they are using it in context of their parenting. The design that we used was that we tested 50 women, 25 mothers. The women were all between 20 and 50 years old, and they saw pictures of people either being in a painful situation or in a neutral situation, and those situations were well matched. So for example, someone uses a knife, and the picture of them just using the knife versus a picture they used a knife and they actually cut themselves. So that was the first manipulation of the pictures. The second manipulation of the pictures was whether the protagonist of this picture was a child between 4 and 6 years old or an adult. And the task of the participants was to put themselves into this person's position, kind of imagine that you were this person who is in this neutral or painful situation. And additionally, because sometimes it was quite hard to tell whether it's a child or an adult, we had a face at the beginning of each block showing, for example, please imagine that you're in this child's position or in this adult's position. And also to ensure that we actually have a difference between these painful and neutral pictures, and to ensure that we have the participants' attention, we had painfulness ratings after 50% of our pictures. And just very briefly, those are the behavioral ratings for this painfulness question inside the scanner. So we have here separated the neutral and the painful pictures, and we can see that there is a huge difference between the neutral and the painful pictures, but there is no significant difference between the adult stimuli and the children's stimuli. And there's also no significant difference between the mothers and the non-mothers ratings. So there are no differences in the ratings, just between the painfulness manipulation of our design. Our pre-processing and analysis, we used fMRI prep, which is an automated processing pipeline for fMRI. And then we did multivariate analysis where we used linear support vector machines to classify the data either into mothers versus women who don't have children, or into this was when someone saw an adult stimuli versus this was when someone saw a child stimuli. And we used leave one pair out cross-validation, so either one pair of subjects or one pair of stimuli was left out in the training process, and then we tested whether this is significantly above chance. And we also did a univariate analysis with a full factorial model in SBM12 with the factors motherhood and protagonist, and these were based on first level contrasts that already subtracted the neutral pictures from the painful pictures. And this we also used for region of interest analysis, where we extracted the activation in those regions and then did a Bayesian ANOVA in JASP to test for possible differences. The multivariate analysis showed that it is possible to predict the motherhood of a never before seen participant above chance with 55.6%. This was significantly above chance. And then we also saw that it's possible to predict the protagonist of a never before seen stimulus above chance with 63.9%. This was highly significant above chance as well. And this is the analysis of the region of interest. The analysis, the univariate results. We had 4 regions based on a recent meta-analysis. And in 2 of those regions, we found a difference between the mothers and the women who don't have children, with mothers exhibiting higher activation in response to the empathy for pain. And then in one region, we actually had the effect that adults led to a higher activation than children, which was quite unexpected. So this is kind of the conclusion. We did find that mothers had a higher empathy reaction. We obviously, based on this, we don't know whether it's simply because more empathic women tend to have children compared to less empathic women. And we also don't know whether it's due to actually the parenting leading to this higher empathy reaction. Although we were able to rule out relationship status and age and socioeconomic status and stuff like this. And with the protagonist, it was a little bit less clear-cut because we actually expected children to lead to a higher activation, but that's not what we found. We found in one region that adults led to a higher activity. So it might be due to the similarity between the people in the stimuli and the people in the scanner. And we didn't find any interaction with motherhood. So basically this effect that mothers had a higher empathy reaction was completely independent on whether they were watching a child being hurt or an adult being hurt. Thank you.
Thank you very much, Irene, for your talk. And let's see if we have questions from the attendees. Okay, we have one. So Sevgis asking, what will be the follow-up of this study?
Yeah, so we have actually one follow-up that we're doing right now where we're looking at other more cognitive social processes. So if it's not about an emotional reaction to children, what if it's about figuring out what a children is thinking right now, for example, so theory of minds that we're looking at. And also a follow-up would be to try to get to this difference before people actually have children. So maybe look at people when they're young, ask them, do you want to have children in your life? Is there already a difference in empathy reaction there before people have children, to kind of get at this causality effect?
Okay, thank you. And another question from Katarina. How do you explain that there were no differences between mom and non-mothers in the behavioral results, but significant classification results of fMRI data?
So our behavioral question in the scanner actually didn't ask about empathy. It asked about how painful does this look. So that's not really getting at the same reaction that we're actually measuring with the fMRI. So with the fMRI, we are measuring the brain reaction of the painful pictures compared to the neutral pictures in regions that are associated even with self-pain, but also watching others' pain. And our ratings really are just about, does this picture look more painful? And with those, we actually hope that we wouldn't get any differences, because otherwise we could argue this has nothing to do with the factor adults versus children or the mothers versus non-mothers. It's just that this looks more painful for some people than for others. So it's in the stimulus material and not in the actual reaction. So we were hoping for that.
Thanks. We have another question from Mert. So, and this is something that I also wanted to ask you. So, was there any control on the gender, sex of the children participated in the study? Do you think that there will be any difference?
So the stimuli were 50% male, 50% female. So both in the adult and in the children's stimuli. We did not control though for whether the mothers just have daughters or just have sons, simply because it was too hard to already match it as it was. So we didn't do that. Yeah.
Okay, and a follow-up question of it could be from my side. Have you also looked at the if the let's say time after having the child is influencing it? So kind of if there are some relationship with the age of their own children, of the mothers participating into the study?
Yeah, so we had the mothers had to have at least one child in the age of our protagonist of the stimuli. So like I think it was between 4 and 8. So they needed to have at least one child in this age range to assure that they actually have this kind of like experience with children that age. But then when we also tried to look at like, is there a difference between mothers with a lot of children versus mothers with one child in this age range and stuff like this? And for this, we didn't find anything, but it might also not be enough variation for this, because we obviously had the typical case of 2 children quite often. Not really a lot of people had more children than that or less children than that. Yeah.
Okay, perfect. Thank you for answering it.
Thanks.
And if we don't have any more questions from the audience, I think we should all. Okay, we have one, and from Daniel. Would you expect the same results for fathers? That's an interesting question.
That's a very good question. I like that one. If we're talking about primary care fathers who've spent as much time with their children, then yes, I would expect the same result. We did not include it because it's just really hard to find enough primary care fathers that spend that much time with the children. But if that was given, then yes, I would expect the same. And there is also a paper that looks at primary care fathers, primary care mothers, and secondary care fathers. And the primary care fathers do have a lot of overlapping changes in the parental brain compared to the primary care mothers there.
That's very interesting. Okay, thanks also for taking this last question. So I think we can wrap up our session. I thank you, Irene, Sophia, and Natasha for their wonderful talks and for showing us their work. I encourage everyone who wants to know more about their research to reach out to them on Twitter or by email and so on. So thank you for all the questions that the audience has asked. Very, very interesting questions, and thank you for having appreciated this this session. So I would just tell you to enjoy the conference, and also to post updates on Twitter. Use the hashtag NMC3. And thank you also to my backhand in this room, Jelana, for providing all the assistance, the technical assistance with the chat and so on. And again, enjoy the conference and see you soon. Bye all. Thanks. Introduce you.
By the way, we should also be careful about timing because the way the webinars are set up is that by like 15 minutes before the end of the hour, the next webinar starts, and then they will kick us out automatically because they will use the same account. When the next webinar starts, our webinar will terminate automatically.
So the speaker has 30 minutes slot, no?
We should quit by the 50 minutes before that.
Hi, Simon.
Hello. Hi, Simon. Hello.
Hi. How are you? Yes, very good. Thank you. Thank you for for accepting the invitation and everything. Thank you.
Yeah. Well, thanks for having me. This is really, it's kind of my first time with this neuromatch, but it seems very exciting.
Yeah, it's also my first time. So you were saying that he was telling me about the timing, that we have to be really...
Jonathan, do you hear the voices?
You know, they use the same account,
yes.
and then we are out from the...
Do you know what's going on?
I see.
So
I'm
you have
sorry.
30 minutes for talking and...
Oh.
Is that not our room or?
Oh, okay, now they are out. So maybe someone who started. Okay.
Oh, okay. Sorry. So it just pulled up when I did live streaming. So.
Okay. So maybe that was the recording of another of previous days.
I'll just. Should I just start the webinar?
Yeah.
Okay.
So we can have the attendees to join us, because that takes a couple of minutes usually. And then we should have a first speaker, but I'm not seeing him, and also he didn't reply to my mails these days. So I assume that he will just skip. But fortunately, it's the last talk. So no, he's no that. So Jonathan, if you see Daniel Mitchen
Okay.
in the attendees, maybe he couldn't join as a panelist. So if you can just promote it, if you just watch the attendee list. Thank you very much.
Okay.
Okay, so I think we can start. We are already a bit late, but since we don't have our last panelist, that could work. So hello everyone, welcome to this session of NeuroMatch 3.0. I'm Alberto Antonietti from Blue Brain Project in Geneva, Switzerland. And together with me, we have Jonathan as a backend host. So he will help me in carrying out this session. So welcome you everybody again. So today we will have for now two talks, and they will be about some tools for neuroscience education, or which are the challenges and opportunities for this virtual situation that we are currently living in. And I will start with our first speaker. She is Eden, and she is a research consultant and technician at the Rice University. And she will present the development of interactive visualization for neuroscience education. And between other things, she also loves coding, cooking, music, photography, learning foreign languages, and she also raised a hamster at home. So that's her presentation, and I am glad that I just look forward to hearing the talk. So please, Eden, the floor is yours.
Thank you, Alberto. And hello, everyone. I'm Eden. Today, I'm going to present to you about the development of interactive visualization for neuroscience education. First, why do we want to make neuroscience interactive visualizations? Because in neuroscience teaching, many students are visual learners, but the concepts are very abstract, especially those high dimensionality ones. Those are the main challenges. So we get some inspirations from the NeuroMatch Academy. There are many interactive widgets in Python that students can play with and stay on top of their learning. And also these visualizations make the concepts very tangible. So we aim to develop a repository of useful visualization widgets in teaching. The structure of our code is, first we develop the widgets on Godot and Unity. There are two game engines. Then we release our products to HTML forms. In the meantime, we export to computer standalone for normal tests and to APK for on-screen mobile tests. Then we release them to GitHub open source. So we welcome contributions and collaborations from the community. And we are happy to have more people contribute new things. And also, we use Jupyter Notebook to hold widgets with added text and background. Teachers can embed and those widgets they want to use into their own Jupyter notebooks and add the web links to their PowerPoint to directly interact with them. At last, we use GitHub webpage to display all of our products. And next, let me show you some of our developments. The first is a head neuron in mice that change activity with head rotation. And we can adjust the camera and view it from different angles, and this is the head rotation. And the cursor on it will show the corresponding activity of the neurons. And next one, 1D Gaussian. This is mainly emphasizing the on-screen gesture development. Again, use the sliders to change the two main parameters of the Gaussian. Or we can use a joystick, like those in games. Or we can just put our finger to where the peak is. And the third one is very intuitive because it goes to where the finger is and the parameters change simultaneously with the position. To the 2D Gaussian, we have two gestures. One is the rotation. We have two fingers on a mobile screen or any tablet that rotate in this way, and that we can rotate the camera and see the mesh from different angles. The other one is a two-finger drag. If we put two fingers on the screen, the code will recover an oval. The oval uses the point further away to the origin as the main axis and cross the second point. When we have the equation of this oval, we can recover the covariance of the two-dimensional Gaussian by a mathematical equation. And also, this oval represents the first standard deviation of the Gaussian contour when it's projecting to the XY plane. So the width of the oval in the x-axis and y-axis is the standard deviation of 2 dimensions. So let's see some effects. And first, the rotation. We rotate the camera by 2-finger slowly, and then the 2-finger drag to change the contour shape. So the Gaussian changed correspondingly. And next is a 1-finger pen. That is a drag, which can change the center of the oval. And that is the two main parameters of the Gaussian. Next product is linear dynamics. And this shows how some random points in the 2D space change along the traces with steps. And it's completely controlled by a matrix, and we can play around it. And it draws some eigenvectors with the length of the eigenvalues. We find this is very similar to the ordinary differential linear system in Neuromatch Academy. So we think that we want to compensate what Neuromatch Academy has already done, not to reinvent the wheel. So this product is no longer developed. It's a first trial of the Goulart software that we picked to develop those things. We have some other ideas. The first is Hopfield network. It's a binary neural model. All the neurons can be white or black, and we click on it to flip its color. The aim is to have the connection between any two neurons to sum together to have a maximum connectivity strength. And this is related to the associative memory in the brain. And the second idea is we want to show some neural wiring. And the first is a game of metabolic cost. Within the cost, we want people to wire a group of neurons and that you can't exceed that limit. And this might help people to infer the architecture of a neural circuit. And the second idea is to show some connectivity chains during a learning process. And third is to see how different inputs are passed down a network among different layers, etc. The third plan is to have a widget of place cells of mice. It's about some cells in the hippocampus of the mice that will be excited at certain places in the space. We want to show people a map and let the users move the mice around. When it arrives at a place with high excitation, there will be a circle showing, and the radius of the circle corresponds to the level of activity of place cells at that place. So I think it might explain why my hamster would like to stay in this trunk during most of his pastime. And last, I want to acknowledge that we have recently started collaborating with Aya Betty from Harvard University, who brought us interesting ideas of this waterfall animation. And this is completely developed by JavaScript. If we choose a distribution like a normal distribution and sample some points, and if we sample it 100 times, we'll see a waterfall effect. And it will automatically give us this distribution of the sampling. It's very cool. And we want to have a meeting with the developers, November to talk about it. And also, she brought us some this GitHub page to host all the projects, which saves us the effort to maintain some external servers. So we're happy to have people contribute to our open source codes. If you're interested in collaborating, contributing, or having exciting ideas, please let us know. And thank you for your attention and questions and discussions.
Thank you. Thank you very much, Eden, for your nice presentation. And is there any question from the attendees? If so, please write them. Otherwise, I have a small request for you. Maybe it would be helpful if you type in the chat after your talk the GitHub addresses and so on, so people could check out the code directly, and possibly also contributing to your effort. So we have one question, from Higher. Do you have any visualizations for neuroimaging?
What type of neuroimaging is that for? Experiments, some fMRI data, that kind of neuroimaging data?
Yeah, I think so. Kind of fMRI or this kind of data, yes.
And we can think about it because it might be some high dimensionality data that will be very interesting to develop.
Okay, so thank you very much again, Eden, for your talk. And I invite our next speaker to step on the floor. So now we are again in this virtual room, and Jasper will tell us about the how neuroscience societies can work in virtual spaces. And he's a PhD student in neuroscience at the University of Oxford, and is also the co-president of the Oxford University Cortex Club. And we will put all the details about it in the chat. So please, the floor is yours, Jasper.
Thank you. So as Alberto mentioned, I'm here to talk about the challenges and opportunities for neuroscience societies operating in a virtual space. And I wanted to start off by saying that I'm sure that most of us have encountered a lot of these challenges and opportunities from the side of a user, whether you attended the Federation of European Neuroscience Societies 2020 conference virtually, or if you've attended any of the SFN events, or even just your local university's neuroscience society. We've seen from the other side what kinds of challenges and what kinds of opportunities there are for operating in a virtual environment as a society. But I wanted to talk about the other side, the organizational aspects of operating a neuroscience society in the times of COVID-19 and beyond COVID-19. And so this talk is going to have two main sections. The first, I'm going to just describe what the Cortex Club is. And then the second, a bit longer section, is going to be talking about the challenges we've faced organizationally and what lessons we can learn and how these will influence what we do going forward, even after COVID-19 makes it no longer a necessity to host events virtually. And so I mentioned the Cortex Club, and Alberto said that I'm the co-president. This is Oxford's Student and Postdoc Neuroscience Society. And so what we do is mainly categorized into three main realms. First is just the communication of scientific facts and discoveries. These are the kinds of research seminars that you think of, like keynote addresses, that kind of thing. We've been fortunate enough to host speakers like Eve Marder and Mu Ming Pu in the past. And even this afternoon, we have a virtual presentation from Aaron Schumann that we're very excited about. We also host career development events, so things that provide opportunities for early career researchers, as well as more senior researchers to network with other researchers and learn career skills that will prove useful for a continuing career in academia or outside of academia. And then finally, we have events about scientific policy and culture, addressing things like open access publishing and reproducibility in neuroscience. And so the challenge was, when the COVID-19 pandemic caused all of us to have to move to a virtual space, how can we reproduce these events in a virtual setting? And so, for instance, we used to take speakers to pubs local in Oxford after their events and have an informal pub chat with a small group where we could talk a bit more about the science, but also talk more generally about issues of career progression and getting the background story about how many times they had to try this experiment before it finally worked, and just this kind of informal setting. And so, for instance, one challenge that we've had is, how do you recreate this in a virtual setting? This is an example of one of our virtual pub chats where the Oxford members of the Cortex Club set some local pubs from around Oxford as our backgrounds on Zoom. And we try to have an informal chat with the speaker to recreate this pub atmosphere. And so I want to take the Cortex Club as a case study to ask questions more generally about how the neuroscience community is going to adjust when virtual events are no longer a public health necessity. And to answer this question, I'm going to focus on a question that's only slightly different, which is, how should the neuroscience community adjust when virtual events are no longer a public health necessity? I think that using what we've learned over the past 6 or so months, we can find ways to adapt and inform our events to make them even better when they no longer have to be virtual. And so first, I'm going to talk about some of the benefits we've had from switching to a virtual format. The first is geographic accessibility. People from anywhere in the world can attend a virtual seminar, so long as it's made open to the public and the links to join in on YouTube or Zoom or whatever other platform are advertised to a geographically diverse audience. That wasn't the case before our events started being virtual. If we were hosting a speaker here in Oxford, it was generally only people from Oxford and perhaps London who could come over to view, to see the seminar or to come to the pub chat. And also on the other side, there's a greater accessibility of speakers from a geographically diverse range of areas. For instance, we're having a panel on reproducibility in neuroscience that's going forward in November. And we've been able to invite speakers like Russ Poldrack, who's from Stanford, California, where it would be quite difficult to organize this before. He's able to come and speak for us because we don't have to fly him out to the UK. And so that's a major opportunity that we've had thanks to the virtual format. Another benefit is highly related, and that's that of budgetary concerns. It's much less expensive to host speakers virtually because perhaps the only recurrent operating costs we've got right now are subscriptions to the premium Zoom service and a few other similar odds and ends related to hosting our events virtually, and perhaps some costs related to our mailing list, but that's really it. Whereas before, if we were flying a speaker in, we would have to pay for the airfare, we would have to pay for hotels, we would have to pay for any catering that occurred. And so it's much cheaper, and that allows societies which are not as well funded to host speakers in a greater number, and also from a larger number of parts of the world, just due to the elimination of a lot of these major budgetary concerns. And then lastly, a final benefit is the opportunity for diverse collaborations. The Cortex Club recently had an event with some of our sister societies in South Africa at Witwatersrand and the University of Cape Town in South Africa. And we're planning to host more events with these sister societies going forward. We had a mini symposium on cortical development, and going forward, we're hoping to hold more events of a similar nature. And that was nowhere near as easy before we moved to a virtual format. Getting speakers from South Africa up to England would have been quite challenging to organize. And while it's still not trivial to organize a virtual event, it's much easier now than it used to be. And so now I'm going to talk about some of the drawbacks, and I've put a question mark on this drawbacks to a virtual format question mark, because I'm not sure that these drawbacks are inherent to the virtual format, but there's certainly drawbacks that we have encountered. The first is domestic inequity. And what I mean by that is that when the events are being held virtually, most people attend from their own home. And that leads to some inequities based on things like domestic caring responsibilities. Regarding the panel that we said we were hosting with a speaker from California, we had invited another speaker, and we decided to hold the panel at 5 p.m. UK time so that our speaker from California wouldn't have to wake up before 8 a.m. to give this seminar at 7 in the morning or some other terribly early time. For his local California time zone. But unfortunately, because we're holding the event after 5 p.m. in the UK, one of our speakers had to decline because this person has care responsibilities in the home that require that they be unavailable after 5. And then there have been some wonderful articles published in the magazine sections of journals about the difficulties of working on a Zoom call with kids screaming in the background. And so a virtual format does highlight some of the domestic inequities when people don't have the same access when we're expanding sort of the time range of our events to accommodate this geographic diversity. The next is merely that socializing can be awkward. A virtual pub is never going to be the same situation as a real pub. And that is a drawback to a virtual format because it does in some ways stifle the kinds of networking and communication that you can do with speakers at these events. And I'm sure that most of us have felt that some of these connections, while they've been wonderful in a virtual format, we're not sure how long they're going to last or how strong the connections are compared to if we met someone at a conference or during the Q&A at a seminar. And lastly is health and wellness. We've had some attendees express regret about some of our virtual events because staring at a computer screen for a long time can cause migraines for some people. And if you're in Zoom meetings all the time, it's easy to feel that you're quite stagnant in your chair staring at a screen all day. Whereas before at a conference or at a seminar, it was an opportunity to get up and move around a bit. And so that's another potential drawback to the virtual format. And so going forward, the question is, what role will the virtual format have for us when it's no longer a necessity? And so I want to highlight the opportunity for successful integration of in-person and virtual settings for single events. And I want to talk about an event that occurred in Oxford in March. It was the International Women's Day Wikipedia Edit-a-thon. And so what this was, it was organized by Tai-Ying Lee and Christiana Vagnoni, two students at the University of Oxford. And it was a Wikipedia edit-a-thon where a bunch of groups around the world all got together to celebrate women in neuroscience and women in other specialty fields by promoting their representation on Wikipedia where they're severely underrepresented. And so our local chapter of this event was one of many across the world. And it was a hybrid of an in-person and a virtual event. There were speakers virtually who were broadcast to local workshop groups where we would all watch the seminar about women in these different fields being underrepresented on neuroscience. There were virtual sessions on how to edit Wikipedia. And then we got together, our group in Oxford and many other groups got together to sit down and actually edit Wikipedia together. And so by having both virtual presentation and in-person workshops combined in a hybrid setting, I think this is one example of a highly successful way to achieve many of the benefits of a virtual format while mitigating those drawbacks about the socializing being awkward, the health and wellness not getting up and moving, and some of the domestic inequity issues that we've encountered with our events. And so what I would advocate for going forward is that all of us strive with our local neuroscience societies to create more hybrid events where we can achieve the geographic accessibility, the budgetary benefits, and the diverse collaborations that virtual events offer, while having it be a hybrid event to mitigate the limitations of the virtual format. Thank you for listening, and here's a bit of information about the Cortex Club if you'd like to come to our talk this afternoon or if you want to attend any of our other events. Our mailing list is open to anyone from anywhere, and you can sign up at cortexclub.com, and you can follow us on social media. Thanks.
Thank you. Thank you very much, Jasper, for the wonderful talk and for the analysis of the situation of virtual or in-person meetings and the advocating for this hybrid format that could merge the benefits of both formats. And I am sure that we all can relate to your words and to what you said about the benefits and the drawbacks, possible drawbacks of both formats, both online or in-person meetings. And so, any further question from the audience or any comments? Otherwise, I really suggest you to have a look at the links that I posted in the chat, so to explore the possibilities of the Oxford University Cortex Club events that are, as Jasper said, open to everybody, not only for people living in Oxford or London, but right now to everyone.
Also,
okay.
The name is a bit of a misnomer. We're not just cortex. We have some events about the thalamus, about the basal ganglia, about tons of brain regions, not just cortex.
Okay, yeah, better to clarify that. Otherwise, non-cortex people can just say, okay, that's not my club.
Exactly.
Okay. Thank you again, Jasper. And so I've seen that Daniel reached us. So he's our last speaker of today's session. So I invite Daniel to share his screen with this presentation while I introduce him. So Daniel is a data scientist, and we can kind of go back to the first the topic of the first talk, visualization of data. So the talk of Eden, and please Daniel, enlighten us with your talk. The floor is yours.
Okay, thank you very much. So actually, the previous talk is a very nice segue into my talk, because it's also about a wiki system. And I actually participated in that Women's Day hackathon that you mentioned. So here we're not talking Wikipedia, we're talking Wikidata, which is a sister project to Wikipedia. The two are closely interconnected. And what I want to talk about specifically is how we can leverage the data that is in Wikidata to profile things that are of relevance to the neuroscience research ecosystem. And while the talk is available as a PDF on Zenodo, I also uploaded a recording here, so you can download that. And since that video is available and also on YouTube, I'm actually now, due to the previous talk, inclined to do a bit more freestyle to explore some things that I haven't included in like the static materials here. So one thing, for instance, would be that initiatives like the Cortex Club could consider expanding their range of wiki-related activities from the classical Wikipedia editing hackathon to Wikidata components. And what this means is hopefully clear after I finish my talk or during it. So first I'm going, well, I tried for this session a better poster thing. So better poster is a Twitter hashtag that tries to change the way posters are being presented. And here, this was not meant to be a poster, but I thought I'd give it a try nonetheless. And so the idea here is during a poster session, as long as we're in the paper world, you often walk by a wall of papers, and then you don't get much of those papers other than maybe the occasional logo of an institution and some bits and pieces. And here, this design of the poster is meant to enhance the main message of the poster or the presentation. And so for me, this is, well, we have a tool that is based on Wikidata, and it allows you to profile essentially the neuroscience landscape or any other research landscape. So if that's the main message that you retain from this talk, that's fine. You can switch off now and then explore it on your own. And so that was one reason why I did this and why I did it this way. Then, of course, you can zoom in, and I have already, in Zenodo, you can't really click on anything. I do have links in my presentation. So I'm now switching to the PDF version. So briefly, how does this work? So how do these profiles work? Now, actually, Zoom comes in the way with the additional little things here. So before we profile something like a research topic or a journal or a clinical trial or a pathway or a gene or a protein, we sit down and basically think about what kinds of questions would you like to ask about a protein or a gene or something like this on a standard basis. And then once we have a set of such questions, we translate them into queries that are in the Sparkle language that Wikidata understands. And then basically, once we ask those queries in a certain order and in a certain fashion, then we get back results from the query service from Wikidata, and then we render those, and that gives us the profiles. So that's about the mechanics. A little bit of background about Wikidata. It is a sister project to Wikipedia, as I already mentioned, but it has also been called the edit button to the semantic web. And it is collaboratively curated. And so if you ever wanted to contribute to the semantic web, Wikidata is the way to do it if you don't have a more formalized mechanism. It is one of the largest public knowledge graphs. It's over 90 million entries, and they are described using 11 billion triples. And still, it's far from complete. But there are about 20,000 people who are making use of the possibility that anyone can come in and improve it in a certain area. And so it's overall, it's growing quite quickly. All the data is public domain. Wikidata is multilingual, so you can feed data in, let's say, English and then pull it out in Chinese or Vietnamese. That's really amazing sometimes. It's cross-disciplinary, so there's not just neuroscience in there, there's all sorts of research topics, and then all sorts of things that you would find in Wikipedia, like popular entertainment, those kind of things. It is fair, so findable, accessible, interoperable, and reusable to both humans and machines, and it's used by major search engines and many other tools. So how are the neurosciences covered? There is a little wiki project neurosciences, but it's not that active on Wikidata yet, partly because many of the efforts that are relevant to neuroscience actually take place at a larger level. So for instance, we have a very active genetics community. All the human genes are indexed in Wikidata. All the human proteins are indexed in there. Essentially, all human cell types are indexed in there. And we're also almost complete in terms of clinical trials, PubMed index publications, or taxa of vertebrates. And from all these different angles and through integration with thousands of databases across different domains, Wikidata has become an identifier hub that actually allows you to basically convert between identifiers in different systems that don't normally talk to each other. Or if you have questions like which researchers won, like let's say, a Nobel Prize and also an Academy Award, these kind of things, that's something that a normal database can't answer. Or things like PubChem might have information about chemicals, but might miss information about what those chemicals do in a biological system. And Wikidata allows you to basically filter the data sets from one database by criteria that meet another database. Some of the visualizations that Scholia now does on the basis of this data are profiling individual people or the journals they publish and the articles they write, the organizations they work for, the topics they work on, or the clinical trials they are participating in. And now I'm going to switch back to the browser to give you a few examples. There are more examples in the PDF, but yeah, let's just do it here. Also, you can, that's the kind of identifier hopping that I was mentioning. So if you know, for instance, the Twitter handle of someone, you can just go there, Scholia/Twitter, and then that Twitter handle that will lead you to that person or organization's profile. These data are live rendered. If there's a Wikipedia article about this person, for instance, then it is also included here. By the way, I've lost feeling of time, so please interrupt me or give me a hint when I have 1 minute left. So for this author profile, we now get a list of publications, the most recent publications by this person, and we see a list of their co-authors. All of this is clickable, so here you can then get to a profile where these two people are being compared. If I click on any of those, here their individual works, then I get a profile for that particular work. If I click on this, I get the profile, since we just had the Cortex Society, I get a profile for the cerebral cortex, the journal, and gets the most recent publications in there. So all of this is clickable, explorable, and also verifiable. That's the basic idea. So in Wikipedia and in Wikidata, we put a lot of effort in making it easy to verify these things. And so one way is you can actually edit all these queries. Here they have a little link to Wikidata, which lead you to the query interface. And there you see, this is about Uta Frith. And so I can change that to anyone else, like Chris Frith, for instance. And then the query runs, and I get essentially, so here we see they have different preferences in which journals they publish in. Yeah, so this is editable, it's verifiable. If there is any of those things that I think is problematic, or there is a need to change something. So for instance here, child psychology as a topic, I can always go to the underlying Wikidata item, which then has certain statements. For instance, here it matches or maps to the mesh ID or to Encyclopedia Britannica. It also gives us Wikipedia articles on the topic. And then if there's anything to fix or to add, I can just go there, add statement, and then it will be part of that linked data graph. Going back to Uta's graph here, yeah, we can see her co-authors labeled by gender if known to Wikidata, and the topics she mostly publishes on. In order, all of this clickable again, we get some images. These are only the freely available images from Wikimedia Commons. And those images might be her collaborators or the institutions or the topics she's worked on or the places she's been to or something like this. We can map the works to individual topics, where you can get a timeline of things that she's done. So she won this prize at this point in time, for instance, and we see her academic tree. We can click on the profiles of these people. We see locations that are associated with her. Then citation statistics, and also which people are citing her, or if she went to some events like conferences. This could also pop up here, but for her there is no record yet. So this actually points me to a last point.
You have 1 minute.
Okay, yeah, there are gaps in Wikidata in various fashions, and Scholia also helps you deal with some of those gaps. So for instance, here we're looking at some things that we know are likely missing. So for instance, she has a co-author named Sarah White, and there are a number of papers in the database for which an author name string is indicated as Sarah White, and so similar for these others. And so you can then go through with a separate tool to identify those papers and see whether that's that Sarah White or another one. So yeah, I'll stop here. I encourage you to explore the tool, and I shall be happy to address any questions or comments that you might have. And I'll stop sharing.
Thank you very much, Daniel, for your presentation, and very cool tool, the one that you have presented. Well, more a framework rather than a tool. So I didn't know about it, so I'm just trying to look for my own name and to look at the different reports that it outputs. So it's really nice. So congratulations for this effort. We have one question from Carlos. He says, a wonderful tool. I agree. Is there any API we can use in order to retrieve this information using Python, for instance?
Yes, there are APIs, because it's like Wikipedia, it's a MediaWiki you can use it just like any other MediaWiki instance. Plus, it has the Sparkle endpoint. There are also dumps, and there are a number of Python wrappers for different, actually interacting with different aspects of that system. So if you want to interact with the Sparkle endpoint, there are several Python libraries. And if you want to interact with any MediaWiki API, there's the PyWikibot library that is also aware of Wikidata. And so you can read data, you can write data, and it's used widely. I will dig out some links and put them in the chat here. I know the session is closing later on, right? So yeah, I'll post it on Twitter, or maybe I'll put it on the Zenodo description later on.
Yeah, that's better. Then we have a last question from Jasper. How is it determined what topics a researcher publishes on? For instance, for Utah, I saw both autism and autism spectrum. Is it mined from the article's titles, abstract, and keywords?
That is a mixed thing, because Wikidata is a crowd-sourced platform. And so all the data in there might come in there through different channels. Some people or tools might use the titles. Some people might actually mine the abstracts. Some people might have actually read the paper. And all these different channels are used. And for each individual case, we would have to look it up. So for her, I can't fully say. I think I have contributed some of it, but yeah, this is always a collective effort, and so it's mixed. But ideally, we would harvest keywords if they're available already, like mesh terms. But this only works in certain cases, like for instance, in geography or in the humanities, there is no such system as mesh, so we can't use that. But when such data is available, we try to use it. And sometimes we do, sometimes we don't, because not everybody is aware of the tools that could be used for this, and sometimes the tools don't work. So it's complex an ecosystem. But I still think it is useful to explore the landscape, and even if it helps you visualize the gaps, sometimes that's also easy, because especially if you're new to a field, you're usually very lost. You have no idea where the gaps are. It's all fascinating. And Scholia actually helps you identify the hotspots and the gaps rather easily. And it's all open data. So there are other tools that do similar things, but here they are usually very pricey. And this one is entirely based on open data. And if you see a problem, you can go and fix it, which you can't in the other tools.
Thank you very much, Daniel, for the answers. And since we are running out of time, I would like to thank you, Eden, Jasper, and Daniel, our speakers, for showing us those great resources that they have been developing or proposing. And I also would like to thank you, Jonathan, our backend here, and all the attendees that asked questions and that participated to this NeuroMatch session. So thank you, everyone. Enjoy the conference, the rest of it, and see you soon. Bye-bye.
Thank you.
Thanks.
Join us.
She tweeted about it. I'm pretty sure she will come.
Okay, cool. Well, a possible problem is that if she doesn't join with exactly with the same email that she has provided, then she can join us in the green room, but only as an attendee, and then we have to fish her from the attendee list and to promote her. But
I see.
We have 6 more minutes.
Yeah, 6 minutes. She can make it. Okay, and also we are going live. You guys consent that, right?
Yeah, of course.
Okay.
Oh.
I've seen the previous NeuroMatch conferences were on YouTube, and people can go and
yeah, that's the
them.
thing. We are technically going live on YouTube as well. So
yeah, but is there gonna be a registration later on, or is this just now?
No, it's recorded and
will be posted.
Yes, yeah. And as far as I read, so you can already find let's say the live streaming and the registration of the live streaming, for example, of yesterday, but they are also kind of recording the session in high quality, high definition, and then they are going to kind of put the good version after. But still, there will be for sure the recording also on YouTube.
Okay.
And Zainab, do you
yes.
have received all the information of our speakers to be sent to the chat? Otherwise, I can send it to you.
If there are the ones that are in the email list that I was CC'd, yes. But if there's any extra, yeah, I'd be happy if you can send me or just, yeah.
So these are for Maria Antonietta, our first speaker.
Okay.
Then for Daniel. And then for Shinjin, I only have the Twitter handle.
Nice, thanks.
Okay, so I would suggest that we start the webinar.
Really, it's 2 minutes early. Can I?
They were suggesting to open it a little bit earlier because it takes a couple of minutes for the attendees to roll in.
Okay,
yeah,
sure.
there's a technical delay. It happens to me a lot that they started the main session, and then when it's loading for me, someone's already talking in the middle of talking.
Okay, so I'm gonna be muted and out of camera from now on, and you can reach us through chat.
Thank you very much, Zainab. I think we have a Shinjin in the audience list.
Why didn't go online?
It could be that she entered with another email. But you can promote her to be a panelist.
Okay. The webinar should have been started, right? Or we are technically online.
Yeah. Yeah, we are online. Okay, also Shinjin is, hi.
Sorry about
Welcome.
that. You're right. I'm probably like by default using a different email with this.
Okay, yeah, that happens. Okay, so it's the hour, so we can start. So sorry. Welcome everyone to this session of NeuroMatch 3.0. So I'm Alberto Antonietti from Blue Brain Project Geneva, Switzerland. And so today we will listen to 3 wonderful talks. They will be traditional talks, so 12 minutes for the presentation and 3 minutes for question and answers. If you have questions for the speakers, please use the Q&A box. And if you type your question before the end of the talk, it will be better because I can read it to our speakers. so the first speaker that we have today is Maria Antonietta Tosques. She's an assistant professor at Columbia University in New York, and she just started her lab 1 year ago, and she works on a species of amphibians that are called Spanish net. And she's a fellow Italian, but then he did her PhD at Heidelberg, and now she's in the US. So please, Maria Antonietta, the floor is yours.
Thank you, Alberto, and thank you everyone for organizing this exciting conference. It's like the future of conferences. So in this talk, I will just take you through our recent work, and mostly through my postdoctoral work as in the lab of Gilles Laurent in Frankfurt, to explain what motivates us to study reptiles and more recently amphibians. These are species that you will not normally see in a neuroscience lab, or not so frequently. And the deep motivation is that is coming from the history of neuroscience itself. Neuroscience started as a comparative discipline. If you look at the work of Cahal, for example, he's been using, is switching between different species, even between vertebrates and invertebrates, to compare and to try to infer general principles of brain organization. In this example, it's a comparison between the retina of flies and vertebrates. And later on, major discoveries in neuroscience came from exploiting the animal diversity in species, so exploiting species that are not so much studied anymore nowadays. And the work I'm going to talk about today was done on 2 reptilian species: an Australian dragon, Pogona viticeps, and an American turtle, the red-eared slider. And now in New York, my lab is starting new work on the Spanish ribbed newt, which is an amphibian. Just to remind everybody where we are in the phylogenetic tree, reptiles and birds are together sauropsids, and they split from the ancestors of mammals 300 million years ago, 320 million years ago, so a very long time ago. And the last common ancestor of amniotes and tetrapods was an animal that lived shortly before, so 350 million years ago. We're looking at deep phylogenetic splits, but nevertheless, we know that the overall organization of the brain in all vertebrates, it's conserved, because we can identify a telencephalon very confidently in all vertebrate species. And we can also identify a dorsal compartment of the telencephalon, which is also called pallium. And you see this highlighted in orange in these brain sections. And the pallium is the part of the brain that also includes the cerebral cortex. The cerebral cortex is defined as a layered structure. It exists as a layered structure only in reptiles and mammals. And in mammals, it goes from the most medial part of the pallium, which is the traditionally called archicortex, the hippocampus, to more lateral regions where you find the olfactory cortex, the piriform. And in reptiles, it retains a simpler organization. So you don't find the fully elaborated 6-layered cortex that exists in mammals, but there is a more rudimental 3-layered organization. In amphibians, the dorsal telencephalon, or pallium, is not defined as a cortex because it does not have a properly layered organization. But nevertheless, the regionalization of this part of the brain into medial, dorsal, and lateral compartments is conserved. So if we zoom in, you will see what I'm talking about when I refer to layers. So this is the traditional 6-layered mammalian neocortex. The reptilian cortex that consists indeed of 1 layer of cell bodies. These are the principal neurons or pyramidal neurons, and 2 layers that are mostly populated by axons and dendrites. and the reptilian pallium, where you don't find, essentially you don't find the layer 3, the layer 3 that you would find in a reptile. So we and others over the years, we've been asking, is there anything in common between these parts of the brain besides the common developmental origin? And with people have looked, for example, at diversity of morphologies of pyramidal neurons, and there is indeed there are indeed important novelties on the mammalian side, which I will not have time to discuss. But besides these morphological descriptions, there was really limited molecular work. So what we've done in the past 5 years is to take this problem and to focus on comparisons of cell types across species and transform this problem from an anatomical and morphological problem to a genomics problem. So we've collected single cell data sets from turtles, lizards, and more recently also from the Spanish newt. And we're setting up methods to compare these data sets across species in order to make cell type comparisons over these large evolutionary distances. And just to outline our workflow, what we're doing is to dissect out our brain regions of interest, capture single cells with microfluidics device, obtain single cell sequencing data where we have gene expression data for each individual cell that we've captured. We cluster cells to identify cell types that are operationally defined in our case as clusters in the single cell data set. And then we take these clusters and we do two things, and this is what the most time-consuming aspect of our work. We do spatial mapping, so we identify cluster-specific genes and we look at their expression in the tissue in order to connect anatomy and gene expression. And we do cross-species comparisons using data integration algorithms that take into account the entire transcriptomic data set that we're handling. And by doing this, we came to two broad conclusions that I want to share with you today. The first is that with this cell type resolution, you can identify ancient cell types, even in tissue architectures that would be otherwise very diverging. And one case is the case of the GABAergic interneurons. We found the GABAergic interneurons in reptiles, and when we integrate this data with mammalian data, we can see that the neuron types from these two species align together, indicating that broad classes of GABAergic neurons are evolutionarily conserved. Another case is the case of hippocampal cells. So in this plot, you see the mapping of lizard cells, glutamatergic cells on mouse clusters, and you see that there is significant mapping of clusters between the reptilian hippocampus, which is in rows, and the mammalian hippocampus, which is in columns. And we've confirmed using more traditional approaches that marker genes that demarcate the different hippocampal subdivisions of mammals are also expressed with the similar patterns in the reptilian hippocampus, even though the reptilian hippocampus is not folded on itself, but is stretched out. So we have identified the integers C1 and C3. And with a similar approach, we've also discovered that there are cell types in the reptilian pallium that map, because of their high transcriptomic similarity, to cells in the mammalian claustrum. And the claustrum is this brain region deep into the telencephalon, which had a very mysterious anatomy and function. And by using this single cell approach, we have identified this claustrum homologue in reptiles, and we have and this has led to further analysis of connectivity and function that you can read more into our recent publication. Okay, so this is about ancient conserved cell types. And the second broad conclusion is that the mammalian pyramidal neurons that make up the 6-layered cortex are instead evolutionary novelties. So it's not all conserved, but there is a mixture of conserved cell types and new cell types. And this is more evident when you look at this plot. It's a complex plot, but it's essentially showing you gene expression of very well characterized mammalian layer markers, which are the markers that define the cell types in the mammalian cortex. And this is on the left side. And on the right side, you see the same markers in the turtle dorsal cortex cell types. And you can see that these mammalian markers are not identifying any cell type in turtles, but they tend to be either co-expressed everywhere or not expressed at all. So we cannot, with these transcriptomics data, unlike the case of GABAergic neurons, we cannot define glutamatergic pyramidal neurons in turtles that are homologous to individual layers of the mammalian neocortex. But what we can do is still to find broad similarities at the level of upper layers and deep layers. What they're looking here is a correlation score between the different turtle and mouse cell types. And you see that there are these two correlation blocks that define upper layer-like cells and deep layer-like cells in the turtle. And the surprise was that these upper layer-like cells and deep layer-like cells are arranged into sublayers themselves within this layer 2 of the reptilian three-layered cortex. So indicating that there is a hidden layered organization in these reptilian cortices. And this disorder is actually matching the birth order of the cells because the deep layer-like cells of the turtle are the early born neurons of the turtle cortex. And they end up being in a superficial part of layer 2 because at the transition to mammals, there's been an inversion of the corticogenesis gradient as represented in this diagram. So to summarize, by studying cell types in these unusual species, we have discovered that GABAergic types, at least the broad classes, are evolutionarily conserved, and that glutamatergic neurons in the mammalian neocortex have probably evolved through a process of diversification, where the gene regulatory programs in the ancestors of mammals and reptiles have been highly elaborated in the mammalian lineage to produce new cellular phenotypes. And this work shows that with the new genomics technologies, high-resolution cell type atlases can be generated from new animals, and this opens up new avenues for further investigation. That mammalian cortical circuits are evolutionary mosaics of ancient and new cell types, and that other ancient, deeply conserved neuron types are hidden in brain areas that otherwise have very different neuroanatomy, different morphologies. And that's the case for the hippocampus and the claustrum. So what's next? My lab is exploiting the simplicity and the genetic tractability of Spanish news to focus on 2 points. One is the mechanism of neuronal diversification, and the other is looking at principles of neuronal coevolution to understand whether there is any deeply conserved circuit motif that is made up of these conserved cell types. And with this, I want to thank my postdoc lab, my current lab, and thank you for your attention.
Thank you very much, Maria Antonietta, for the wonderful talk. And yeah, for sure, reptiles and amphibians are not the primary choice for most of neuroscientists. So it's interesting to also to look at ancestors of mammalians and other species that are more used in neuroscience. So if the attendees have questions, please use the Q&A button to ask the speaker. So in the meantime, I have one question myself. So you have studied the neurons, let's say, from a genetic point of view. Have you also looked, or do you know if there are some, let's say, other shared properties that, for example, the cortical GABAergic neurons share among different species, so morphology or some electrical behavior?
That's, I mean, that's one of the questions we're interested in exploring now, because we think that we think that the molecular conservation of these cells may indicate the conservation of circuit motifs. It's of course a speculation that we need to verify experimentally, but there are some good indications and that support that idea. First is the fact that when we look deeply at what are the genes that have similar expression, you find neurotransmitter receptors and that indicated, for example, some of these neurons might have a conserved function in integrating or relaying brain state information in cortical networks. So that's one thing that we're interested in understanding further. And also another interesting point is that these cells, these GABAergic cells, are not distributed randomly in these cortices or tissues, but they have they're distributing in depth in a way that is resembling their mammalian counterparts. For example, in the turtle, we find that the neurogliaform cells, homologs, are also enriched in layer 1. So they're also in the sub-P position, very similar to the neurogliaform cells in mammals. So that's another thing that would be interesting to explore.
Thank you very much. I see that there are 3 great questions, but I think our time has run out for this first talk. So, Maria Antoinette, I thank you for your talk, and I ask you if you could please answer to the question, let's say offline. So typing the answers, you can find them in the Q&A. It will be great to have this interaction even offline.
Of
so
course.
Thank you very much.
Thank you.
So I will pass to our second speaker, which is Daniel Muller-Komorowska. He is a PhD student at the University of Bonn in Germany. And we will see, let's say, a kind of continuum from the previous talk, in the sense that we will look at some GABAergic interneurons and hippocampus. So Daniel, the floor is yours.
All right, thank you very much for this introduction. As said, my name is Daniel Muller-Komorowska, and today I'll be talking to you about our work on interneuron diversity in hippocampal CA3, and hopefully convince you that we have discovered a new interneuron subtype that co-localizes both somatostatin and VGLUT3. But first, about interneuron diversity more generally. Interneurons are, of course, a subtype of neuron that inhibits other principal cells. And these interneurons are very diverse, and they are so on 3 different categories. First of all, they can have very different electrophysiology. So they exhibit different firing behavior. They can be very regular firing, or they have very irregular firing, just as examples. They also can receive different inputs and send out different outputs. For example, here in green, we have a cell that receives both feedback drive, but also feed-forward drive. In orange, on the other hand, is a cell that receives almost only feedback excitatory drive. So these cells participate in very different circuit motifs, and they also send their outputs to another set of principal cells. Third, interneurons are also diverse regarding their molecular markers, as we just heard a lot about. And there's a lot of methods to get at the molecular markers. One of the most common ones right now is transcriptomics, which oftentimes appears to you in the wild as a color map like this. And if you take these 3 criteria to talk about interneurons, there's, of course, hundreds of different interneuron types in the brain, but one of those that's somewhat interesting in the hippocampus is the cholecystokinin positive basket cell. And this one is electrophysiologically fast spiking, so fast firing, but it also adapting. It receives mainly feed-forward drive, and its molecular markers are cholecystokinin and VGLUT3, the vesicular glutamate transporter number 3. And recently, the McBain group has a paper where they show that VGLUT3 is not only a marker for these cells, but also these cells release both glutamate and GABA onto principal cells with both excitatory and inhibitory effects. So we were interested in uncovering potentially other cell types other than the cholecystokinin positive basket cell that also co-localize VGLUT3. And we found the a subset of somatostatin cells co-localizes VGLUT3. This work we've been doing together with Eric at the laboratory of Professor Sandra Bless. And here you see the green arrows mark two cells that have somatostatin antibody signal, somatostatin mRNA signal, and this is visualized with the RNA scope technique. And they also have VGLUT3 mRNA. And if we quantify this per layer, we see that this is predominantly, these cells are almost exclusive to CA3. So in CA3, there is about 30% of somatostatin positive cells co-localize VGLUT3 mRNA. So we wanted to characterize these cells, and the only way we saw to characterize them was to use a technique called PatchSeq, which gathers both electrophysiological and transcriptomics data from the same single cell. I will first talk about the electrophysiology of that. So because we're interested in somatostatin and VGLUT3, we used mice that express Cre under the somatostatin or VGLUT3 promoter, and we use viral injections to get EYFP into those cells so we can visualize it. And then here's an example from an electrophysiology recording of an SST positive cell and a VGLUT3 positive cell. We can patch those cells in the 3 layers of the hippocampus, stratum radiatum or stratum oriens. But as a control, I also patched a lot of cells in the pyramidal cell layer, and I call those putative PCs, pyramidal cells. And from each of those recordings, we can quantify 28 different electrophysiological properties. So first of all, then, of course, it's interesting to do dimensionality reduction on those 28 electrophysiological parameters. And the first finding was that this was anticipated, but there is a clear cluster that contains all the cells from the pyramidal cell layer. So these are the putative pyramidal cells. So it was good that I patched those, because this tells me that there's also some that were in the stratum oriens and stratum radiatum that have pyramidal-like properties. So we throw those out because we want to get more into the interneurons, recluster them, and the electrophysiology alone does not form very well-defined clusters of these interneurons. So then we need to move on to the transcriptomics. So these cells here, these are 155 cells that I patched, and out of those we could get cytosol of sufficient quality, mRNA quality from 97 cells, and these are shown here. And first finding was, do we also find the somatostatin and VGLUT3 co-localization in the transcriptomics data? And the answer was yes. Out of these 97 cells, 12 are both somatostatin, and SLC17A8 is the gene name for VGLUT3. So these cells are both somatostatin and VGLUT3 positive. And then we looked at some known interneuron markers to see if any of those characterizes these co-localizing cells. And interestingly, found that tyrosine hydroxylase, it was the one that significantly can classify these co-localizing cells. And by the way, this transcriptomics work we're doing with the precise core facility that exists here in Bonn, which have done great work with our samples. So tyrosine hydroxylase is a putative marker for these co-localizing cells. But now that we have the transcriptomics data, we can look again at the dimensionality reduction and annotate the co-localizing cells. And unfortunately, neither the transcriptomics nor the electrophysiology embedding the co-localizing cells form well-defined clusters. And we've not only done the dimensionality reduction, but we've also, of course, done more traditional statistical tests to see if any electrophysiological parameter defines these co-localizing cells, and we found none of our 28 parameters does so. So something that I'm working on right now, and that's more directed towards my scientific future, hopefully, is that here you see an example of one electrophysiological parameter, which is called maximum frequency. In the middle, you have the co-localizing cells. See no statistical difference between these groups, but even within the groups, there's a pretty wide range. So you can have around 100 hertz between of cells within a group. So I'm interested in modeling this. So I've started to use the Brian 2 model fitting toolbox And here you see these are recordings from one cell, and sweep 1 is a very low current injection that in blue you see the data, in orange the model. And this model that I fitted captures that there's only 3 spikes here, and they are all only in the beginning. And then at sweep 3 at much higher strength, we get a more sustained high firing frequency. And then I want to later use these kind of models to get a biologically plausible model of CA3 that incorporates the diversity of different interneurons. So in summary, a subset of hippocampal CA3 interneurons co-expresses somatostatin and VGAT3. PET-seq allows for the characterization of such rare molecular subtypes of interneurons. Somatostatin and VGAT3 co-localizing cells are not electrophysiologically unique. And tyrosine hydroxylase is a putative marker of these co-localizing cells in the hippocampus. With that, I want to thank you for your attention. Thank my supervisor, Heinz, Sandra and Eric for the great work on the RNA scope, Christian and Heidi from the core facility that have done great work with our RNA samples, my wife, who always helps me improve these presentations, and my presentation, my figures in general, and also the NeuroMatch organizers and the community. I'm somewhat of a NeuroMatch veteran right now. I was at the first one, the second one, 98 at NeuroMatch Academy, and it's pretty great to kind of grow with the community here. And if you want to follow up on the interneuron grind, my Twitter and my email, and yeah, contact me.
Thank you very much, Daniel, for the wonderful talk, and also for your constant presence at NeuroMatch. So one of the early adopters of the NeuroMatch idea, and that's great to see. So if you have questions for Daniel, please write them in the Q&A. And we have maybe one from Elena. Do you think it is possible that instead of there being separate clusters for which we have not found the parameters to define them, there is a continuum that might be more dynamic than we realize? For example, cells changing class, for example.
Yeah, so I'm personally, I subscribe to the continuum theory of interneuron diversity. There is some work in the dentate gyrus from the Bartos lab that claims that they show, they provide evidence against the continuum hypothesis. So they use electrophysiology, molecular markers, and morphology. And using all 3 of those, they're able to get clustering. But in the end, you can always get clustering, but the question is, how meaningful is this clustering? So it's hard to say. I'm not perfectly convinced that their clusters are the end all of interneuron diversity in the dentate gyrus. And I think even if there are clusters, they can still be they can exist. Clusters and continui can exist, I think. And I'm personally, because I had so many clustering failures, as you can see, I'm more on the side right now for CA3 that it's a continuum. Yeah, that's
okay.
I think that's a good question, and right.
And we have a last question. So from Jason, SST positive neuron in hippocampus tend to target dendrites. Do these SST VGLUT3 neurons show similar targeting?
I would love to know that. And we didn't really have a method yet to specifically get an opsin only into the co-localizing cells. We can get fluorophores into somatostatin cells. We can get them into VGAT3 cells, but getting fluorophores only into these co-localizing cells, we are trying now a bidirectional genetics with SST flip recombinase and VGAT3 Cre recombinase, and hopefully this will tell us, but we don't know yet. But this is definitely something that, as I said, our input-output is a major criterion for interneuron diversity. So this is next on the table to kind of find that out.
Okay, and we have a very last question from Jan. Cool talk, I agree. What might be a functional role specific to those SST VGLUT3 co-expressing cells?
I don't know yet. and it's hard to speculate. We'll be doing some knockouts of the VGLUT3 in these SST cells and do some behavior to see maybe if it does something to memory. But what specific? I mean, there are cells that are CA3 specific. So probably something sequence related or ensemble related or attractor related. But yeah, beyond that, I'm not really sure. Generally though, if maybe they also co-release glutamate, that would be cool. But the functional role or the role of glutamate co-release is not actually known computationally. So if this would be known though, that would be really cool. But I don't know yet.
Thank you very much, Daniel, for your answers and all the attendees for their interesting questions.
Thank you.
Let's move on to our last talk. Xinjin is our last speaker for this session. She's a junior fellow at the Harvard Society of Fellows at Harvard University and Broad Institute. So we stay in the gene area, in the gene topic, and please, Xin, the floor is yours. And you are muted.
Is this better?
Yeah, now we hear you.
Great. Good morning, good afternoon, everyone. And I want to thank the organizers for putting this very special conference together. And especially thank you all for the opportunity to allow us to share our work here during this very special time. Thank you, Alberto, and all the panel speakers. It's an honor to be here with you. This project I will talk about is done in collaboration with Feng and Paola's lab and strongly closely working with Aviv Regev. In general, we're interested in developing a systematic approach to study gene function inside an intact developing tissue. And this is really driven by the sort of the interest thinking about diseases and disorders. Because now we've accumulated long lists of risk genes implicated in different disease and disorders. And the next challenge is really to define the function of these variants. And more specifically, we want to define the type of tissues and cell types listed here that these variants act on. And we also want to know how these variants can actually affect different cell states and gene programs in the cell intrinsic manner at the start. But there are really long lists of genes, and this kind of genes are likely to be growing when we deepen our understanding of diseases through the human genetic studies. And there are also long lists of potential tissue type, cell type, and molecular pathways that any of these variants can act on. This just create a huge search space for us. We develop in vivo perturb-seq to address some of these issues. And the tissue I care about is the brain, which has amazingly diverse cell type that you heard from the two speakers today. And these, when during development, these diverse cell type need to be made in the right time and space. And if any of these program goes awry, this will lead to neurological disorders and diseases. So for autism, one of the relatively well-known and well-studied neurological disorders, in the past 2 decades, we've actually known a lot about genes that's strongly implicated in autism. So for example, one of the syndromic cases of autism called Rett syndrome is caused by the mutation of MECP2, which is a transcription repressor that's required both in development and in adult stage. But another set of gene, for example, SYNGAP, is a gap protein that operates at the synaptic junctions of excitatory neurons. And they are part of a larger glutamate receptor complexes to regulate the receptor trafficking and neurotransmissions. And I'll give you one last example, which is Fragile X, caused by the mutation in a gene called FMR1. And in this case, FMR1 actually regulates very different classes of biological processes, such as protein translation, among many other things. So we could go on and on with these single genes that are strongly implicated in autism and other disorders. And these are mainly de novo loss of function variants, implicating that they have large effect sizes and are very penetrant. And these are very important entry point for us to understand autism. And in the past almost 2 decades, these single gene studies have set the foundation for the field, allow us to think about the problem by building important animal models, allow the physiology of molecular studies, and even build behavior models in different kinds of organisms. But we should also realize that this gene list is growing. And therefore, in the recent cohort studies, it is shown that there are at least hundreds of such de novo variants across the genome. So in this plot, you will see that on the y-axis, resembling the statistical significance of these risk genes, and on the x-axis, these risk genes across every single chromosome in the human genome. About a third of them encode transcription factor and chromatin modifier, some are structural proteins or synaptic scaffold proteins. So the nature of these genes and proteins are just very different. They might be operate at very different cell types and biological processes. Just based on sort of the nomenclature and the nature of the gene and gene product, we really do not know in priori what could be the common mechanisms. So our question here is, are these hundreds of risk genes actually are hundreds of different problems and affecting hundreds of different things? Or are they actually converge and diverge into different subtypes? And what I meant here is that is it possible that some subsets of these variants and mutation actually affect common cell types? And in these common cell types, are they actually affect similar biological pathways that can be grouped together for mechanistic studies? And to solve this question, we really need a genetic approach to study loss of function of these genes systematically. And we want to establish such a screen because making individual knockout animals and cell lines and study individual knockout animal cell line can be very time consuming and costly. We need a scalability here. So in general, in our design, we focus on development and we want to do this in vivo in a developing brain. We could inject the genetic modification. In this case, it's a guide RNA library through virus. And we do this very early in utero during the embryogenesis and the corticogenesis in the early stage. And postnatally, we can harvest and enrich for these cells after perturbation. And we want to use cell type specific readout or single cell readout to understand what kind of cell type are most vulnerable across different cohort of risk genes. We want to identify the cell type changes, cell state changes that's related to the loss of function of the risk gene during such a dynamic neurodevelopment. So standing on the shoulder of the giants who developed in vitro perturb-seq, we use similar molecular approaches to conduct this kind of experiments. So we want to inject lentivirus that carry the expression of pooled guide RNA that targeting individual 35 different autism risk gene. And we inject this very early at embryonic day 12 at the onset of corticogenesis. So you can imagine this time, the cortex is quite thin. There will be very sparsely some of the neuroepithelial and radioglial cells being labeled like this very lucky blue cells. But lentiviral will integrate into the genome and carry out the expression of not only the guide RNA, but also the blue fluorescent protein that label the entire lineage during development. At postnatal day 7, you can imagine the brain will be a mosaic and each of the clone will carry the blue fluorescent protein expression as well as the loss of function for the given gene. So we can enrich for these perturbed cells in the postnatal stage and use single cell RNA sequencing to obtain their transcriptomic profile. And this will allow us to computationally later on deconvolute what cell types are they, what transcriptomic profile are each of the cells, and as well as the perturbation identity of these cells. So altogether, we want to ask what kind of cell types are most vulnerable for different autism risk gene perturbation and what are the molecular pathways. And this approach is very efficient because we are asking the effect of many different risk genes at the same time. And precisely because of that, it's less noisy or less likely to be biased by other confounds. So in this approach, we can achieve reasonably high loss of function for this in vivo CRISPR, but we're analyzing a mixture of homozygous and heterozygous knockouts. We are using a very mosaic approach, and therefore it allows us to focus on the cell autonomous effects, and the time window is focused on the mouse development. But in the future, this approach can be applied to other systems, and hopefully one day in various reptiles as well. In total, we analyzed about 40,000 single cell data from this data set. And from there, we can actually analyze major cell type that's being targeted as expected in this stage of development. And we also observe that not only in the cortex, but also in hippocampus and other brain regions, we can also target using the in vivo perturb-seq approach and allow it to study development in different brain regions in the future. So we can analyze these 5 major cell types, but any of these cell types can be further divided into other subcell types, such as the excitatory neuron can be divided by the subtype coordinate to their laminal position. And using the machine learning-based topic modeling, we can extract gene program from these single cell data. I wouldn't go into the detail of the gene programs, but some of these gene programs represent subtypes of, in this case, excitatory neurons. But some of these gene programs actually share across different cell types. For example, the neurite development is a feature that's conserved in all the different layers of excitatory neurons, and we will be able to extract such feature from the single cell data as well. So ultimately, we use these gene programs shown here by row, and we can analyze each of the perturbations shown here by column and whether they affect the gene expression using the linear model. So you can appreciate that even in this reasonably small pool of 35 autism risk gene perturbation, we can already observe certain level of convergent phenotype, and namely different knockout can affect the same module and the same cell types. In total, we observed the changes in 4 different cell types and across 5 modules, and some of them are specific to layer 4, probably correspond to the timing that the thalamic input is being widened to layer 4 in this very interesting timing. And some of them are related to a subtype of cell type, such as the interneuron and neurotrophic factor, such as NDNF1 positive interneuron subtypes. So it's interesting that we see some of these modules and cell type recurrently occur in different perturbations, and both neurons and glia are directly affected by different perturbations. We validated many of these results through orthogonal experiments, including the role of how anchoring protein ANC2 in an interneuron subtype. And this is a role that, in addition to its known role in regulating the calcium signaling and axon morphology in excitatory neurons, that's known in the literature. So in the next 1 slide or 2, I will tell you a validation in a germline mutant animal about a glia phenotype. So oligodendrocytes is not very highly represented in this data set because of the early stage of the harvest. We can see that majority of the cell types in our data are the progenitor cell type labeled here in purple, and the ODC1 module we extracted were highly expressed in this progenitor cell type. We discovered that the perturbation of the 2 genes, one of them the CHDA, actually lead to the downregulation of this module in oligodendrocyte. At the same time, when we see these results, we want to really validate this data. So we took the heterozygous loss of function animal model of CHDA and use the gene module that we extracted to perform in situ hybridization validation, where we do observe a similar decrease of expression for the progenitor marker PDGFR and CSPG4. So we validated these results in CHDA, but what does that really tell us? I think this is an interesting time to think about not only cell intrinsic factor, but also the cell extrinsic factors, such as the perturbation of autism risk gene can directly affect glia cell type, as well as how the glia cell type can exert cell non-autonomous phenotype to modulate nearby excitatory neurons, as well as their final refined connectivities during development. So here, just to summarize, I told you how our attempt to build a technology to study gene function at scale. And using this approach, we studied the function of a panel of autism risk genes in mouse development And we discovered that both neuron and glia can be directly affected by panels of perturbations. And specifically, we identified really a very rich data set that we start to appreciate and hopefully in the future learn more about it. We did multiple different lines of investigation and validation, and specifically, we identify how oligodendrocytes and specific gene targets that may lead to the related autism risk gene perturbation effect. And briefly in the future, we also
Sorry, can I ask you to conclude very briefly since we are kind of out of time?
Right. This is the last slide. So in the future, we're hoping to work on a complementary technology that bring in the spatial information. And namely, we want to perform perturbation, but at the same time using spatial transcriptomic to read out not only the morphology of the cell, the anatomy position, but also their neighborhood and how intrinsic and extrinsic mechanism is involved in the perturbation. And I want to thank my mentors, my collaborators, my funding, and most importantly, your attention of being here with us. Thank you.
Thank you. Thank you very much, Shin, for wonderful talk. Very, very interesting and impressive, the amount of data that you have produced and analyzed. And with this very cool technique that you are kind of applying at a large scale. So I encourage questions since we have very few minutes before we are kicked out of this meeting. And if no questions comes from the audience, I have a brief one. So are there other pathologies other than the ASD that you think this technique could be ideal to target or to investigate?
Yeah, I would say this technology, although we piloted in the brain, but broadly it's actually technology that could be applied in many other tissue type as well. As long as I think we should start focusing on genes that are likely to give an intrinsic phenotype, because we're not really analyzing the neighboring cells or where they are. So I think this is really just the first pass for us to know what kind of, let's say, developmental timing, what kind of brain region we should focus on, just because the gene list is enormous and we need to start somewhere.
Okay, well, we have other two questions, but unfortunately we are kind of run out of time. So I encourage you to reach out to Shin or also to the other speakers to continue the discussion. I'm afraid we have to leave before the other session kicks in. So I really want to thank you all the attendees and of course the speakers, Daniel, Maria Antonietta, and Shin, for their wonderful insights in their work. And thank you also, Maria Antonietta, for writing typing your answers in the Q&A. And so that's it. Thank you again for being here and enjoy the rest of the conference. Thank you very much. Bye, everyone.
I mean, you're not going to change your talk live.
No.
We are live now?
No, we are setting it to be live. So the moment that I started, it goes live. No, whatever you say now is not going to be anywhere. Yeah. Okay, so I think in like 2 minutes, I'll press the button, we go online. And because usually there is 1 or 2 minutes delay, even the moment that you go online till the moment that everything starts.
So how will we know we are online?
I think the screen shows that you're online. It's on the top right, top left, you see something is recording.
Because here it's live.
It says live. Something comes next to that and says it's recording. Okay. So, I mean, that is the first thing I can say. And you don't see this thingy on that you're in a practice session on top, can you? Do any of you see like you are in a practice session on top of your Zoom? Okay.
I can see live.
I see live.
Yeah, live is the one that's the setting, but you don't, okay. In my thing, I can see that we are in a practice session, so it's not started. But I think I can post it on chat if you don't, I mean, the moment that I press the button, it's like 1 second, so you can start like a second, wait a second and then I start talking.
Okay.
And I be off camera, and in the chat section, you can technically, it's written to the panelist, so you can technically chat to me if you need anything. That would be private. And then if you want to post anything to the audience, you can just say to all the panelists and attendees. So technically you can post anything you want. You want everyone to see that. Okay. Okay, great. So let's see, I check with Rosa. Now I say, hey, can you see that, Rosa?
I say, hey, yes.
Okay,
Hey.
so that would be like our communication. If you need anything from me, just say to me, room, whatever, room
So
2.
you're Zoom room 1.
Yeah, we are on technically, yes. So,
okay.
Okay, so it's 10 o'clock. So you're going to be on air. I'm gonna press the button.
Good morning, good afternoon, good night, everyone. It's a great pleasure to welcome the next speaker, Dr. Rustem Kazipov. I have the privilege to have Rustem as a colleague at the Institute of Mediterranean Neurobiology for many years now. Rustem is not only an INSERM research director, he's a team leader at INMED in Marseille, and he also runs a prestigious neuroscience institute in Kazan, Federal University in Russia. So he's very international. And well, before that, Rustem graduated as a MD, PhD in Kazan, and he has been a research associate or a visiting scientist in the US, in the lab of Gregory Holmes, and as well as in the lab of Yuri Pujaki. So just a few words on a more personal note. As I just said, I know Rustem for a while. In fact, since I was a master's student, he was a young recruit at INSERM in 1997, so more than 20 years ago. Well, yeah, much more than that. And he's been, I must say, an amazing source of inspiration throughout the years. I can say that Rustem is maybe one of the most creative neuroscientists I know, an exceptional experimentalist who has pioneered many techniques, including patch clamp recordings in pups in vivo. And he made really seminal contributions to the understanding of the mechanisms and of the role of early network activities in the developing cortex. So I would say that Rustem is really a pioneer in the field of systems developmental neuroscience. So today, Rustem will talk about his latest work, characterizing early sharp waves in the neonatal hippocampus. And so I will give you the floor, Rustem. Thank you.
Thank you. Thank you very much, Rosa, for this warm world. I'm happy. And I'm happy to be here. And so Rosa has made already introduction of myself. My talk will be divided in 3 parts. First of all, I will describe those patterns of activity which are expressed in the hippocampus of neonatal rats in vitro, in slices, and in the intact limbic structures preparation. Then I will move to the short description of the activity patterns that we see in the neonatal hippocampus in vivo. And in the third part of my talk, I will present recent non-published data on the activity patterns which are evoked by sensory stimulation in the neonatal hippocampus. So most of we have learned through the past years about how activity is organized in hippocampus comes from studies using in vitro preparations of slices. In the pioneering work by Enrico Cherubini, Jean-Luc Gaillarza, Ezequiel Benari in late 80s. We used slices to discover this pattern of giant depolarizing potentials. Giant depolarizing potentials, do you see my mice, by the way, or not? You see, okay, good. So giant depolarizing potentials are characterized by polysynaptic network-driven discharges, which occur at a frequency of about 1 event per 5 seconds. They are characterized by large conductance mediated by GABA and glutamate ionotropic receptors. During these events, interneurons and pyramidal cells discharge action potentials from 1 to 5 on average in a pyramidal cell or an interneuron. And basically, entire network is firing during these population bursts. In addition to electrophysiological bursts, we can also see calcium increases in cells during GDPs. So here you can see an example of 3 cells which are loaded with calcium-sensitive dye and voltage-gated calcium channels which are activated during GDPs are producing synchronous calcium transients. Also, there are little spots which are activated at dendrites of pyramidal cells, which are likely reflecting conduction of calcium through NMDA channels at the early synapses, which are quite few at this developmental epoch. Importance of GTPs is evidenced by this schematic drawing of development of hippocampal network. As you can see here, immature pattern of GDPs is present between birth up to the end of the 2nd postnatal week. And this period is characterized by the most intensive and most rapid increase in the length of dendrites, branching of dendrites, axons, but also increase in the number of spines and synaptogenesis. So this immature pattern, many people think, is linked to the developmental plasticity in this developing network. When these GDPs disappear at the end of the 2nd postnatal week, they are replaced by adult patterns which are supporting integrative functions which are in which hippocampus is engaged. GDPs have been extensively analyzed through recent years, and here I would like just to show you example of one of such analyses in which we have addressed conductances and ionic currents which are contributing to generation of giant depolarizing potentials. So this is based on recordings of pyramidal cells in CA3 using Gramicidin patch clamp recordings and simultaneous extracellular field potential recordings. Here on this plot, on the bottom, you can see a discharge of pyramidal cells in CA3. Time 0 is the peak of maximal firing of pyramidal cells. And here you can see on the plots above changes in membrane potential during GDPs. So cells slightly depolarize from minus 75 millivolts to about 55 millivolts. During this events, we can see activation of GABA and glutamate conductances. GABAergic conductance is much, much higher than glutamatergic conductance. But driving force for GABA is relatively small. From resting driving force of plus 15 millivolts, it drops to almost 0 and even change polarity at the peak of response of GDP. Whereas glutamatergic conductance maintains high driving force. When we calculate in keeping with change in driving force and in keeping in conductance change for glutamate and GABA, we can see that at the onset of GDP, GABAergic and glutamatergic currents equally contribute to depolarization of pyramidal cells. Whereas at the peak of GDP, glutamatergic conductance remains excitatory. However, GABAergic conductance transiently switches to hyperpolarizing and inhibitory to stop population burst. Otherwise, it will raise and develop to epileptic discharge. In vitro preparations also have enormously contributed to understanding how GDPs are propagating within limbic system. For instance, here is a photo of limbic system in vitro developed by Ilgam Khalilov. So here are shown some trajectories of generation and propagation of GTPs along septotemporal axis. Also, GTPs are highly synchronized in left and right hippocampus through ventral commissural connections. and they also propagate to septum. Within hippocampal network, they mainly originate in CA3 region, from where they propagate through Schaffer collaterals to CA1, and through GABAergic connections, they also activate dendrogyrus granular neurons. It should be said that in addition to hippocampus, very similar patterns of intermittent bursts, very similar to GDPs, are also present in many other structures in vitro, for instance, in retina or in spinal cord. In spinal cord, they are generated in the ventral parts of the spinal cord. They are present in the isolated spinal cord. And notably, they are also very similar to what is seen at the level of behavior of the neonatal rats. So here you will see a short video describing behavior of a 5-day-old rat. And you can see that this animal is producing limbs and whole body startles and local twitches. And these are very likely generated at the level of spinal cord by these intermittent bursts of activity. And I will show you later some importance of these events for the generation of hippocampal activity in vivo. So from this data, which we have gained from in vitro recordings, we came to conclusion that internal network of hippocampus is sufficient to produce self-generated activities in the form of GDPs, as well as many other networks in our brain do during these very early developmental stages. However, question is, does this type of activity also express in vivo? And we were very lucky at this moment because Yuri Buzhaki has joined our team during his sabbatical in late 90s. And during fantastic 6 months that we have spent together with him and with Xavier Lincugel in the lab, we have developed a system to record from the head-restrained rat pups in vivo. And here you can see example of one of such recordings where we recorded with local field potentials events, which we called early sharp waves, which are associated with activation of polysynaptic GABAergic iPSCs, synaptic currents at the level of pyramidal cells of CA1. And the same sharp waves are also associated with large glutamatergic currents at the top of sharp wave. And this is followed by after discharge, which we call tail at that moment, also composed by barrages of synaptic glutamatergic EPSCs and GABAergic currents. So at this moment, we could see that there is a lot of similarity in electrophysiological appearance of GDPs in vitro and this in vivo sharp waves. However, then we started to think about the origins of these glutamatergic EPSCs. And basically in vivo, in addition to internal circuitry, which may be at the origin of these glutamatergic EPSCs, there is another source which is provided by external inputs, and most notably by entorhinal cortex, which plays a main role in conveying information from various sensory modalities to hippocampus. And in order to verify this, we have performed recordings from entorhinal cortex and hippocampus. And we have found that actually early sharp waves are reliably preceded by network bursts in the medial entorhinal cortex. And we could see that medial entorhinal cortex, its superficial layers 2 and 3, fire in advance to early sharp waves. And current source density analysis of sharp waves in hippocampus confirmed very important contribution of the external inputs coming from entorhinal cortex in generation of early sharp waves. Moreover, we also found that bursts in entorhinal cortex are in turn preceded by movements which occur in the body. These twitches and startles, which I have shown you on one of videos before. These have led us to a scheme in which activation of early sharp waves involves much more complex circuitry than GDPs that have been described in vitro. Let me remind you that GDPs are expressed in a circuit which is completely isolated from entorhinal cortex, and it is supported by a CA3 network and GABAergic interneurons and depolarizing GABA. In vivo, situation is much more complex, and activation of early sharp waves involves activation of entorhinal inputs through tempoharmonic and perforant pathways. And important roles are played by twitches and startles, which are hypothetically through sensory feedback, activate sensory cortex, and then activate entorhinal cortex. So if we write this scheme step by step, all starts in spinal cord, which generates spontaneous bursts of activity, which activate spontaneous movements. And these spontaneous movements through tactile and proprioceptors and sensory feedback activate thalamocortical circuit, which generates events which are called gamma and spindle bursts in somatosensory cortex. And further, this information goes to entorhinal cortex and ignites in hippocampus early sharp wave. If this cascade of events is correct, then you could expect that sensory stimulation could also evoke and trigger similar activation of the circuitry. And this is something new that I would like to share with you today, new data which we are actually analyzing right now. And these are based on explorations of sensory evoked responses in hippocampus using silicon probes, which enable to record activity in different layers of hippocampus. For stimulation, we use bipolar electrodes, which are positioned in whisker pad or subcutaneously at the level of hind limbs or forelimbs. In addition to electrophysiological recordings of hippocampal activity, we also place piezo detectors on the limbs of the animal to detect movements. So here you can see example response that we are getting by stimulating whisker pad in a P5 animal. Red dot line shows the stimulus of the whisker pad, and black traces show LFP, and red vertical bars show spikes recorded at these electrodes. You may see that response, which are evoking, is very robust. It attains millivolts in amplitude, and the remarkable feature of these responses are things which are located at the level of stratum radiatum, stratum lacunosum moleculare, and molecular layer of dentate. So these are projections for Schaffer collaterals to CA1 and projections for entorhinal cortex to apical dendrites of CA1 pyramidal cells and the molecular layer of granular cells. These responses are associated with increase in the frequency of multiple unit activity, both in CA1 pyramidal cell layer, also in granular dentate cells. In addition to large sharp potential deflections that you can see here on average response, we also record tails which are characterized by high frequency oscillations. They are much smaller in amplitude than sharp waves, but they are quite prominent in almost every response, and they are manifested by increasing the power of oscillations that are recorded at the tail of response in the beta frequency range from 10 to 30 hertz essentially. And another thing which I would like to pay your attention on is the fact that when we stimulate whisker pad, we record very sharp increase in piezo detectors, which are recording limb movements. So basically what happens is that when we stimulate whisker pad, we trigger startle response, reflectory startle response in the animal. And this again occurs at a very short latency, and this is followed by hippocampal response. So here, these relationships between movement and early sharp waves in hippocampus are shown in more detail. Here you can see raster plots of 80 events which were triggered by stimulus, and you can see that after stimulus, movements occur very reliably and at very short delays. Early sharp waves also occur often, but at much longer delay. On average, we have about 20 milliseconds between the stimulus and onset of movements, and about 150 milliseconds from stimulus to the sharp wave. As I said, response that we get in response to stimulus in hippocampus is composed by a sharp wave, which we quantify by filtering at low pass 10 Hertz to measure amplitude of the sharp wave. And then we also, at the tail of response, we measure frequency and power of oscillations at the frequency of 10 to 30 Hertz. By comparing spontaneous sharp waves and sensory evoked sharp waves, we can see that these events largely overlap on this circular histogram, which expresses power of beta oscillations at the tail and amplitude of sharp waves, basically. And there is a little difference in the sense that sensory-evoked sharp waves have a tendency to have larger sharp potential component, whereas spontaneous events have this cloud which is located at the segments with higher power of beta oscillations. But these differences are minor. Comparing current source density profile of spontaneous sharp waves and sensory evoked sharp waves, here you can see example of recordings from 10 animals from P5 to P7. You can clearly see that spontaneous and sensory evoked sharp waves have very similar current source density profiles. And here you can see a grand average obtained from 10 animals in CE1 and 8 including 8 with indentate gyres. And basically on current source density analysis, we see 3 sinks. Sink number 1 is located in stratum radiatum of CE1, exactly at the place where are projecting Schaffer collaterals from CA3. Sink number 2, so it's shown here on this schematic drawing by red. Sink number 2 is located in stratum lacunoso moleculare of CA1, close to the fissure, which is a projection of connections from antrinal cortex layer 3. It's here it's on this schematic drawing, it's number 2. And finally, we also see sink number 3, which is located in the molecular layer of dentate. Here it's shown in blue color as a number 3. And depth of these sinks is quite similar for spontaneous sharp waves and responses that we are getting after stimulation. Similarity between spontaneous and sensory evoked responses is also evidenced by histograms of multiple unit spikes which are activated during spontaneous and sensory evoked responses, both in CA1 and in dilated gyres. Then by analyzing the movement intensity during different types of events which we are recording in hippocampus, we could see that more amplitude have movements which are associated either with spontaneous events or with events evoked by sensory stimulus. So they have also larger sharp waves. And this is true for both spontaneous and evoked responses. In addition to that, evoked responses are also characterized by stronger beta component for the movements that have larger amplitude. Then there is kind of a difference between spontaneous and sensory evoked sharp waves in the sense that delay between the sharp wave and movement seems to be slightly shorter for the evoked ones than spontaneous ones. But and this is quite significant, but we don't have idea yet how to explain this for the moment. Now, from the data I have shown you till now, it looks like sensory stimuli are evoking sharp waves and that movements are preceding these sharp waves. So basically we have about the same sequence as during spontaneous events where we have also sharp waves triggered by movements. However, when we sort all responses evoked by sensory stimulation by the amount of movements that we are evoking, which is shown here on raster plots, from the maximal response to minimal response, we can see that a relatively small fraction of events which is evoked by stimulation is not associated with any movement, as you can see here, right? So this dashed line separates those events which are not associated with movements. However, early sharp waves in hippocampus are present, and you can see it here as an example where stimulation does not evoke any movement. However, sharp wave is very nicely present in hippocampus. So this poses a question of how this can be possible, and perhaps somehow these movements that we are evoking are obscure and we haven't detected them with our limb detectors. So we started to think about how to solve this problem, and we went to urethane, which is a general anesthetic, which completely suppresses spontaneous and sensory evoked movements. As you can see here, both spontaneous movements and sensory evoked movements are suppressed by 1.5 gram per kilo of urethane. So taking this as an advantage to suppress movements, we compared effect of urethane on movements and on sharp waves. And you can see here example responses recorded in control conditions without return. You can see here that stimulation evokes movements and sharp waves, as I told you before. Also, there is a presence of movements and sharp waves before stimulus, which are spontaneous events. However, after return injection, so this red line indicates half an hour interruption in recordings, but then once a return exerts its anesthetic action, we lose almost completely movements. However, responses in hippocampus, sharp waves in hippocampus persist, and moreover, the amplitude of these sharp waves increases by factor of approximately 2 to 4. Examples are shown here. So this is done under urethane. All movements, spontaneous and evoked, are blocked. However, we still see also at much lower frequency spontaneous sharp waves, and we see also sharp waves evoked by stimulation. And this evoked sharp waves, as I said you before, are much stronger than controlled spontaneous sharp waves. And this is about it. And to conclude, I would say that into the scheme of activation of long-range scheme of activation of hippocampus following startles, we may add this interesting pathway, which is provided by direct stimulation, which is also a natural signal acting on neonatal paps during development. By the way, we have previously shown that activation of somatosensory cortex largely depends on direct stimulation of baby rats by their litter mates and by mother. So this direct stimulus is capable of evoking sharp waves, and this involves two pathways. One is a direct pathway, which is independent on movements upstream through upstream activation of somatosensory system and activation of entorhinal cortex and sharp waves. And second pathway involves a reflectory startle and then activation of sensory feedback, which will contribute to driving about the same pathway. One could think that this direct pathway would be activating a little bit earlier response, earlier sharp waves in hippocampus, and we are working on these time differences right now. To conclude, I would like to make 3 conclusions from my talk. First of them is that it is very important to verify what we see in vitro in slices or in other preparations in vitro with in vivo. And so in the talk which I presented you today, we could clearly see that hippocampus is capable of generating activity in vitro in isolation of any inputs from external world. However, in the intact animal, very important drive comes from sensory periphery, and it is conveyed through entorhinal cortex. And as a result, early sharp waves that I have described you today are very different in their generative properties from giant depolarizing potentials in vitro. Second conclusion is that sensory input provided by sensory feedback from movements, startles, and twitches is very efficient in triggering early sharp waves. And finally, somatosensory stimuli which evoke early sharp waves in neonatal rat hippocampus act through 2 cooperating mechanisms: sensory feedback from evoked startles and direct bottom-up somatosensory circuit, which is activated by sensory stimuli. I would like to finish by thanking all contributors to this work. Maybe I shouldn't read this. I mean, you can read it yourself, right? But very important were those who contributed to the last part of my talk: Lorenzo Fabrizi from UCL, and Guzel Valeyeva and Misha Sentsov, who worked on a part related to sensory evoked responses. And I thank you very much for your attention, and I'm ready to answer to your questions.
Thank you, Rustem, for a great talk. There is no clap, but I can clap.
Thank you.
Okay, so the talk is now open for discussion. So I'm looking at the Q&R. I don't see any question yet, but I can start.
Please.
I mean, if there are no questions, I can start. So I was curious to know whether you've looked at the the developmental profile of the spontaneous sharp waves and the sensory evoked ones, whether there is whether they follow the same developmental timeline. I guess you're saying that they are probably the same phenomenon, but is it something that may differ with the age of the recorded mouse?
Right. So what I have described is true for ages maximum. We had P7, P8 rats. From some recordings that we have made at older animals, we could see sensory evoked sharp waves at ages of from up to P18. And basically we have this question for our future research, right? And what we know is that startles which are evoked by sensory stimuli in different modalities, which are presented as unexpected stimuli, they trigger startles also in adults. It's not a feature of babies. In babies, they occur at spontaneously, these startles, and much more frequently than in adults. However, in adults, startles are also present, and it is quite possible that if a circuit of startle generation is somehow associated with events which go upstream to entorhinal hippocampal system, then it's very likely that this will be also activated, but this is something to investigate in future research.
You started by saying that there was a role of these early activities in maybe promoting synaptogenesis, spinogenesis. And so how do you reconcile these two views, that you have a pattern that is there to convey this startle information to the hippocampus, and the same pattern is also promoting the development of circuits? Is it?
Yeah, I think that this is a great question. And I think that we should also think about the roles of hippocampus in general. And there is quite strong evidence that hippocampus, which we know is involved in spatial navigation, learning and memory, etc., it's also involved in such phenomena such as fear conditioning and emotions. And then it's quite possible that the development of startle-dependent hippocampal activations that we describe here are representing development of a circuit which is engaged into hippocampus development dependent fear reactions and fear conditioning and avoidance reactions.
Your recordings are done in dorsal hippocampus mainly, right? Have you recorded from ventral as well? Because one would expect ventral hippocampus to be more critically involved in such.
We haven't done dorsal recordings, no. Well, we haven't done ventral, it's mostly dorsal.
I see we have two questions in the chat. So the first one from Yuri Rodriguez, who asks, do you know why backpropagation seems to be inefficient in immature rats?
Backpropagation? Of what? Can you precise?
That's complicated too. I'm not asking, I'm just relaying the question.
So maybe it's meant backpropagation of the action potentials.
Action potential backpropagation, yes. Yuri.
Okay. Why it is less efficient? I guess that backpropagation is a phenomenon which has been described in juvenile animals mainly, and we do have some recordings of with multi-shank silicon probes from neonatal pyramidal cells. Unfortunately, I didn't prepare a picture of that. So basically, we have recorded with multi-shanks CA3 pyramidal cells in slices, and did whole cell recordings from CA3 pyramids. And then by triggering the waveform of LFP signals, we could see that actually there is a backpropagation of action potentials to dendrites, even in neonatal animals. Also, backpropagation has been described in a number of studies in Sackman's lab in juvenile animals, right? Moreover, I would say that there is overall tendency of spikes being initiated to some extent at the dendritic spots where supported by calcium spikes, these events are propagating to soma and trigger action potentials there. So I'm not sure that this is true to say that spikes do not backpropagate in immature neurons, unless Dr. Rodriguez has some other opinion and some other evidence about that.
To check. There is action potential backpropagation in young rats, but they adapt very fast when compared to adult rats. That's the answer of Yuri.
Yes, that's true. Another thing is that it's, when you simply present the polarizing step to immature neurons, then they barely fire fast action potentials and trains of action potentials in the response. They usually produce 1-2 spikes and then they get silent. That's another feature. And I guess that also action potentials are much longer from study which we published with Pierre Pascal, where he measured duration of action potentials. He could see that even action currents are much slower by like factor of 2 or 3. They are slower than action currents in adults. And another problem is that you also cannot distinguish because of that interneurons from pyramidal cells, which very nicely separate on the basis of duration. So there are factors such as expression of ionic channels like sodium channels, fast sodium channels, and potassium channels, which are involved in after hyperpolarization. So these are not the forms of these channels which are supporting fast spikes and ability of cells to fire bursts of action potentials, like sustained trains of action potentials. They are not yet expressed in immature neurons. This is true. And this is, by the way, one of factors which is supporting bursting mode of activity of immature networks as opposed to kind of tonic modes of activity which we see in adults during active states in cortex.
So we have another question that relates to the modulation of early sharp waves by noradrenaline or other monoamines. Do you know if they are modulated by?
No, we don't know that. We don't know about that. What we learned from work of Mark Bloomberg, by the way, I should say that this is a researcher who has enormously contributed to the field, Mark Bloomberg and all his group. And they have described in a little bit older animals, and we show here, starting from P7, P8, they show that during transitions from active sleep to wake, and I believe that this is the moment when endogenous noradrenaline goes up, then they see that sharp waves are depressed. And also from our recordings, we can say that we have responses which are evoked by sensory stimuli, which are getting much, much smaller during wake states. So clearly, adrenaline and other arousal systems, they should have a major impact on spontaneous activity and suppression of sharp waves. Basically, it's very similar to what we see in adults also, right? Sharp waves are present only during sleep or periods of immobility, rest, right? And once animal gets alert and goes to exploration behaviors, then sharp waves are suppressed. So perhaps there are some similarities in terms of modulation of early sharp waves.
That's what I was about to ask also. If the sensory and evoked sharp waves, you could evoke them the same way during the active sleep state or during
so they're getting the response is getting much, much smaller. Another element I would like to add is that sensory evoked responses have been also studied in hippocampus in adult animals. And this was done in 2 works by one from laboratory of Yuri Buzhaki and another from laboratory of Menendez de la Prida. So both of them have shown that basically you will never evoke sharp wave like we show here in baby rats. And responses are much, let's say, much weaker. Also, current source density profiles suggest activation of inputs from entorhinal cortex, similar to what we show here in baby rats. But if you talk about modulation of units, it's much more heterogeneous. And if you look at the multiple unit activity, kind of average metric of population activity, you see no change in CA1 pyramidal cell layer. Some units are slightly increasing their firing, others decreasing firing, and third part does not change firing. Here we see that sensory stimuli are evoking response much more reliably. And in these studies, what they also have mentioned is that during theta activity, if theta activity is present in hippocampus, then sensory evoked response is much, much weaker compared to immobility state. So this goes, again, along the same ideas, right? What we have discussed now.
We have another question from Yuri Rodriguez, who is asking whether, so I asked him to rephrase whether the CA1 frequency spectrum that you show changes with age.
Okay. We see, so first of all, I should say that these beta oscillations that we I showed you today, so they are present mainly starting from age of P4. In younger animals, these population bursts, which we have described as tails previously, and also population bursts which may occur in any without any relationship to sharp waves, by the way, in the form of increase of population firing in CA1 without any prominent LFP event. And they last from half of a second to 3 seconds approximately. So they are not, as I said, are not associated with prominent LFP oscillation until age of approximately P4. Starting from P4 to P5, you start seeing that these population bursts are modulated by LFP potential, LFP oscillations at beta frequency. Then there is a very strong increase in the power of these beta oscillations during the second postnatal week. And this has been very nicely described by work of laboratory of Dirk Isbrandt, where maybe you should see this works. It was published in Nature Medicine. And also they have another very nice work describing changes in NKC1 knockout animals. So basically, beta oscillations strongly increase in amplitude, and they become really very prominent phenomenon. And another thing is that from this, they are modulated by slower rhythms, and they increase in duration with age. And I think that these events are prototypes of future theta oscillations that will be modulating gamma oscillation. So to me, something to check, of course, but these beta oscillations that are very prominent phenomenon during the second postnatal week are very likely, or at least hypothesis to check, precursor of gamma oscillations nested by theta oscillations. At least the current source density profile shows that major sinks are generated by entorhinal cortex, similar to early sharp waves. They are driven by entorhinal cortex.
Okay, I think it's about time to leave the stage. So if there are no more burning questions, I think we can end this session. And I would like to thank you, Rustem, again for
thank you, Rosa.
You have a lot of thank yous. Thank you for a great talk and a nice discussion. And See you soon, Met.
Yes.
Bye-bye.
Bye-bye.
That your laptop will what?
Blue screen in the middle of my presentation.
Okay.
If I drop out for whatever reason, just move on to the next speaker.
Okay.
It would take probably about 2 minutes for me to get back into the session, and might not be worth it. Just fair warning. I don't think it's going to happen, but if it does, yeah.
Okay. Thank you for
don't panic. It's a technical issue on my end.
Okay.
I'm happy to try my best and butcher your slides.
Yeah, well, collectively we could give your presentation. That'll be fun. Yeah. We've done that in my lab before. You make a slide deck and you have somebody else for somebody else.
It'd be fun.
Okay, so we're live. I'm gonna get started. Hello, everyone. I am Adam Steele. I'm a postdoc at Dartmouth College in Hanover, New Hampshire, USA. I've been told that we have a few moments to let the webinar configure and people will enter. So I'm going to use that time to say for everyone in the United States, please vote, make a plan to vote. If you have a plan, call somebody and make sure that they have a plan. And if you are not able to vote in the US elections, call one of your US colleagues and make sure they vote because it's super important. So with that said, we'll get started with the science. We've got 3 really great thought-provoking talks this session about the integration between parietal and prefrontal cortices. And I'm really looking forward to hearing all 3 of these. As a reminder, we've got the chat where you can post messages and clarification questions. Just make sure that you have your messages configured to panelists and attendees, so that way everyone can see what you're saying. If you'd like to introduce yourself now, or if you'd like to introduce yourself so that way people know you're here, now is a great time to do that. Also, we have the Q&A tab for questions. You can post questions at any time and make sure to upvote your favorites. And for those of you who are on Twitter, please tweet about this session using the hashtag NMC3. So with that said, we'll get started with our first talk. We have Lina Ali Ibrahim, who is a postdoc at Harvard Medical School. You can find her on Twitter at Lina Ali Ibrahim, and she'll be giving her talk titled Developmental Dynamics of Bottom-Up and Top-Down Input Integration into L1 Interneurons in the Sensory Cortex.
Thank you, Adam, for the introduction. So I'm going to talk about what I've been doing in the Fischel lab for the past few years. Basically looking at how layer 1 interneurons in the sensory cortex integrate inputs from our sensory environment together with top-down inputs from prefrontal areas. So organisms use sensory inputs to uncover the structure of their surroundings and create a representation of their environment that allows them to perform many complex behaviors. And a lot of these behaviors and computations rely on inhibitory interneurons, which are essential building blocks of cortical computations, and they're very diverse in terms of their morphology, physiological properties, and connectivity that help gate excitatory flow in very precise manners. The interneurons in layer 1 are very understudied and will be the focus of this talk. So if we look at how sensory information flows within the brain, as we all know, light from the environment goes to the LGN, the thalamus. That input then is transferred to V1. So this DLGN to V1 signal is very important for perception. And then that information to get further transferred along the visual hierarchy to higher cortical areas, such as the anterior cingulate and the medial prefrontal cortex for behaviors and decision-making, motor planning, and stuff like that. That information then is fed back for further modulation of sensory inputs at the level of primary visual cortex. So if we schematize this, the externally driven sensory signals is what I'll be referring to as bottom-up, whereas the internally generated feedback signals are top-down, and then looking at how they integrate in layer 1 of both auditory and visual cortex. So if we look at the laminar organization of top-down inputs in the primary visual cortex, so for example, one of the structures, the retrosplenial cortex, which is involved in visual navigation, if we inject a tracer in this area and look at what the axonal organization looks like in the primary visual cortex, we can see that a lot of these axons preferentially target the very superficial layers of the primary visual cortex. And similarly, if we inject a tracer in the anterior cingulate or the premotor cortex, we see a very similar laminar distribution of the axons. And as I mentioned, a lot of these structures are involved in decision-making, attention, prediction, as well as expectation. So the question that we wanted to address was, how are the cortical top-down inputs to layer 1 interneurons established during development? So the first thing we wanted to do was figure out how to target layer 1 interneurons, and RNA-seq data from the Allen Institute figured out NDNF as one of the major markers that labels about 70% of layer 1 interneurons, and they're all inhibitory, as we can see with the co-labeling with GAD67, which is an enzyme that makes GABA. So we have a molecular handle to be able to target them. And so a Cree transgenic line was made, and we're now able to characterize these cells. I just want to mention that NDNF does not label a homogeneous population of layer 1 interneurons. There are two subgroups that are labeled by the NDNF line, ones that express NPY, and those are the classic neurogliaform cells. They have late spiking properties and are able to have both GABA A and GABA B responses. And the other group are the NDNF non-NPY positive. They have morphologies that are similar to neurogliaform cells, but they are not classic neurogliaform cells. And then two minor groups, the alpha 7 and VIP, which I will not be talking about. So the first thing we wanted to do was brain-wide monosynaptic input tracing to layer 1 neurons. So we used the NDNF Cree line. And for those of you who don't know how this works, very briefly, it's a combination of two viruses that we inject, an AAV helper virus that's going to only transfect the neurons that are Cree positive, so NDNF Cree layer 1 neurons, and then a rabies virus that's going to then replicate in these neurons and cause only monosynaptic tracing. So we inject this in the NDNF Cree line in the visual cortex and are able to map out all of the inputs that these neurons receive. So this is what it looks like. This is a labeling of layer 1 neurons, and the green label just shows that these cells carry the AAV helper virus. The middle panel shows the retrogradely labeled cells, and on the right, we see the starter cells. So these are the cells that we'll be looking at the inputs to from across the brain. So I'll be focusing on two areas, the anterior cingulate cortex as a source of top-down inputs, and the dorsal lateral geniculate nucleus as the source of bottom-up inputs to layer 1 of V1. So if we look at the upper panel here, during development, the DLGN inputs seem to be very predominant to layer 1 neurons, and the cortical top-down inputs, so from the retrosplenial and anterior cingulate, don't count for much. When we look in the adult, we see that we still see DLGN inputs to layer 1 neurons, but now a lot of these inputs are replaced by top-down inputs from the retrosplenial and the anterior cingulate cortex. So essentially, the overall dynamic seems to be a shift from bottom-up thalamic input driving layer 1 neurons during development to a more top-down cortical input circuitry that drives these neurons. So we wanted to confirm these using optogenetics. We injected chirodopsin in the DLGN, assess the fiber innervation pattern in layer 1, and as we can see, these fibers contact layer 1 neurons. We optogenetically stimulated them and recorded from both layer 1 and layer 4 in the same column. And even in the adult, we are able to elicit responses in layer 1 and layer 4. During development as well, the neurogliaform cells, more specifically compared to all the other subtypes of neurons that are located in layer 1, receive the maximum amount of inputs from the DLGN, though they're much smaller compared to layer 4. And this input persists in the adult, where the bottom-up thalamic inputs from the DLGN still innervate neurogliaform cells in layer 1. And this is a normalized response between the response from layer 1 compared to that of layer 4, and we see no difference in terms of how strong this response is between layer 1 and layer 4, between development and the adult, basically suggesting that layer 1 neurons are still being driven equally by the thalamus. So the question we then wanted to address was whether these thalamic inputs are common between layer 1 and layer 4, or whether they're distinct. And so to address that, we used a rabies flip approach, wherein the rabies virus carries a flip recombinase presynaptically. So now in the DLGN, we can go ahead and inject a flip-dependent virus and assess its innovation pattern. So to do that, we used a layer 4 Cre driver, SCNN1A Cre, and as you can see in the top panel, the green cells are the layer 4 neurons in V1 that carry the helpervirus, and then in the bottom are the retrogradely labeled neurons in the DLGN. And then we injected a flip-dependent cherodopsin virus in the DLGN. So these are presumably the presynaptic neurons to layer 4. And as expected, we can see a lot of the axons innervating layer 4, but then we also see axons that go up to layer 1, suggesting that at least some of the inputs that go from the DLGN that innervate layer 4 are also innervating layer 1 neurons. So far what we've seen is that there are common thalamic inputs between layer 1 and layer 4, and that these are more dominant during development. So what about the top-down inputs? We injected a cherodopsin virus in the cingulate, assessed its projection pattern in V1 in both development and in the adult, and then recorded from neurons in both layer 1 and layer 2-3 in V1. So based on the rabies, we actually expected that there probably won't be any inputs to layer 1 or to V1, in fact, around this developmental age between P0 and P15. However, we actually see a lot of diffuse fiber patterns at that early age, which is not as well pruned compared to what the pattern that we see in the adult. However, when we quantified the number of synapses onto layer 1 neurons at this early age compared to adults, there are far more number of synapses in the adult that these axons make onto layer 1 interneurons themselves. And then when we optogenetically activated the anterior cingulate axons and recorded from layer 1, we basically see a much smaller current being driven during development compared to the adult. That increases by about 10-folds. And sorry, I'm gonna go quicker. In fact, the responses of layer 1 neurons are also much stronger compared to those of layer 2-3. So the next question we wanted to address was whether sensory inputs are required for the strengthening of these top-down inputs, because the timing seems to be very similar between when the top-down inputs are being strengthened. So to do that, we enucleated the eye during early postnatal stages, waited till the mice were adults, injected cherodopsin, and then recorded from the neurons 2 weeks later. And what we see is that removal of sensory inputs, in fact, decrease the strength of these top-down inputs onto layer 1 neurons. We then wanted to make a more specific manipulation wherein we are disrupting only the thalamic inputs to layer 1 neurons, and then again do the same experiment. And basically, the way we did this is using the the rabies flip approach, and injected the stereotoxin to kill off the projections to layer 1 neurons. And this basically shows that all of the DLGN neurons that are presynaptic to layer 1 are killed off in this experiment. And again, we recorded from the anterior cingulate axons stimulation onto layer 1, and we can see that the strength of these inputs are reduced as well. So basically what we think is happening is that there is a predominant sensory input during development and very diffuse cortical top-down input. And perhaps layer 1 neurons are instructed by this large repertoire of sensory information, and there's a selective strengthening of the coincident signals between bottom-up and top-down inputs onto layer 1 neurons. I'm going to skip over this paradigm, but essentially we wanted to test this, and we prematurely co-activated DLGN and ACC inputs and recorded from using GQ dreads, and then we recorded the strength during development. And essentially what we see is that when we artificially co-activate both of these inputs, there is a premature strengthening of the top-down inputs to layer 1 neurons, suggesting that we need both thalamic and long-range cortical inputs to be able to strengthen these projections. So in summary, layer 1 interneurons receive direct thalamic inputs which persist in adulthood in both A1 and B1. Layer 1 inhibitory neurons in layer 4 receive common thalamic inputs, and that layer 1 neurons receive direct projections from top-down cortical areas that strengthen during development. Sensory experience is required for the refinement and strengthening of these top-down inputs, and when we co-activate thalamic and cortical inputs prematurely, there is a strengthening of top-down inputs to layer 1 interneurons. And with that, I would like to thank my PI, Gord Fischel, and the rest of the Fischel lab for all their help, as well as my funding sources and the NYU Abu Dhabi Virus Core and all our collaborators. Thank you.
So thank you so much, Lena. That was an incredible amount of work and very, very impressive. Unfortunately, we don't have time for questions, but Lena is here. If you have any questions, you can post them in the chat and she'll be able to take them there. So next up, we have Jorgi Lur, who will be giving his talk titled Cell Type Specific Nonlinearities in Frontal Sensory Integration in the Parietal Cortex. Yuri, you're muted.
Yeah.
Oh, okay.
Yeah, thank you for the introduction. I managed to unmute myself. All right, hi everyone. Thank you for tuning into this session. My name is Yuri Lur. I'm an assistant professor at UC Irvine, and I'm going to tell you a little bit about some synaptic integration in the parietal cortex. So I think if you turned up on this session, you can probably appreciate that all except maybe the most basic behaviors require sort of a delicate interaction between top-down and bottom-up processes. This feels kind of intuitive if you think about any kind of basic behavior that we as humans do nowadays, even just walking down the street or driving. Some of the environmental sensory clues, something like the change of a traffic light, would be completely meaningless without sort of the internal context that we have all built up during our lives, our memories, our previous choices, our motivations, etc., etc. So what I'm trying to dive into today is what may be some of the synaptic basis of these interactions. And so we actually happen to know a thing or two about the interaction of multiple information streams, in the large part thanks to the now classical experiments of Barry Stein and colleagues. In this body of work that has predominantly focused on the superior colliculus, they have shown that under the right circumstances, when you display two sensory stimuli, in this case a visual and an auditory, if the two stimuli are displayed coincidentally, this can increase the action potential output of multisensory neurons exponentially. A question remains, though, whether these principles of multisensory integration can be extended to non-sensory modalities. And if so, where would one look for such interactions? Now, I mean, if you listen to the previous talk, and I think in general, I think that if one looked really carefully, you would be able to find signatures of these top-down, bottom-up interactions in almost any cortical area. However, there are regions that are extremely appropriate for this type of experiments. One such area is the posterior parietal cortex that has been well recognized as a cognitive hub that receives extensive excitatory input from both sensory regions and from these higher order cognitive type regions in the brain. So this is going to be our target of investigations. And essentially, if you want to boil down our question to a very simplified cartoon, we're trying to ask if when a parietal neuron receives excitatory input from two different sources, two different cortical areas, how do these two things add together? Now, of course, reality is always a little bit more complicated than a cartoon. First, we need to be able to manipulate two separate input pathways specifically. Then we need to find cells that receive input from both of these. And then we need to be mindful of the temporal dynamics of these interactions. All right, so lucky for us, the first problem has really been solved for us already by a whole lot of really brilliant people who spent probably a better part of a decade developing novel channelrhodopsin variants that allow dual innervation. The way this works in practice is that we inject the classical blue light sensitive channelrhodopsin into the anterior cingulate cortex. From now on, I will probably just refer to this as the frontal input, and the red shifted crimson into a sensory area like V1 or A1. On this image, you can see the expression of these two opsins in the parietal cortex. And then we can cut acute slices from the PPC and make whole-cell patch clamp recordings. And basically all we need to do is use the right wavelength to activate the correct opsin. So this sounds easy enough, right? Well, there is a caveat. Crimson, although it's sensitive to red light, it actually retains quite a bit of sensitivity to blue light as well, which makes the separation of these two opsins non-trivial. So we went ahead and built a rig with some super fast LED lights that allowed us to titrate the amount of blue light that we use in these experiments to avoid the activation of crimson. All right, so we have a system where we can separately manipulate sort of frontal and sensory inputs to the PPC. Next, we wanted to see whether there are any cells that receive dual innervation. And to do this, I'm not going to go into all the technical details. I think if you work with opsins, you're probably familiar with this TTX4AP method. And honestly, sort of measuring synaptic latencies doesn't call for too much imagination. So let it be sufficient to say that we explored two input combinations, frontal auditory and frontal visual. And we found that for both combinations, approximately 90% of layer 5 pyramidal neurons in the PPC receive dual innervation. All right, so that's great. We have our technique. We have dual innervated cells. So what does an experiment look like? Well, we can activate the sensory or the frontal input separately or in combination. We typically used either coincident 0 millisecond ISI interstimulus interval or 50, 100, or 250 millisecond delay. On these traces, the red trace is the actual real recording, while these black dashed lines are the arithmetic sum of the two separately activated inputs. So if you look at a blow-up image of the 50 millisecond delay, you can see that the amplitude here is significantly larger than what you would expect if you predicted a linear interaction between these two inputs. We can display this in sort of an integration graph where the x-axis will show the delay, the interstimulus interval between activating these two inputs, while the y-axis is sort of an integration ratio that's calculated very similarly to the way one would calculate paired pause ratios if you were looking at a single input. But so here we're looking at two inputs. So this integration ratio is kind of a mixed pulse ratio, but for the sake of simplicity, we will just call it integration from now on. So On the population level, we found that if the two inputs are activated coincidentally, which is this 0 point here, you basically get linear interaction. However, if there is a delay between the two inputs, there is significant supralinearity in the processing in these cells. And then again, at large delays, that goes away. And so this pattern actually held true for frontal auditory and frontal visual integration. Of course, working in a cortex, in the mouse cortex or in any cortical region is not quite as simple as this. In layer 5, there are at least two very well defined and well studied neuronal types. These thin tufted regular spiking cells that typically project out of cortical regions, and thick tufted bursting cells that are known to project subcortically. We have established with the previously not detailed methods that both of these cell types receive dual innervation. However, when we started these experiments, we just recorded randomly from all layer 5 cells. So predictably, our results were all over the place. However, when we separated the two cell types, we found that the bursting neurons actually showed strong superlinearity at coincident input, but linear integration when there was a delay. So we have two cell types here with completely different integration patterns. And so the next logical question is, what may be the reason for this? We know that dendritic nonlinearities are typically driven by either sodium channels, calcium channels, and NMDA receptors. So looking at this 50 to 100 millisecond delay, we predicted an NMDA receptor involvement in here. And indeed, in regular spiking cells, when you look at this 50 millisecond integration, when you block NMDA receptors, you can actually linearize the interaction. Of course, NMDA receptor block didn't do anything in the burst spiking cells at coincident integration. However, when you block low threshold calcium channels, that had a partial effect on coincident supralinearity that was accompanied by another partial effect from blocking sodium channels. When you block both sodium and low threshold calcium channels, you actually manage to linearize the coincident supralinearity in these burst firing cells. All right, I blasted through this last mechanism bit quite quickly, so I'm going to quickly recap what I told you guys about. The question we set out to look at is, how do parietal neurons integrate frontal and sensory inputs? So we activated two different inputs into the same cells, and we found that about 90% of these cells receive monosynaptic dual innervation. In burst firing layer 5 pyramidal neurons, coincident activation of these inputs resulted in massive supralinearity that was likely driven by cooperation between sodium and low threshold calcium channels. Conversely, in regular spiking neurons, we saw supralinearity when there was a delay between the activation of the two inputs, and that was predominantly driven by NMDA receptors. All right, just to finish up, I want to acknowledge the person who actually did most of these experiments, an exceptional graduate student, Daniel Rinner, in my lab, and our funding from the White Hope Foundation, the Center for Hearing Research at UCI, and the National Institute of Mental Health. And with that, I would like to thank you for your attention, and I'm happy to take questions.
Great, thank you. That was a really great talk. We do have a question from Pierre, which we'll start with. And if anyone else has questions, we've got time, so you can post them in the Q&A. So Pierre asks, for what reason is there data points only at 0, 50, and 100 millisecond time steps?
That's a good question. I mean, ideally, you would have sort of a continuous look at this. The reason is really a sort of a practical reason. You can't do infinite number of recordings. We have to sort of interleave single activation with paired activations, and then single and then paired, and then single and then paired. Testing these 4 time points already takes close to an hour. in whole cell mode, and it's just not feasible to do much more than this. The reason why we picked these exact time points, the coincidence is obvious. 250 is kind of obvious. You don't expect much synaptic interaction at that point. And in between, we essentially just broke it up to places where we expected the larger effects.
Do you see any change in the nonlinear interaction with repeated presentation? So do you see any evidence for plasticity at any of these time scales?
That's a really important consideration, and we actually designed our experiments to avoid plasticity, because that would obviously that could completely bias these results. So there is about 20 seconds delay between each individual activation, so that we are definitely out of the LTP regime, and we're not really close to the LTD regimes either. So we're trying pretty hard to avoid plasticity.
Great. So now we'll move on to our final talk. Thank you again. That was really a brilliant talk and very, very clear. So our last talk, we have Blake Joe, who is presenting research they did as an undergrad. Their talk is titled Brain-Wide Tracing of Inputs into the LP-ACC Reciprocal Circuit.
Thank you, Adam, for the introduction. Thank you, everyone, for being here. Today, I'll be talking about work I did in the last year in Mrganka Sir's lab at MIT. And my work was on the topic of mapping inputs to the reciprocal connection between the LP and the ACC in mice using disynaptic rabies tracing. So I first want to start off by giving a brief overview of the two central brain areas in this work and why the relationship matters. So LP is short for the lateral posterior nucleus of the thalamus, and it is the rodent homologue of the pulvinar in primates. The LP is retinotopically organized, and it's highly involved in transthalamic pathways. It's mostly known for its role in regulating visual attention through dense connections to the superior colliculus and cortical areas, which is exemplified here in work from the Allen Institute, showing LP's reciprocal connections to various visual and frontal regions. For this project, we focused on the relationship between LP and the anterior cingulate cortex, or ACC, which you are all familiar with by now. It is involved in action selection and the allocation of attention. So since a main goal of our lab is understanding how the brain integrates sensory inputs to make decisions, we were really interested in finding out what information the ACC is getting from the LP and vice versa. Now, inputs to these two regions have been extensively quantified by other groups, but not with specificity to their reciprocal connections. So what we wanted to know is what brain regions are contributing to bottom-up processing from the LP to the ACC? And conversely, what brain regions influence ACC as it conveys top-down behavioral priorities via LP to other subcortical regions and beyond? To characterize the circuit, we used disynaptic rabies tracing to selectively target inputs to either the ACC to LP projection or the LP to ACC projection. And Lena did a really great description of the concept of the rabies virus with the deleted glycoprotein. The strategy basically involves injecting a retrograde AAV with CRE in a target region, but the CRE travels presynaptically, and we can take advantage of that to label a second node upstream of your target region with the rabies virus deleted glycoprotein construct, along with CRE-dependent helper AAVs, which enable the rabies to spread one synapse further into the rest of the brain. So here, just to recap, we have injected our AAV CRE into the LP and rabies virus, along with helper viruses, into the ACC, with the result of brain-wide labeling of cells projecting specifically to the ACC and then to LP, which is why we have the term disynaptic rabies tracing. Conversely, we can also reverse the two nodes of injection, and we derived specific inputs to LP that then fed to the ACC. We were able to quantify these inputs by collecting images from across the whole brain, aligning each section systematically to the Allen reference atlas, and then annotating the location of labeled cells that we observe. So looking at the data, we found that the ACC to LP projection and the LP to ACC projection receive significantly different inputs. In broad strokes, ACC to LP receives most input from the cortex, whereas LP to ACC has less cortical input, but receives significant input from the midbrain. Zooming in to the specific brain regions that contribute to the circuit, what is immediately clear is that LP and ACC projections are modulated by a diverse array of inputs. Starting with the ACC to LP side, the main contributors are frontal areas, such as the prelimbic area, PL for short, and infralimbic area, ILA. Associational areas also contribute a large proportion of inputs. So notably the retrosplenial area, which we heard about earlier, RSP for short. Looking at the LP to ACC side, the distribution is quite different. We see that while the cortical input is split between multiple sensory modalities, subcortical input is dominated by the superior colliculus. We can also look at this data by organizing it by function. So here we have cortical data divided in this way. Looking at it, we see that overarching differences between ACC to LP and LP to ACC in the cortex are more evident. We see the bias of the ACC to LP side towards these retrosplenial areas and prefrontal areas, as well as the broad spread of sensory inputs towards LP to ACC, which we'll get more into later. In the same vein, by summing up the regions that make up these cortical categories, we see what functions are important to the respective sides of the LP to ACC reciprocal circuit. I think this data also provides perspective to how LP is viewed, because LP is primarily thought of as a visual thalamus, but clearly there's so much more going on. And I also want to take a moment to point out that motor cortices also provide a significant input to both sides of this and should not be overlooked. So here we have primary motor cortex, and this is the secondary motor cortex. The visual motor associations of the circuit are emphasized by superior collicular input to LP ACC. It is the biggest input to this projection. The literature around superior colliculus pulvinar projections largely regards retinal input and visual attention, which retinal cells project to the superficial layers of the superior colliculus. But looking in closer detail at the contributing superior colliculus subregions projecting to the LP-ACC circuit, we see that these inputs actually reside in the intermediate and deep layers and not in the superficial layer, which is interesting because while these deeper layers are still visually responsive, they are more strongly associated with motor and multisensory functions. These inputs from the superior colliculus to the LP and then onto the ACC also implicates the LP in a modulatory role in communication between the superior colliculus and the ACC. Because while the ACC projects directly to the superior colliculus, this is a one-way street. The reverse is not true. So LP could serve to provide a path for visual motor information to be filtered and returned from the superior colliculus back to the ACC. The LP and ACC also receive visual information from the cortex, but again, the inputs are asymmetric. Looking first at the top-down ACC to LP projection, the visual input is mostly from higher visual areas, namely the anteromedial visual area, vis-AM, and the posteromedial visual area, vis-PM. In contrast, inputs to the bottom-up LP to ACC projections are much more spread out between the various visual areas. And it's worth noting here that all anatomical regions were defined using the Allen Reference Atlas, and vis-A actually overlaps with PPC a little bit. So the difference in visual input between these two directions is further highlighted by the broader context of other sensory modalities in these projections. Visual input dominates ACC to LP sensory inputs, and this dominance emphasizes the relative contribution of these higher visual areas. In contrast, in the LP to ACC direction, there are multiple modalities contributing significant input, which kind of overshadows the visual input, because you can see that primary somatary sensory input is much higher than primary visual input. And the potential importance of these non-visual modalities has actually been shown by work from other labs as well. There's recent work showing that neurons in the LP respond to whisking, which shows the somatosensory effects. And other work has shown that the LP can moderate auditory tuning via reciprocal input to the auditory cortex. So to summarize, today I showed you an anatomical mapping of the specific areas which influence the communication between the LP and the ACC. The ACC to LP projection is characterized by associational, frontal, and visual inputs, while the LP to ACC projection receives a broad multimodal array of sensory inputs, as well as superior colliculus and inhibitory inputs from the midbrain. Taken together, these data highlight the LP as a switchboard of sorts for thalamical cortical communication across many areas, and it provides a roadmap for understanding how the LP interacts with frontal areas to modulate the selection and allocation of attention. I'd like to thank everyone in the SIR lab for support in my work over the last year, particularly my PI and mentors who provided help and guidance throughout this project, the DCM staff for animal care, and finally Ian Wickersham's lab for providing virus. And thank you to all of you for listening. I'll take any questions now.
All right, great. Thank you, Blake. That was a great talk. Very impressive amount of work. We do have time for questions. So if people have questions, post them in the chat. I'll lead off first. It's amazing how diverse the connections are within this circuit. So you have really it seems as though it spans most of cortex. Is there anywhere that's relatively silent between these two regions? So is there kind of a space where you might say these areas don't get information from?
Well, like you said, there was a remarkable variety of inputs. I definitely received a on-the-go education on tons of brain regions I'd never heard about. I think the hippocampus is very sparsely labeled, which I think is expected. Honestly, I think we were also expecting more primary sensory input, whereas we see much more secondary and higher order input.
Yeah, that's really interesting.
I have a question. So you basically looked at the specific neurons that are, either receiving inputs from the ACC or sending out projections to the ACC. So what's the proportion of these neurons compared to the total number that is present in the LP and vice versa? Like what proportion of LP neurons are the ones that project to ACC what percentage of ACC neurons are the ones that project to LP?
So we can actually go back and look at LP and ACC in this data. So here we have LP to ACC to LP. Is this kind of what you're getting at? The proportion of inputs that you see? Or
the question was most not in terms of inputs, but because you kind of use this special strategy, right, for looking at the inputs that, if I understood it correctly, that you looked at the inputs that LP LP neurons that project to ACC, what their total number of inputs are, right? Like that specific, those specific neurons. And so I was just wondering, what is the total proportion of those neurons compared to the rest of the LP population?
I would say it's pretty sparse. We were targeting medial LP specifically, and just because of the methodology that, you have to have the Cree virus travel, and it depends on all the viruses being in the same place at the same time. The specificity is very high, so I think it is relatively sparse, especially in the LP. I think the rabies virus was actually designed for cortical labeling rather than subcortical labeling. So definitely, yeah, more sparse in the LP compared to the ACC. But given that, there's a remarkable number of projections that resulted from that relatively sparse population, which speaks to how well connected LP neurons are.
Great. Well, that was a fantastic talk. Actually, 3 fantastic talks. So I want to thank our speakers again for that wonderful session, which we'll wrap up now. Hopefully we'll see everybody again at future sessions. All right.
Thanks, Adam.
Then I'll show you that replay appears after a single experience. Then I'll show you how it slows down with subsequent experience by adding detail, and then a little bit of some summary, and if time, some implications. So for background, place cells in the hippocampus represent space. So on the right, showing place fields of 4 different cells in an open arena. And on the left, you can see that if you record from enough cells, place cells cover the entire track. So here, looking at a linear track, our lab records in rats, and all the experiments that I'm going to tell you about today are rats running back and forth on a linear track from one reward to another reward, back and forth. So because we have the encoding model of place fields, we can use really simple Bayesian decoding, memory lists, no prior, to decode the animal's position at any one time during behavior, because we have the place fields of all the neurons, and then we have when they're spiking. So you basically can decode where the hippocampus thinks the rat is at any one moment in time. So this means that it's possible to accurately decode where the animal is while he's running across the track. So here I'm just showing a schematic of those about 100 neurons that were recorded simultaneously. And you can see that as the animal runs across the track, the rat's actual position is in blue. You can see that each cell fires in a different location across the track. And if you look at the decoder estimate of position, so this is high probability in white, low probability in black, you can see that you can accurately track where the animal is as he's running across the track. Then when the animal gets to the end of the track, stops running, starts to drink the reward, what you see is that these cells seem to fire in bursts of activity. And so if you use a shorter timescale decoder to decode what the spatial information is within these bursts of activity, then you can see that they are not actually all firing all at once. They're firing in these sequences where the cells fire in the order in which they fire during behavior. And so you can decode that as this trajectory that goes across the track. And this is associated with a sharp wave ripple in the LFP around 150 to 250 hertz. So, you know, we think that these replays are really cool and they could be important for a lot of things, including maybe memory. If you disrupt sharp wave ripples, you see deficits in learning. And, you know, it would be very advantageous to be able to replay experiences in order to consolidate those memories for later decisions. So one question we have is how flexible is replay? For instance, you know, the hippocampus is thought to be important for episodic memory. Could replay also be supporting those types of functions? Episodic memories happen one time, you know, they're not like when a rat is running up and down across the track over and over again, which is usually the way that we look at replay. So we wanted to ask whether replay would appear after a single experience. And so to look at that, I looked at data recorded by Brad Pfeiffer, a former postdoc in the lab. And what he did was he allowed a rat to sleep in a familiar rest box before a run, then placed the rat on a novel track and allowed the rat to only run either 1 or 2 passes across the track. And then took the rat off and put it in the same familiar sleep box. And so the way that we identify replays is with 2 criteria. So this is a nice replay. You can see it has a trajectory across the track. This is the decoded posterior, so the position on the y-axis and the time on the x-axis. And here are 2 other events. So we're looking at just any bursting activity where there's lots of hippocampal neurons firing at once. And these don't look like replays here. And so the 2 criteria that we use to pull out good replays are the weighted correlation, which will be high when the posterior is tight and it's traveling in a direction across the track. And then the max jump distance in between time bins as a proportion of the track. So in between each time bin that is decoded, how far is the replay moving? And so this event does not pass criterion because it has a low weighted correlation, even though it has low max jump distance, whereas this event seems to just, you know, represent one side of the track and then have a huge jump to the other side of the track. So that wouldn't qualify as a replay here. It's a large max jump distance. So this is how we pull out good replays. And if we look at, say for one session, the 3 best events in this pre-task rest, these are what these 3 best events look like. So they don't, you know, look like good replay, but after just one pass across the track in this session, you see nice replay events that pass our criteria to be replay. And if I look across all of the rats that Brad recorded, what you see here is what I'm doing is comparing all of the, so for each session, I take the number of replays that actually happen, compare it to a shuffle distribution where the time bins of each replay are shuffled. And so what you're seeing here is a p-value for each of these across both of these dimensions of our criteria. So max jump distance and weighted correlation. So nice replays are in this green box. And what you can see is that in this pre-rest period, there's no more replay than you would expect by chance. But after just one lap on a novel track, you see that there's robust replay that appears after this single experience. So this was really cool to see. And then the next question we wanted to ask was, after that first experience on this novel track, does replay change as the animal gets more experience in that environment? And so this is data that it was collected by a former graduate student in the lab, Delia Silva. And what I'm showing here is the behavior, an example session. That rat is running up and down this track and pausing at the ends. And I just pulled out 7 different example replays that passed our criteria to be replays. On the top, I'm showing the decoded posterior, so the position across time. And on the bottom, the spiking of the hippocampal cells that went into that posterior, ordered by where the place field is on the track. And so what I hope you can see here is that as the animal takes laps across the track, the replays start off rapid, really short in time, and they just dart across the track. And then with experience, these replays get longer and longer. And we looked at across across all the animals in dozens of sessions, and saw that replays do increase in their duration and decrease in their slope with experience. So these are all on novel tracks. And I won't show you this today, but I've narrowed this down to local replays that are significant in an environmentally novel experience. So one thing I wanted to look at is, when I was thinking about the mechanism that could support this, is whether the experience is really what's necessary to do the slowing down, or whether being in a novel environment just triggers some process that happens on its own in the hippocampus. And so to look at that, I looked at the replays during this rest period that Brad recorded to see whether after a single pass on a track, the replays were slowing down on their own. And what I found here, I'm looking at just the duration across the period during the rest period, and I found that they did not change. Their slope did not change either. Then when you put the rat back on the same novel track that it's only experienced one pass on and let it explore this novel track, then the replays start to get longer and slow down. So it really does seem that it's the experience of the lapse, not just time, that is making these replays slow down. Another thing about replays is that they don't move smoothly across the track. They actually jump and hover in these steps across the track. So I also wanted to look at how replays slow down at this time scale. And so they slow down locked to this slow gamma rhythm that's about 25 to 50 hertz. And what I found is that replays slow down by adding smaller steps. So these jumps and hovers across the track that are thought to maybe be a signature of like underlying attractor dynamics don't seem to change in their frequency or any of the temporal aspects of them, but the replays just add more of these steps on. So in this schematic, you can see in the beginning, there's 3 big steps across the track, which is how the replay gets across the track. But then in later replays, you could have 5 smaller steps across the track. So across all of our replays, the number of steps goes up and the step size decreases across laps in a novel experience. And stuff that I won't tell you here, we actually think that the addition of these hover points where the replays hover themselves are experience dependent. There are fewer hover points than you would expect on the first lap. So it may be that you need to have experience in order to insert these hover locations into these replays. So in summary, what I told you today is that replay appears after a single experience and it lasts irrespective of where the rat is. I did some other experiments to look at that. That significant replay of a local environmentally novel space slows down with experience, and that replays slow down by adding detail to the representation in these hover points. And so I think that this could be, this could make sense as a good mechanism to have when you first are in a novel environment. You want to have quick representations that get you across the track to review, the safe spaces, to update your values of the reward that you just got in this environment quickly. And then later with experience, you may want to slow that representation down in order to add detail. Any downstream area that is listening to these representations would be able to have more detailed representation of experience to maybe build on some cortical or other schematic representation on top of that, or to be able to update states with more granularity. Yeah, so in summary, I want to acknowledge all the people whose data went into this work. Ting Feng did a lot of the early data organization. Delia Silva did the novelty data. I didn't get to show this PFC work that went in, but Jiajing Wu did that. Ellen Ambrose did this reward manipulation to test whether reward would change replays, which we found did not, which I didn't get to tell you about. And then Brad Pfeiffer did the single lap experiment. And that's all. Thank you all for listening.
Great, ended right on time, perfect. Thank you for that excellent talk, Alice. We already have quite a few questions, so let's get into that. Remember, we have 3 minutes. First is from Eleanor Duvel. Did you look at interneuron activity and could they contribute to slowing down?
Yeah, so I sort of skipped that. just due to time. But yeah, we do think that they could contribute to the slowing down. It might be that they are less active in the beginning of experience. And I have this sort of idea that the connections between the pyramidal neurons are not as strong in the beginning because you haven't had theta sequences across the track, or you haven't had replays. But because there's a low interneuron drive, you are still able to have these jumps from these to these locations and these rapid replays. But then as the connections get stronger and inhibition goes up, you need stronger connections in order to have get across the track. But you have that because you've had theta sequences, you've had behavioral timescale plasticity and replays in order to get those connections to be stronger. So yeah, we think that inhibition may be a good potential mechanism.
That's a great answer. Thank you. So the next one is from Aditi Bishnoi. Sorry if I butcher any names. What is the length of the track? How many cells is it allowed to record in a single lap? And how do the number of cells affect your replay analysis?
Cool, great question. So the data I showed today, they're all from about 1.2 to 1.5 meter tracks. There's not that much variety between them. It would be really cool to look at this in a larger environment and see replays slowing down like really long replays slowing down. And yeah, we believe that getting the most cells is really what we're always trying to do. And so sessions where there aren't as many cells is usually around 100 or so neurons recorded simultaneously to 200 to there's a lot of variability, but sessions where you have fewer than maybe like 50, it's really hard to get replay. And so in the analysis that I was showing, all of that I was showing were only significant replays, and you really don't see a lot of significant replays when you get down to something like 30 or 40 neurons, unless you get really lucky about the way they are. But yeah, so we have to have a lot of neurons simultaneously.
All right, great. And let's see if we can answer this last one really quick. Eleanor Duval says, nice talk. Have the rats experience another track before the novel track? And do the one-shot replays also represent that other track?
Yeah, so all the rats have experienced another track before. And yeah, there definitely are like remote replays of other tracks. And that's also something that I looked at when I was seeing whether this effect was about the actual like gaining of experience. I looked at on a novel track where where the animal was just on a different novel track. And I can see whether the replays of the local environment slow down, which they do, but the replays of that first novel track don't slow down while the animal is on this second novel track. So they exist. There are these remote replays, but they stay the same as they did after the animal got off that track, which is consistent with the idea that you actually need to be gaining experience in order for these replays to slow down.
All right, great. Thank you so much for that. So next up is Aaron Milstein from Rutgers. He's going to be talking about offline memory replay and recurrent spiking neuronal networks. So thank you. Go for it.
Hi, everybody. Thanks for coming so I can share my work with you. I'm going to be asking a particular question today, which is what selects the content of an offline replay event? So I'm also thinking about the hippocampus like Alice was. And so, during the behavior of an animal, if an animal is exploring a spatial environment, then these sort of classical hippocampal place cells will be selectively activated for little snippets of the animal's experience and will be preferring to fire at particular spatial positions. And so these cells will fire in a sequence that's on the time scale of the behavior, multiple seconds. But then when the animal stops to lick reward or goes back to its home cage to sleep, the hippocampus will emit these population bursts that are called hippocampal sharp wave ripple events. And then some subset of cells will get recruited to spike, and those neurons will fire in an order that often corresponds to the order that they fired during the run phase. But my question is, well, what selects which cells are going to spike in a given sharp wave event, and what decides the order that they fire in? And I'm going to try to address this question with a really simple network model of spiking neurons. So I just have a population of feed-forward inputs. I have a population of excitatory cells that are recurrently connected. And I have a population of inhibitory cells that receives both the feed-forward excitatory input and the recurrent feedback excitatory input. And I won't be able to tell you the details about how I tuned this network model, but suffice it to say that I'm able to get a network model like this to reproduce many of these features of the behavior of the hippocampus. So individual cells can exhibit cell activity for spatial position. As a population, the individual place cells will tile the space, and at different positions, different subsets of cells will be active. And so we can use these sparse representations to try to decode the position of the animal if we have access to all of the activity of these cells. And then the hippocampus during run tends to have oscillatory network synchrony that's in the gamma band that's 30 to 100 hertz and in the theta band that's 4 to 10 hertz. And so I'm also able to get this network to emit theta and gamma oscillations even when the input is arrhythmic. And for the cell models, I'm using a very simple single compartment cell model that's a type of integrate and fire spiking neuron. It has conductance-based synapses. And then the only fanciness is that I can optionally have some spike rate adaptation in these cell models. And so I chose as an inhibitory cell, just sort of a fast spiking cell model so that these cells can kind of maintain high firing rates. But for the excitatory cell, I chose a bursty cell model. So the sort of first couple of spikes that a cell emits, given a constant input, will be at high firing rate, and then the firing rate will tend to slow down. And this helps reproduce this finding that, place cells tend to emit these kind of punctuated bursts of spikes that are paced by a theta rhythm. So you'll have to imagine that we're taking a snapshot of this network after some learning process has happened. And so whatever your favorite learning rule is, you can imagine that that's how this network was able to produce its spatial selectivity. And what that looks like is biased synaptic weights over both the feed-forward inputs and also the recurrent excitatory inputs. So a given cell in the network can receive random connections from any other cell. So any of these feed-forward inputs or any of the other excitatory cells. But then what gives a cell a nice sort of signal-to-noise ratio, high firing rate in field and low firing rate out of field, is some biased distribution of synaptic weights. So we've simulated the run phase now. This would be like an animal running down a track. It takes 3 seconds to run the track, and all of the cells are spatially tuned. So then how do we mimic the offline rest period? So this network is inhibitory dominated, and this network is not generating spontaneous activity on its own without any extrinsic drive. So we need to provide at least some kind of a seed of an input to get some spiking out of this network. So I sort of first chose like the most null stimulus that I could think of, which is just to choose random cells from the feed-forward inputs to be active together during a sharp wave event. So for 150 milliseconds, there'll be some randomly chosen subset of cells that are just firing at a constant rate. And so because these feed-forward inputs themselves were also place cells, each spike is like a vote for a spatial position. And so we can take the spiking activity of this feed-forward input population, and we can try to decode a spatial position. Where would the animal had been in space if we had seen this pattern of activity during run? And so you can see, since this was just a random input pattern, the decoded positions are kind of hopping all over the place. They span, they can jump half the track length in 20 milliseconds. Now, the inhibitory cells in this network, they respond to this random input by self-organizing into an oscillation. So these vertical columns of spikes that you see are the interneurons firing together with high frequency. And then the excitatory cells in this network model, they respond to this input pattern by selecting some sparse subset of cells to spike. And then different cells are spiking at different elements of what looks like a sequence. And so if we try to decode spatial position from this pattern of activity, we see that there are relatively short hops and sort of spatially contiguous positions are being visited. And this would correspond to like a trajectory that the animal might have been able to run. And so we can do this many, many times. We can have a bunch of instantiations of the network. We can throw 1,000 different random input patterns at the network like this, and we can look at what the output offline events look like. And so first of all, you can see that all of the positions along the track are visited at some point in some offline replay event. So we get sort of uniform coverage of the track. And then while the feed forward input was just garbage, and so it often had these sort of squirrely trajectories that have big hops and trace out a path that's even longer than the track itself, up to 3 times the length of the track. The excitatory cells in the network are tracing out trajectories that are much shorter and tend to be closer to like half of a track length. And then we can also decode velocity. So, every time bin is 20 milliseconds, and then there's some change in position. So we can compute a velocity of movement. And so many of the offline events have a mean velocity that's positive. So that would be corresponding to a sort of forward trajectory along the track. But then some of the events have a mean velocity that's negative. And so that would correspond to a reverse replay event, or like backwards movement along the track. And what's interesting is that this corresponds to about a 10-fold temporal compression. So in the run phase, it took 3 seconds to go 1 track length, but now we can go up to 3 track lengths in a second. Okay, but then what if we have the network try to learn something salient about the environment? So we know from experiments that if a reward is presented at a fixed location in a circular treadmill like this, that the hippocampus will allocate more neurons to have place fields that are nearby the rewarded site. And so we can mimic this in this network model as well. We can sort of allocate more cells to have their place field locations be nearby a simulated reward. And then we can play this same game. We can play in these random feed forward input patterns. And what we get out is sequences that are, spatially contiguous and tend to visit this overrepresented simulated reward site. And so again, we can do this many, many times, and we see that the positions that are nearby the reward site occur more frequently in these offline events. So this is sort of this network that sort of started off with like a null stimulus that didn't have any structure to it, is kind of already reflecting like the stored memory or the stored experience that's in the recurrent weights and in the feed forward weights of the network. So why is this working? So we can think about some of the previous literature looking at neuronal sequence generation, and something needs to break the symmetry. So one way to do it is with biased connectivity. So you could have a population of cells that corresponds to element A in a sequence, and they're kind of unidirectionally connected to the next set of cells that corresponds to element B. You could do this with synaptic weights too, where you kind of have stronger weights that are going in the forward direction than in the reverse direction. An interesting idea by Sandro Romani was that you could actually use synaptic dynamics for this. So if when a cell starts to spike, it has a high probability of releasing transmitter, but then that depresses over the course of additional spiking, then you can again kind of have some momentum that pushes a sequence forward. And then an interesting study by Kanika Rajan and colleagues had a network that had on average symmetric synaptic weights, but then some interaction between the external input and some fluctuations in the weights were able to drive a sequence forward. So what's going on in this network? So I can tell you a couple of things that will break this sequence generation mechanism. So one is that if I scramble either the feed forward weights on their own or the feedback weights on their own, that disrupts the sequence generation. So there does seem to be some sort of required interaction between the feed forward input and the feedback generated by the network. And then what's kind of interesting is that if I take this bursty excitatory cell model and I replace it with a regular spiking cell model, this also disrupts the sequence generation. So it seems like we were getting some extra benefit by having some spike rate adaptation. And this kind of reminds me of the synaptic depression mechanism, that if you kind of have something different about the beginning of the sequence versus the middle of the sequence, that can kind of add some momentum. But by having symmetric synaptic weights, this network was able to generate sort of both forward and reverse sequences without having like a fixed unidirectional bias. And then the last little bit that breaks this sequence generator is if I just leave out that the objective that this network needs to generate rhythms. So if I tune this network to be sparse and to be selective, but I don't require it to generate theta or gamma oscillations, then the sequence generator also gets disrupted. And so it seems like there is some extra benefit by having the interneurons provide these kinds of intermittent windows of synaptic integration, and that helps to kind of discretize the sequence and add some push forward. So thank you for listening. This work was started when I was at Stanford with Ivan Šoltes, and there were two fantastic Stanford undergrads that helped me with this project, Sarah Tran and Grace Ng, and I have a couple of other great collaborators for other projects. I'm at Rutgers now, and this work was supported by an NIH Brain Initiative grant and an NIMH grant, and used supercomputers that are operated by NSF and by the Department of Energy. Thank you.
Thank you so much for that, Aaron. All right, we have a question, just a sec. All right, so this is a question from Mengsen Zhang. Does the random FF induce a shape wave?
Well, so a sharp wave would be detected like with an extracellular electrode and would reflect, current flowing across dendrites and at synapses and like the spiking activity of like populations of cells. So I don't have that, but I can just take all of the spiking output of the cells and kind of sum them up and look at the total population activity. And that's what I filter to try to look at the oscillations, the theta and the gamma oscillations. And so, yeah, I mean, when you start to provide the feed forward input, then you see this big increase in spiking, and it has a ripple oscillation that's riding on top of it. So maybe that's as close as I could get to mimicking like the actual sharp wave component.
Great to know, thank you. And let me just, close that. And we had another question that referred to Alice's work for you, Aaron. This is from Pierre Orhan. Would you explain Alice's velocity? How would you explain Alice's velocity slowdown in your model? Or
yeah, actually, I really liked Alice's idea that sort of initially you would form sort of a coarse representation of some bits of the environment, but then with additional experience, you'd be kind of refining the resolution and the sort of completeness of your representation. So it's probably that, like, I've taken a snapshot here of this network kind of at equilibrium, like after all of the weights have been learned, but it might be interesting to instead climb in at different parts of the training phase. And look how much sequence generation do you get when there's kind of an incomplete representation after the first lap or two. That's an interesting idea.
Yeah, that would be interesting. And I think I may have, there was a clarification to that question. So if you take a look at that question in the Q&A, you can provide any further refinement to that. And we have another question from Diogo Santos Pata. Hi, really nice to see this. Have you looked at phase precession?
Right. So what's interesting is that I build up the template for doing my decoding from the activity during the run phase. But I've also kind of binned it such that I'm mostly trying to capture the spatial selectivity of the cells. But if you do something like, if you really just bin it with really small bins, then you can try to decode position in time intervals that are smaller than a theta rhythm. And then you actually do see that if you try to decode the run phase, that you get these little kind of sweeps ahead and then a reset that kind of looks like something that would be like theta phase precession. And so it could be that, yeah, just by virtue of having sort of rhythmicity that you already get a little bit of this for free in this simple network.
Great point. Thank you for that excellent talk. We're going to move on to Ryan Harvey. So Ryan's from University of New Mexico, and we'll be talking about hippocampal replay alterations following moderate prenatal alcohol exposure. Go ahead, Ryan.
Okay, yeah, thank you for the introduction. Yeah, so today for this, I'll be talking about hippocampal replay and sharp wave ripple activity following moderate prenatal alcohol exposure. So kind of as a broad background, globally around 9.8% of individuals drink or consume some amount of alcohol during pregnancy. And so up above, I'm just showing kind of the world map color coded by percent of individuals that drink per country. And this is an issue because even in pretty moderate amounts, and when I say moderate, I mean kind of like around 2 standard drinks in a day, so well below like the legal limit, you do see effects in hippocampus and other areas. So for one, there's a recent study that came out that showed even given a moderate amount up into adulthood, you see decreased numbers of GABAergic interneurons throughout the entire hippocampus. Also, there's been several papers showing reduced dentate gyrocell TP, and there's been several studies looking at how memory performance and spatial memory in particular is at a deficit following moderate amounts of alcohol. And so a recent paper that came out of our lab is we recorded hippocampal place cells in a rat with moderate amount of alcohol exposure. And for the rest of the talk, I'll just call it PAE for prenatal alcohol exposure. And so recording the place cells, we found that their place fields were slightly unstable over time in a couple different environments. Right here, we have a cylinder, and here we have a linear track. And so the field was kind of moving around slightly over the duration of the session. Also, along with that, we recorded we measured the intrinsic theta frequency of the individual units and found that following alcohol exposure, the intrinsic theta rhythms were actually slowed down or decreased. And so when you consider theta sequences, that has some pretty broad implications to have a slower theta rhythm. And so with that kind of background, leads me to my broad aim is to explore the integrity of sharp wave ripples and replay events following prenatal alcohol exposure. And so gone over pretty well in the previous two talks, sharp wave ripple events and replay have been really implicated in memory. If you lengthen the duration of the ripples, you can recruit more neurons, pyramidal cells, and interneurons, and actually might increase memory performance. Alongside that, if you kind of truncate or shorten the duration of ripples, you can decrease memory performance. And because with this PAE, you do see decreased interneurons, as well as you do see decreased memory performance, I expect that these ripple events and this replay events will be somehow altered following alcohol exposure. So going to my methodology, throughout gestation, we exposed the rats to around 5% of ethanol, which is a pretty moderate amount. They're born, aged to adulthood, and we implant their hippocampi with electrodes to record single units and LFPs. And the task is fairly simple. First, we put them on a pedestal where they kind of hang out for a little bit of time while we're recording them. We drop them into a linear track where they kind of just move back and forth and have some time at either end to eat some Fruit Loops, and so we can record some sharp wave ripples as they occur. And after this, we put them back on a pedestal. So kind of like a traditional kind of paradigm for this. So getting to the results of this, what you see here are two ripples, one from a control rat and one from a PAE rat. And the white is just a wideband signal over the spectrogram, and beneath that is the bandpass filter signal. And from these ripple events, you can kind of take out several different features, such as duration, or like how wide they are, the frequency of the ripple, the amplitude of the ripple, and like the ripple interval, just to see how they're doing between groups. But along all these dimensions, I didn't find any differences between the two groups. So if there are any differences caused by PA, they're not along the dimensions of the ripples themselves. So the next thing I did was look at what the cells are actually doing during the ripple events. So on the left right here, we have an interneuron, and the right we have a pyramidal cell from hippocampus. And you can see that both these cells are actually pretty highly modulated by the sharp wave ripple events, and that they fire maximally during the ripple. But when we kind of move out and look at all the cells combined across groups, you can see that there is slight decrease of modulation of the fast spiking interneurons in the PAE group. And there is slight decrease in the pyramidal cells, but just not to the same extent as the interneurons. And alongside that decreased modulation, I did find fewer significant interneurons that were modulated by the ripple events. Okay, and you might have a next question of how does this affect hippocampal replay when you have decreased modulation of the interneurons and fewer interneurons being recruited into the replay events? And so here's like a kind of standard schematic where I have a good number of place cells, and here are their tuning curves. On each row, we have the cells raster over a good amount of time. And from both these things, we can use a Bayesian decoder to decode out their current position. And white right here is the actual position of the rat as they make their way down the linear track and kind of have a rest point right down here. And if you kind of squint your eyes and look in here, you can see there's about 3 kind of bursts of activities. And so those would coincide with sharp wave ripples. And if we zoom into this last one right here, you can see that the cells are kind of firing in this sequence, and using a more finely tuned or more temporally precise Bayesian decoder, we can decode out this trajectory that the rat is kind of possibly thinking about making as it goes back to the other side of the track. And so from those trajectories, we can, again, kind of pull out several different features, such as linear weighted correlation. And right here, it's just represented in absolute value, the distance of the trajectories, the speed, and the step size. And while the effect sizes between the groups are fairly small, denoted with Cliff's delta right here, I do see like the general finding of a truncation of these trajectory events. And so kind of meaning that while the controls kind of have, on average, more of a complete kind of representation of the entire track during the replay, the PAEs kind of have more stationary replay event. Okay, to summarize all those findings, first, the ripple features seem pretty similar between the 2 groups. Also, the interneurons are recruited in fewer numbers and with decreased strengths into the sharp wave ripples. And finally, the replay trajectories are slightly truncated. And so the next step that I'm going to take is to go in and quantify interneuron and pyramidal monosynaptic connections to see how precisely these interneurons are kind of affecting these pyramidal cells during the sharp wave ripples. And maybe that might help explain why the replay events are slightly truncated. And so with that, here's my acknowledgements. I'd like to thank my team and collaborators and my funding source. Thank you.
Thank you, Ryan, for that great talk. We're just going to wait a second for a few questions so people can finish typing. All right, it seems that I had missed a question that was asked into the chat. And now we also have on the Q&A. So just give me a sec. All right, so I'll do this chat one first because it seems like it popped up first. This is a small question from Cooter Bilal. Sorry if I butchered that. So when looking at your graph showing interneurons firing relative to the SWRs, control seems to be firing rhythmically, but not for PAE. Do you have any explanation for why that might be?
Yeah, so that was one of the big things that I found when I'm doing these analyses. I guess the traditional thought is during the replay events, the rats are kind of motionless, and so theta powers could be down. But for whatever reason, the fast spiking interneurons that I've classified, they were still theta modulated around the sharp wave ripple events. And when I made a null distribution, kind of like taking the same amount of ripple events, but just like taking random points in time and recreating those plots, it was a pretty standard kind of flat line. And so my control cells were modulated in the ripples, but also had a slight theta modulation, which I didn't see in the pyramidal cells. And I looked at literature and didn't find too much about that, but maybe you have something to share with me, or someone does.
Yeah, I guess if anybody in the audience has any insights, we'd love to see it in the chat. In the meantime, maybe we can address this next question from Emily Airy Jones. Have you looked at slow gamma during the SWRs in the PAE rats?
Yeah, hi Emily. No slow gamma so far, but that'll definitely, that's on my list of something to do next. Yeah, but great question and great line of research to look into.
Yeah, that does sound like it'd be very interesting. Emily says hi back. All right, cool. So we're just running into the end of this session. I'm gonna let people send in a few more questions if they'd like. Technically, we're ending now, it's 2:45, but we'll just wait for a second. In the meantime, I'd like to thank the panelists for their fantastic talks. And I really appreciate the engagement of the audience here too. So good job all around, guys.
You did a great job as MC too.
Oh, thanks. Lots of appreciation from the audience for the talk, so I'll clap on their behalf. So we're just hanging out right now, seeing if any questions pop up before we're kicked out. But if nothing pops up in the next minute or so, we'll just close the session. Because technically we're done. So we have a hand raised from Eleanor Duval. All right, and we can definitely invite you to come hang out with us. I think the host can, oh, you were just testing, okay. All right, so we do have a question. I'm sorry, I'm gonna butcher this, Quadr Bilal. Thanks, I see you laughing at me here. Small question again, did you look at sleep SWRs? I think that's for Ryan. Oh, that is for Ryan.
Yeah, so unfortunately, I didn't have any sleep data. Originally, this data was collected to look at place cells and for that place cell paper that just recently came out. If I had a 2-year extension on my PhD, I would probably go and collect some sleep data. Yeah, that would be something for the future to look in this model to see how they respond during sleep, especially because there are slight sleep disturbances in this model.
I mean, graduating, having extra sleep data. All right, cool. So since we're about to get kicked out in a second anyways, I'm gonna wrap up. Thanks again, everybody. This was really a pleasure. We learned a lot about replay. All right, take care all. All right, hi everybody, welcome to the session. I'm going to give kind of a minute before we get started, but I just want to let you know what the format of this is. We'll have 15 minutes per speaker. That'll be 5 minutes for the speakers to introduce their topics, and then 10 minutes for questions. And feel free to come on screen and ask your questions. This is really kind of laid back. We'd love to have as much audience participation as possible. So just feel free to get engaged. All right, and we'll get started in just a second. All right, so let's kick this off. The session today is going to be on neuron development and regeneration, and we'll be starting with Julia Kaiser at the Burke Medical Research Institute. She'll be talking about development of cortical projections to the brainstem versus cervical spinal cord. All right, go for it, Julia.
Yeah, let me share my screen. Someone else is sharing. Is it okay if I just stop? I guess so. Where do we have the screen? Is it this one? Maybe. Can you see the full PowerPoint, or is this just the?
There we go.
Okay, great. Okay, so as I was introduced already, hi, my name is Julia Kaiser. I work at the Burke Neurological Institute, and I'm going to introduce you a little bit into the development of cortical projections to the brainstem versus the spinal cord. First of all, I need to introduce you to this little guy, the corticospinal tract. It's the major descending tract of the cortex that controls our motor skills, and I think this is a function that we all highly appreciated during the quarantine. And in brief, upper motor neurons that are in the cortex project down to the spinal levels, where they directly or indirectly innervate lower motor neurons that then control the muscles that allow us to hold glasses and enjoy life. And as you can imagine, a lesion to this corticospinal tract by any of the mentioned or many other diseases or neurological conditions will leave the patient in, have a devastating impact on the quality of life and leave the patients at a point where they need care and assistance throughout their life. So that's obviously something that we want to be able to help them with in the future by regenerating. One thing that is also known about the corticospinal tract, as for many other cortical areas, is that there is a so-called somatotopy to it, meaning that certain areas in the cortex and also in the corticospinal tract specifically control different parts of the body. And it does that by actually extending the axons to different sites and target sites within the body, for example, found in the brainstem or in the spinal cord on cervical and lumbar levels. So far, it is not clear yet how this segmental axon targeting is specified during development, or in other words, how does a corticospinal neuron know what it wants to be when it kind of grows up? So that is a question that we are addressing in the SANI lab. And my supervisor, Vibhu Sanu, showed in his previous research that there are actually at least two subpopulations of the corticospinal tract that extend their axons either to the cervical spinal cord or to the thoracolumbar spinal sections. And that there's also a transition zone between the cervical and the thoracic spinal cord where axons make a molecularly driven decision to either stop in the cervical spinal cord or to cross this transition zone into the lower levels, like the thoracic and lumbar spinal cord. He established that this decision is made early on in development, but it is maintained into adulthood, suggesting that there's a dedicated molecular program that steers this. And indeed, he found that thoracolumbar projecting corticospinal neurons can be identified by the factor CRIM1, and that a cervical neuron, if it wants to not cross over this transition zone, needs to repress CRIM1. So far, so good. He looked at this mainly based on the fact that the cervical spinal cord neurons are known to innervate hand functions or control hand functions, while lumbar spinal cord projecting neurons are more responsible for leg functions. And similar to that, we actually have another hotspot of motor control, namely the brainstem, where we know that facial movements are controlled, as well as arm muscles and sensory regulation of touch, for example. So there's an open question whether there is an additional transition zone where neurons have to make a kind of active decision to become dedicated brainstem neurons or cross over. And that is something that we look at in our research now. First, we wanted to know whether neurons do make this decision early in development, And for that, we injected a retrograde tracer into the peduncle, so in the brainstem, that labels all of the neurons that have an axon at this point in time, which is P0. So this is where we expect the axons to be here. This actively labels all of the neurons in the layer 5, which you can nicely appreciate here. And if our hypothesis is true that there is a subset of neurons that specifically projects only to the brainstem, if we inject the tracer on the cervical levels later on, we would expect that there is a subset of neuron that is labeled, and indeed that is the case. So we see that first here in the cross section, but also the quantification here. You can appreciate that in the lateral cortex specifically, a lot of the neurons are only labeled by the yellow tracer, not by the magenta tracer, showing that about 80% of the neurons actually become dedicated brainstem projecting corticospinal neurons. We also wanted to know if this decision is maintained into adulthood, and for this we did a so-called timestamp experiment where we switched on a TD tomato receptor, TD tomato reporter that we give in the cortex by injecting a retrograde tracer switch on molecule in the cervical spinal cord at P2. So this is 2 days after birth. And any axon that is here present in this area at this time point will get switched on and will express the reporter, and we can then count the axons on different levels throughout this throughout the neuroaxis, and we identified that there is actually a massive loss of neurons that projected onto the cervical spinal cord. So these neurons, like 90% of the neurons that were projecting to the cervical spinal cord at P2, now at P28, when we looked at this, are actually pruned back. So on top of brainstem neurons making the early decision to become brainstem neurons, cervical neurons now also decide to become brainstem neurons. This is especially interesting because in contrast to the cervical thoracic zone, as I mentioned in the beginning, we don't see this drastic reduction. So this seems to be a very brainstem specific thing. And to cut a long story short, we also have a handle on a molecular marker that projectively marks brainstem projecting neuron. As we can see, a double labeling only with our tracer that we inject into the brainstem, but not when we inject it into cervical spinal cord. So to summarize this, additionally to the cervical thoracic transition zone that was previously established, we also now show that there is a brainstem cervical transition zone that is indeed established early in development, that the decision is maintained into adulthood, but that there is a massive amount of refinements, and that we have a molecular program that steers to this, and CalSCHLAG14 will allow us in the future to look at the moleculars that steer the transcript. We can do transcriptomics to look at what exactly makes a brainstem neuron, a brainstem projecting neuron versus acervically projecting neurons, and it will also allow us to do some loss and gain of function and look at motor behavior as well as axon targeting. So stay tuned, and with that, I'm happy to take any questions.
Thank you, Julia, for the excellent talk. We'll just give people a minute to send in any questions they might have in the Q&A. You can also raise your hand and we'll invite you to come talk with Julia. We'll just give people a minute to type those out. All right, I think that we'll just take just a little bit more time, wait for people to type out their responses, but we can always allow people to ask questions throughout the duration of this talk. So if you end up having a question for any of the panelists, feel free to send those in. I do see that we have a question right now. This is from Kelvin Jones. If CSNC cells express CRIM1, they will grow past the transition. Isn't this true for the CSNA?
So in this case, I am not entirely sure what they refer to with CSNA. I'm just going to assume that that's got maybe means CSNB, which could mean our brainstem projecting neurons. No, Carolyn, no.
Oh yeah, and Kelvin, if you'd like to clarify, feel free to, you can follow up on that Q&A or in the chat, and I'll just relay that. Or you can come on screen and clarify if you'd like.
Yeah, so far the experiments have been done. So this is the work that has been done mainly by my supervisor, Vibhusani. So I'll just briefly refer you to his upcoming paper, but generally overexpression of CRIM1 does lead to to more axons growing into the thoracic zone. Of course, we don't know if these that do then finally grow over, what their molecular identity was initially, if this was a cervically projecting neuron, or if this could have been a brainstem projecting neuron beforehand. But that's definitely something that we want to look at in the future to find out which subtype of neurons can be used or can be manipulated to grow into a different target, because this will actually be really interesting also for spinal cord injury models, stroke models, ALS models, because the idea of this whole research is not only that we get, that we find out the molecular identity, but that we can maybe then in the future use those molecular identities to steer the regenerating neurons to their respective target that we want to get. Like, a lot of the currently applied regeneration models, all they're focusing on is getting more growth and more sprouting of the neurons. But if the neurons don't have a map in their hand, just letting them run doesn't really help anyone. So we want to get them to their targets, and this research will hopefully help with that.
Yeah, those are good. Those are great points. Kelvin was clarifying, said they couldn't read the screen well. So yeah, they meant the cortical neurons that stop in the brainstem is what they were referring to.
Yeah, so I stand by my answer.
Excellent. Perfect. All right, we have another question that just popped up from Rebecca Blume. Sorry if I butcher names, by the way. Is this specific for motor neurons, or does it also apply to neurons mediating top-down regulation of sensory neurons in the dorsal horn?
Now I would love to have a clarification of what this means, if this is true. So this is probably the disadvantage of Zoom meetings. This would be easier to discuss in person. So I'm just going to assume that this specific targeting, it's not going to just be in the cortex. I mean, we also know from the Hox genes how certain neurons in the spinal cord get their own identification. And I am just going to go out on a limb here and say that for sensory, we can expect something similar. As it's a common pattern throughout development. So I'm just going to assume that bluntly, but I'm not aware of any research being done on this. So very interesting point though.
I agree. And that sounds reasonable to me. And Rebecca, feel free if you want to clarify that in the chat or to come on, feel free to do that. I see a question from Noah Golan. Hi, Noah. First of all, good to see you here. And she says, hey, Julia, lovely talk. I was wondering where the CRIM1 thoracic/lumbar transition zone data were published.
It's not published yet. It's in the revision at the moment. So stay tuned. It's going to be out. Anyhow.
All right. We look forward to that.
Yes. In the meantime, feel free to reach out to our supervisor, Vibhusani. I'm sure he's happy to talk about his research.
Excellent. All right, so I'm going to sit here. We should sit and just wait for any other questions that come in. In general, since this is a longer question period, we're just going to kind of hang out and see if anyone wants to come forward and talk on screen or have questions in the Q&A box. I've been enjoying our conversation so far, and thank you guys for all your lovely questions. All right, and we'll just sit tight for another, let's say, half a minute or so. All right, great. I think we're going to move on at this point, but feel free to send Julia any questions in the Q&A and I'll field them to her, or through the chat for those of you coming from other places like YouTube. All right, and I'm going to mark both these done. Okay, so we are ready to transition. Next up is Noah Pardell from the University of Alberta. They'll be talking about facilitation of transmission of axonal branch points by nodal GABA receptors, simulations with a reconstructed proprioceptive axon model. All right, go ahead, Noah.
All right, thank you so much. I'm just going to share my screen, and I hope everyone can see the screen. So I will start. Yeah, thank you for coming to my talk. I'm talking about branch point failure and the rescue thereof using ionotropic GABA A channels. I'll just move the slides. So we have a synapse, but a computational way of looking at the synapse is an input signal that gets modulated in some way to create an output signal. Now, one of the ways you can do this is presynaptically versus postsynaptically. So presynaptically, there is this thing called presynaptic inhibition. Classically, what this means is that the output signal is inhibited, but the modulation and why it's inhibited, the vesicular release is diminished, happens presynaptically. So if we look at the schematic on the bottom, we see this lovely action potential that has reached the terminal. And historically, we believe that there is these ionotropic GABA A receptors that allow chloride to, oh no, my thing, there we go, that allow chloride to efflux from the terminal, producing a depolarization. This depolarization was thought to reduce the resistance and create a shunt. And this would lead to less vesicular release. The problem is these GABA A ionotropic channels aren't actually located at the terminals. So what are they doing? They're actually located at the branch points. So here we have a schematic with the 1A proprioceptive axon in the top going through the dorsal columns and sends off an axon collateral that travels ventrally through the gray matter. If the ionotropic GABA A channels are at the branch points, then what contacts them are these GABA interneurons, which are the last of a train of interneurons that produce this phenomenon known as primary afferent depolarization, or PAD. This PAD can be measured more dorsally, and that's historically it has always been measured. So what is this PAD doing? What are these ionotropic channels doing at the branch point? Well, immunohistochemically, if we look at the branch point with this neurobiotin-filled axon, we see a co-localization of the GABA A channels and the voltage-gated sodium, the yellow and the white, respectively. In a more diagrammatic form, it can be illustrated like this. So what is it doing at the branch point? Well, for our model, we took morphometry from a serial electron microscopy study in 1991 and reconstructed it in the computer. On the bottom is a electrical circuit diagram that corresponds to the double cable structure that we created. And this branch point highlighted here is the one that is used in the study, as well as the one that will be used in the next couple slides. So keep in mind this branch point. So to kind of validate the model, we had this proprioceptive afferent that has been filled with neurobiotin in experimental mice, and we see the branching structure, primary, secondary, tertiary branches. We wanted to see if the model and the experiment had kind of the same diameters, et cetera, for these different classes of branches. And so, as you can see, the secondary and the tertiary branches are quite concordant in their diameters, and this would help validate the model against experimental observations. However, there's kind of a discrepancy between the primary branch. There's some reasons for this, one of which might have been that the original electron microscopy study cut a little bit more ventrally than we thought, and therefore it's actually a secondary branch. But there could be some other explanations as well. Lastly, another way that we validated the model is through pairing the action potential heights to make sure that they were somewhat close to the experimental data. And here's just a model trace. So with the ion channels in the model, we titrated the conductance of potassium and sodium over a wide parameter space to try to see how common branch point failure is. In the black sections, this means that an impulse was not even able to conduct on the axon. And the magenta sections mean that there was unphysiological behavior. It's too hyperexcitable. So this region that's bounded by this trace is the one where there was impulse conduction that reached that branch point I talked about earlier, but either in gray, it wasn't able to go into either daughter branch. So the red and the blue were not able to conduct. In blue or red, it means that one of the daughter branches was able to conduct and the other failed. In green means both were successfully permeable to propagation. So in that parameter space, the gray, the colorful one, branch point failure is common, as we see in the pie chart. So if we increase chloride conductance with these GABA A channels at the branch point, we should increase failure. So if we fix potassium conductance on the y-axis and put it on the chart on the right, and on our y-axis we titrate GABA conductance or chloride conductance, we would expect increased failure. We would expect the cells to go from green to blue or red to gray. But actually what we see is gray to blue and blue to green. This is an increase in, a rescue of the previous failure. It's increasing transmission. So actually presynaptic facilitation is what we're seeing. So lastly, we have an impulse which travels down the axon, is not able to go into one branch. But with the addition of PAD and these ionotropic GABA receptors, we're able to successfully go through both branches. Thank you so much.
Thank you so much for that, Noah. Great talk. All right, so again, we'll sit and we'll wait for the Q&A. Feel free to either send in your questions in the Q&A, or you can come on screen. I'll also monitor the chat for any questions that might pop up there.
Sure.
All right, so we have a question for you, Noah. This is from Gregory Piercy. He says, very interesting talk, first off, I agree. Does this suggest that presynaptic facilitation may be required for normal behavior and that a lack thereof might explain dysfunction?
Sure. So what we're thinking in the lab is that this presynaptic facilitation is being implicated in the synaptic priming of reflexes. So if there's branch point failure, that means that there's some sections of the collateral that are silent in everyday functioning. But if we're able to facilitate the impulse to go through that branch, for example, in a reflex when you need to activate certain muscles that were previously quiescent to, let's say, I don't know, on the train to stabilize yourself when the train shifts, then that's an example of a normal behavior that we think that this is acting as, the facilitation is doing. A lack thereof very well might explain some dysfunction or some areflexive states. I'm not too sure about the clinical side of if this priming wasn't there, but that would be my answer to that question at the moment.
Thank you. Yeah, I think that definitely answers that. All right, we are just gonna hang out here and wait for some more questions to come in on the Q&A or in the chat box. Again, you can raise your hand and we'll bring you on screen, but we'll just sit here and because these are the longer question and answer sessions. All right, great. We have a follow-up from Gregory. Would this help to explain a cutaneous facilitation of reflex amplitudes that was once thought to be a reduced presynaptic inhibition?
So actually cutaneous fibers are, for example, I wonder if this is still sharing, correct?
Yeah, perfect.
So where are we? Here we go. So as we see here, the 1A afferent here is in gray, is contacting the interneurons, which then contact the branch point. And this is the classical pad story. Cutaneous afferents are actually also an area that can activate this circuit and create, as you say, a reduced presynaptic inhibition. What we think is actually this facilitation, this increase in conduction through the previously quiescent branches to increase the postsynaptic response because there's just more transmitter across all the branches. So I would say that this helps explain that, yeah. I could have added other little circles here to show, other, central nervous system activations on this circuit, but I wanted to try to keep it a little simple. But cutaneous afferents are definitely ones that produce this tonic pad that we see this facilitation acting. Yeah, for sure.
Yeah, it could definitely get complicated very quickly.
Yeah.
All right. Thank you, Gregory, for those great questions. All right, we'll hang out here for about another half minute just to let people finish typing their questions. Otherwise, we'll move on. I think we're going to move on in just a sec. I just want to mention that Gregory was saying that this seems to be a very intriguing line of work. First saw it at NeuroHike. Oh, cool. And very much look forward to following the progression. Yeah, I feel like we're pretty lucky to be able to have the NeuroMatch conference. Otherwise, we would just all be sitting in a vacuum. But yeah, it's definitely good to be able to see these things progress. All right, so I think we'll go next up to Carolyn Rubin at the Burke Medical Research Institute. She'll be talking about microsurgical lesions identify segmentally distinct responses of exotomized corticospinal neurons. All right, go ahead, Carolyn.
Okay. Okay, hello everyone. So thank you for giving me the chance to talk about my work. So I'm Carolyn Rubin. I'm also from Sony Lab in Burke Neurological Institute. I'm gonna talk a little bit about how I'm using microsurgical lesions to study cortical spinal tract regeneration in development. So corticospinal tract, as already previously introduced a little bit, so it's formed by the neurons in the cortex and their axons that extend to the spinal cord. When spinal cord injury happens in adulthood, then this tract is not able to regenerate. So since this tract is needed for voluntary motor control, the regeneration of corticospinal tract remains a key focus for recovery after spinal cord injury. So it does remain well established that intrinsic regenerative ability of neurons declines with age. So there has been extensive research on identifying what's behind this failure in the adulthood. So they found out that in adulthood, the combination of extrinsic factors and intrinsic factors are responsible for the regeneration failure. So extrinsic factors are at the injury site and are mainly inhibitory for axonal growth. These are, for example, glial scar formation, inflammation, cell death, inhibitory proteoglycans, and myelin-associated inhibitors. These are known, most of these processes are known to become more prevalent with age. So at the corticospinal neuron level, there are different growth-associated genes and transcriptional regulators that control the axon regeneration. So in adulthood, these developmental growth genes are down-regulated. So despite the significant work, we still don't really know how and when actually in this developmental period, this regeneration ability gets lost. So it's also because the current models that are there for neonatal spinal cord injury are very severe and cause massive tissue damage. So in this, so for example, this is one of the conventionally used model, that's spinal cord injury compression in neonates. And as you can see already from the whole mount images, the tissue damage is very severe. And because of that, it's difficult, you're not able to distinguish whether the axons are not able to grow because of they have lost the intrinsic ability to grow, or it's because of these extrinsic factors at the injury site. So therefore, to be able to actually answer the question when and how does the regenerative ability get lost, we needed to firstly come up with a new approach that would leave the spinal environment relatively intact while still precisely disrupting the CST. So for that, we actually established a novel microsurgical approach to transect the developing CST in the neonatal spinal cord. So in our model, we are using a beveled micropipette that here you can see inserted into the XV1 neonatal spinal cord for the size comparison. So this pipette is then further attached to a high frequency vibrating device, and the injury is done under ultrasound guided backscatter microscopy. So here's just a short video clip showing how, the injury process. So to firstly orient you, the animal here, the mouse pup is on the side. With the dotted line, you can see the skin. Here in white, you see this, the spinal vertebrae, and this here is the spinal cord and the cortic spinal tract. So in the red here will be the micropipette. So here you'll see the micropipette inserted into the spinal cord, and then the vibration is turned on to cause the injury. And the wavy movement is just the artifact from the ultrasound. So as I showed before, that conventional models result in this massive tissue disruption that you can already see in whole mount. Then in contrast, after our microsurgical injury, there's barely any visible damage to the spinal cord. And overall, it also results in minimal glial scarring, inflammation, and it leaves the spinal environment largely unperturbed while precisely still guiding the CST. So using this microsurgical injury approach, we studied the cortic spinal regenerative ability at different spinal segments at the neonatal ages when the CST tract is still growing. So firstly, we did the injury at cervical C2 level at B4 time point. So what we found there is that this CST axons totally failed to grow across the lesion site at this age. So then we also went to the injury at the same time point at B4. We injured the axons at T11, thoracic T11 level segment, where then the injury site is closer to the growing CST tips. And what we saw is, in contrast to the previous one, that the axons are actually able to grow through the injury site. So next, we actually went a little further in the development to the P7 age, when most of the tract has grown further. And we saw again that actually the tract is still able to grow through the injury site. And in this T11 segment, actually, we saw this regenerative ability. We started seeing the loss of it at P14. So here's also just to quantify this by showing the amount of axons that make it below the lesion site to the lumbar cord from the cross sections, and you can see clearly that there are no axons making to the lumbar cord after the cervical injury down there after P4 and P7 low thoracic injury, and the decline starts at P14 for a low thoracic injury. So then the main conclusion basically that I wanted to convey today here is that the regenerative ability of CST is lost earlier than previously thought. It's already lost at least by P4 in cervical segment, and the loss of this ability is not uniform across the entire length of the axon. So next, we do actually have some of the some mechanisms that we want to study that might be behind this phenomenon that we are seeing. So to kind of maybe facilitate the questions in the discussion session, I wanted to just bring up some of it and that we're gonna study next. So first, we do think that it might be because of some CSN intrinsic factors. So for example, there might have been a developmental switch in a growth program after the axons have passed the cervical cord. So as previously maybe mentioned already by Julia, that there are different molecular programs kind of targeting the axons either the cervical or thoracolumbar cord. So if in the case that the growth program has already turned from cervical to thoracic growth, then maybe that's why after the injured cervical, the axons don't know how to grow anymore. Other option might be that also after the injury, there is just different injury response that gets activated depending on the injury level. Also, it might be still, we don't exclude that it's somehow CSN extrinsic environment, even though in our model, we try to minimize the effect to the environment. And we don't see much of the glial scarring and inflammation, but still it is possible that either regionally distinct astrocytes, myelination, or maybe microglia can somehow affect the CSN axon growth differently. So before we get to the questions, also a quick acknowledgement for my PI Vibusani and all the lab members, our collaborators, and also our funding. So now I can leave this on for maybe facilitating some of the discussion.
That's an excellent idea. I appreciate this slide. Thank you for that excellent talk. All of these have been really great talks, you guys. I just wanted to mention a comment from Kelvin Jones, just saying that this looks like a really nicely controlled injury. So great job there.
Thank you.
All right. So we will wait again for people to send in their questions via the Q&A box or through the chat. And as usual, feel free to raise your hand and we'll bring you on. We can make this segment be specific for Carolyn. And then after a little bit, I'll just open up to questions for any of the panelists, or people can just come in and have a conversation with the panelists. All right, so we have a question from Kelvin Jones. Where was the injury relative to the transition zones, the Julia's transition zones?
So the cervical injury, the higher one at C2, so it's below the brainstem cervical transition zone, but it's before the next cervical thoracic transition zone. So yeah, so at that point we should be injuring the axons that were heading to cervical or thoracolumbar, all of them.
It's also maybe worth noting, highlighting Carolyn's work again, that she did the same experiments also with a marker only looking at the thoraco projecting neurons, and she sees the same issues. So it's not a subpopulation question, but the question of where the axons are actually transected.
Yes, exactly.
That's great to know. Thank you both. Thank you for clarifying that. All right, we have a question from Gregory, who's just saying, excuse some ignorance about this area. Don't worry, I feel similarly. But I am curious if there's any way to assess differences between ascending and descending tracts in their ability to cross the injury site.
We have not looked at the ascending tracts, but there are stainings that could be done for the sensory tracts to also see if they cross the lesion site or not. So in our model, we don't. We are saying that we're looking at CSD and we're damaging mainly CSD, but we are probably still damaging some ascending tracts, the same dorsal funiculus. So we have not looked at if they are able to grow or not. But this is something we definitely are actually planning to look at.
Yeah, that would be very interesting. Another question from Kelvin. If you injure at that level but have cervical CSN express CRIM1, do they grow past the injury?
I don't think this is an experiment that has been done.
Yes, we have not done. We have done the opposite way. No, we've actually, so we have done, not CRIM1, cerebellum one that is somehow similar, one of the molecules that neurons, and we have actually tried to overexpress this after our T11 injury. And at the time points at P14 when we see already that they don't grow normally. And after we've overexpressed the cerebellum one, we have, our data is still not very conclusive. I don't think there is a big effect, but that was our idea. Maybe it will be a combination of different genes to get them there. But at the moment, it seems it doesn't actually make it. But we haven't tried the CRIM1.
But to also add to that, we don't think that CRIM1 is necessarily involved in the regeneration capacity of the developmental neurons. So I personally wouldn't expect any effect on if we overexpress CRIM1 in the cervical projecting population and then injure.
Yeah. Because also, I mean, as said, the population that I was showing now that we injure at the cervical level, it is mixed population of the ones that go to cervical or thoracolumbar, but the growth stops for all of them. So even if you try to make cervical to go longer, at that level, it was even stopping the long growing ones. So in that sense, also, it's unlikely that it will make a difference for that. But good question.
Yes, thank you. That was a really good question. Your responses were very illuminating. Thank you both very much. In general, thank you all for your questions. They've been really stimulating, and I've appreciated hearing the discussion around these. So feel free to keep those questions coming in, or come on screen, or send some in the chat. And just so everyone knows, we'll be hanging out for about a couple more minutes before the session is over. So I'll just say, feel free to send questions for any of the panelists at this point. We're open to any of it. Or if you want to come in, you don't have to necessarily have a question per se, but you can just have a conversation with our panelists if you'd like to come on screen. All right, I think we'll just hang out for another half minute or so, and then we'll wrap up. So I'll just wait for anybody to type any final questions if they need to. In the meantime, I would just like to say thank you again to all the panelists. Thank you for your fantastic presentations. Thank you for the very illuminating discussion. So it's really excellent to learn a lot from all of you. All right, so I think that's a wrap. I think we'll say goodbye at this point. Thanks everyone for tuning in, and thanks for participating. Yeah. We're just going to get started now. Today we'll be talking about the role of neurotransmitter switching in disease and behavior. We have an excellent lineup of panelists today for you. And we're going to start with Shweta Gudavarti, who's at UCSD, who will be talking about transmitter switching contributes to 2 environmental models of neurodevelopmental disorders. All right, go for it, Shweta.
Thank you, Stephanie. So, thank you all for joining, and hello to people joining us from across different time zones. So as Stephanie mentioned, I'll be talking today about my work, which is examining the role of transmitter switching and how it may contribute to 2 different environmental models of neurodevelopmental disorders. So we're all familiar with the classical forms of plasticity, which involves change in synaptic strength or change in the number of synapses or modulating the extent of excitability of the synapse. But our lab has been instrumental in identifying a novel form of neuronal plasticity called neurotransmitter switching. And this has been demonstrated both in developing and adult nervous system. And it basically involves the loss of a neurotransmitter and gain of another neurotransmitter. So there are usually switch partners. And this occurs in response to sustained stimulation, which is we alter the baseline activity levels of the circuit, which can either be brought about by either artificial means, by misexpressing ion channels, or by using natural stimuli, which can be the sensory or motor in nature. And which one will be coming up next, we'll talk more about this aspect. An important part of this neurotransmitter switching is that the change in the presynaptic neurotransmitter is accompanied by a corresponding match of the postsynaptic receptor, so thereby resulting in a functional synapse. And the switch is often associated with a change in the sign of the synapse, so either from excitatory to inhibitory or vice versa. And most importantly, this molecular level neurotransmitter change is associated with change in the behavior of the animal. So our lab for a long time has looked at neurotransmitter switching in the healthy brain and tried and examined which regions are susceptible to switching and what stimuli can induce it. But I was interested in examining neurotransmitter switching in the context of disease, and specifically autism spectrum disorders, which are characterized by a triad of deficits in 3 major domains: communication, social function, and motor behavior. At the molecular level, both animal models as well as patients we have been shown to exhibit deficits or disruptions in GABAergic and glutamatergic signaling. And eventually, a common phenotype that emerges across different types of ASDs is disruption of the excitation-inhibition balance. And this excitation-inhibition balance could either result in neurotransmitter switching, as we saw in the previous slide, or it could be a result of aberrant neurotransmitter switching. So in order to examine neurotransmitter switching in the ASD model, we decided to look at the prefrontal cortex region of the brain. And if we look at the functions that the prefrontal cortex performs, we see that it nicely maps on to these deficits that are associated with ASD. And importantly, PFC has a protracted period of postnatal development. That is, although initially the initial specification and connectivity is driven by intrinsic factors and genes, environmental factors play a large role in the postnatal development of prefrontal cortex, thereby making it an interesting and a key candidate region to examine ASD in the context of neurotransmitter switching. So this is the broad aim of the work, which is to examine what role neurotransmitter switching plays in the development of PFC, in maintenance of cortical excitation-inhibition balance, and in the emergence of ASD. And to examine this phenomena, we are using 2 different environmental models of autism, unlike the more popular genetic models. So it is known that in pregnant females, if they're treated with antiepileptics like valproic acid, or if the female launches a strong immune reaction, there is a high risk of her carrying a child with autism spectrum disorders. And we are mimicking these conditions in our mouse models by injecting a pregnant female at embryonic day 12.5 with either VPA or poly-IC to mimic the maternal immune activation. And then we follow the fate of the progeny at various time points. And of course, we are interested in looking at the medial prefrontal cortex. So the first question we wanted to address is how and if do developmental manipulation alter neurotransmitter identity in the PFC. And we find that as early as postnatal day 10, we observe a loss of GAD67 positive cells, which is a marker for GABAergic interneurons, and gain of VGLUT1 positive cells, which is a marker for glutamatergic neurons. And here are the stereological estimates for these. And the change in the number of GAD and VGLUT1 is specific because we looked at another marker like NOS1, and we do not find any difference in their numbers. Now, the fact that we see loss of about 5,000 GAD67 positive cells, and there's an equal gain of about 5,000 VGLUT1 glutamatergic cells, and in the absence of any change in the total number of neurons or any change in the cell death, specifically within the medial prefrontal cortex, suggests that these two might actually be switching partners. And this is region specific because we do not see any difference in our cell counts in the lateral prefrontal cortex. This change sustains till up to P21, that is postnatal day 21. So we continue to see decreased GAD67 and increased VGLUT1 numbers. However, by P30, this change that we see in transmitter identity disappears. And of course, it's not seen in the adult animal. So to summarize this part, we see an increase in VGLUT1 and a decrease in equal number of GAD67 positive cells, specifically in the medial prefrontal cortex of our environmental models of autism. And there's a specific window period between P10 and P21 where we see this change, and however, it is not sustained into adulthood. So now the question arises whether the switch that we are seeing, does it occur within the same neuron, or are these two different populations of neurons that are affected? So to address this, we firstly looked at all the various subtypes of GABAergic interneurons that are present in the medial prefrontal cortex. And we identified that parvalbumin and cholecystokinin subtypes are able to account for the loss of 5,000 GAD67 cells that we see in our EMA models. So what we did next is we crossed PV Cre lines with CCK Cre lines, and we treated the pregnant female with poly-IC. And postnatally, the PV CCK Cre animals were injected with a control GFP virus. This allowed us to mark all our PV and CCK cells green. And when we did our poly-IC treatment, we found that there were GFP positive, that is PVCCK cells, that were also VGLUT1 positive, as we can see here. And there are far more number of VGLUT1 GFP positive cells in the poly-IC condition as opposed to the controls. So this indicates that PV and CCK cells contribute to the loss of GAD67 that we see in the AMA mice, and the poly-IC treatment is able to induce VGLUT1 expression within these PV and CCK neurons. We are further validating this aspect using RNA-scope now. So now the most important part is the causal aspect, whether neurotransmitter switching is required for appearance of autism-like behaviors. And for this, again, we resorted to our PV Cre CCK Cre cross, but now following poly-IC treatment of the pregnant female at postnatal day 10, the animal was injected with GAD1, which encodes for GAD67. And so thereby we are able to restore GAD67 expression in these PVCCK neurons that are otherwise losing GAD67. Now the question is, what happens at the molecular level? So the good part, and this was expected at the GAD67 level, is restoring GAD67 expression even in the poly-IC animals is able to prevent the loss of GAD67 positive cells. But interestingly, it is also able to rescue or prevent the increase of VGLUT1 positive cells following poly-IC treatment. So we see a molecular level rescue. What happens at the behavioral level? So we are examining all these three domains of functional deficits, but today I'll be talking about social function and motor behavior specifically. So for motor behavior, we are looking at stereotypy assays by using marble bearing or self-grooming assays. And we find that although both males and females show deficits in marble bearing assay, only males show increased stereotypical behavior in the self-grooming assay. And only males exhibit disrupted social functioning as seen through the social interaction assay, while females are not affected. However, when we restore GAD67 in the PV and CCK neurons, we are able to rescue behavior across all these three assays that we see. So in marble bearing, in in self-grooming, as well as in social interaction assays. So to summarize this last part, so we see that there is a gender dependence in terms of the emergence of autism-like behavior in our EMA animals, and overriding the GABAergic to glutamatergic switch in PV and CCK neurons as early as P10 is able to rescue behavior in the adult animal. So I'd like to thank my supervisor, Nick, for his amazing mentorship, and all my lab members for the wonderful discussions, and of course our funding agencies. And I'd be happy to take any questions that you may have.
Thank you for that excellent talk, Shweta. And yeah, I encourage any questions via the Q&A or the chat box. And we have one right off the bat. This one comes from Shruti Muralidhar. I'm sorry if I butcher any names. Hi, Shweta. Have you seen switching of synaptic identity along with cellular identity?
Right. Hi, Shruti. Nice to get in touch with you here. So we haven't looked at the synaptic level, but that is something that we are interested in. And we have experiments ongoing right now to examine what happens at the synaptic level. And I hope that Marta, who will be presenting as well in this panel, will be able to address this question.
Good point. And Shruti says, great talk. All right, so another one from Sridevi Venkatasan. Sorry, again, pronunciation. Do you predict a role for transiently cholinergic neurons in the neurotransmitter switching and emergence of ASD phenotypes?
Oh, that's an interesting question, Sridevi. We haven't looked at the cholinergic neurons in the prefrontal cortex because from our literature survey, we realized that, of course, GABA and glutamate are the predominant population, but this is something that is an interesting aspect that you suggest.
Yeah, it definitely could be. All right, another one from Priscilla Ambrosi. Is the switching happening in the same cells, or is there a different population of cells that starts expressing more V-glut?
Yeah, great question. As of now, from the data that we have with our poly-IC treated animals that were injected with GFP to mark the PV and CCK neurons, and then we examined V-glut expression, it looks like it is happening within the same cells. So it is the PV cells or CCK cells that are gaining V-glut1. Of course, the question is always whether they have some baseline level of V-glut1, which we are unable to detect with our current techniques. For that, we are using RNA-scope assay, which will be able to better give us, which will be a more sensitive technique to address this question. So whether there's a change in expression levels of V-glut, we'll know through RNA-scope.
That's a good point. All right, so we have a question from YouTube from Kala Alganaras, who starts with saying that this was a really great talk. Did you check if the developing synapses were already functional at the same developmental time that the NT switch occurred?
Great question. We are in the process of testing this, and stay tuned. Hopefully, we'll have some interesting results for you.
Definitely. We'll look forward to that. All right. We're just going to wait a sec for people to type any other questions. Yeah, we'll just hang out for a minute.
Sure.
All right. So I think we'll move on. If anybody has more questions for Shweta, you can feel free to send that along and she can type a response. But we're moving on to our next speaker, which is Huichuan Li. from the UCSD talking about neurotransmitter switches regulate motor learning and stress response. Go for it, which one?
Thank you so much, Stephanie, for the introduction. Hi, everyone. I'm Hui Chun from UCSD, and today I'm happy to share you two stories about neurotransmitter switching in motor learning and stress response. It has been thought like the neurotransmitter identity is fixed throughout a neuron's life, but this dogma has been challenged by neurotransmitter switching that in response to environmental change, neurons can lose their original transmitters and gain a new one, even in adult brain. And the switch usually occurs between excitatory and inhibitory neurons to strengthen its impact on the neural circuits. And as our brilliant audience have already brought out the question that in the field, the two key questions are firstly, whether the gain of one transmitter and loss of the other occur in the same neurons or not. The other question is, does the switch directly lead to behavior change? To tackle these two questions, I used a healthy mouse model where I found that voluntary wheel running promotes motor skill learning. That you can see here is a happily running mouse on the running wheel, and this is a control mouse that has no wheel to run on. After 1 week, I found that for the runner mice, their motor skill learning is enhanced as they learned faster for a road test, which is a rolling road, and the mice learned faster on how to stay on it at a high speed. And also the mice learned faster on how to cross a very thin beam. You can see for both the tasks, the runners, the learning slopes for runners is steeper than control mice, indicating their better learning. And then I searched in the nuclei that regulate motor skill learning and found in peduncular nucleus, which is shown here in green in the chrono section, that there were more GABAergic neurons and less cholinergic neurons in runners compared to controls. To understand whether the gain of GABA and the loss of acetylcholine occur in the same neurons or not, I injected adeno-associated virus expressing career-dependent mRuby2 fluorescence protein into chat-carrier mice. Chat is the enzyme that synthesizes acetylcholine, so it is a marker for cholinergic neurons. And you can imagine that the after DNA recombinase induced by carrier and AAV expression of mRuby2, the chat neurons can be permanently labeled by these fluorescence proteins. And after running, if the loss of chat and the gain of GABA occur in the same neurons, these magenta neurons, these magenta cells will lose their blue and gain some green and turn into yellow. Otherwise, if the gain of GABA occurs in some other populations, I would not be able to see those yellow cells. Indeed, in the control mice, I saw most of the cells are magenta, while in runners, interestingly, there were yellow cells showing up. I then grouped the neurons into 4 categories: classical neurons that express only chat, switch neurons that express only GABA, neither neurons that have neither of them, and co-expressing neurons that have both. And then found that in the control mice, the majority of the cells are classical neurons that express only chat. And this population is reduced in the runner, which you see the difference between in the pink. And in turn, there is a significant increase of the switched in the number of the switched neurons that have only GABA. So this demonstrates that the gain of GABA and the loss of chat occur in the same populations of neurons. Then, to study the behavioral relevance, I constructed an AAV that expresses career-dependent chat to overexpress chat in these cholinergic neurons you can imagine that even when the endogenous chat is lost during running, this exogenous chat expressed by the AAV can maintain the chat level and therefore the acetylcholine level. As a control, I use an AAV that expresses only mRuby2 fluorescence protein, the reporter, but no chat. And indeed, the expression of the mRuby2 alone did not affect the benefit of running, as the runners still do better than controls for both tasks. However, when chat is overexpressed, you see those runners did just like control animals that have never been run on a running wheel, and significantly less well than the runners injected with only a reporter virus. So this shows that overriding the loss of acetylcholine prevents the running enhanced motor skill learning. How about GABA? I then injected acetylcholine for GAD1, which is the transcript encoding the enzyme synthesizing GABA, and see the same thing that when GAD1 is interfered, the runner mice did just like control mice and not as good as the runner mice who were injected with a scrambled acetylcholine that targeted nothing. So this overall tells us that the switch between acetylcholine to GABA induced by running is essential for enhanced motor skill learning, and the switch occurs in same neurons. This is about healthy brain gets healthier. How about disease? We then got interested in how neurotransmitter switching plays a role in stress response. As you know, the fear is an essential emotion that helps us to make decisions, judge risks. And just like now we're having a virtual conference instead of gathering in a room, this is an example of how good decisions are made by the right amount of fear. Being fearless is not good for one's survival. But when fear is sustained and disproportional to the risk, it can lead to fear disorders, such as post-traumatic stress disorder, where patients suffer from their traumatic stress, and then even when the threat is not present, they're still being fearful to a noxious environment and be overreactive to mild stress and behave passively. So to understand whether neurotransmitter switching plays a role, we use a mouse model where we treated mice with intense food shock. The shock was acute, but the effect was long-lasting, that 2 weeks after the shock, when we put the mice into an environment that they have never been exposed to before, the control mice were exploring the chamber, the environment freely, while the shocked mice showed passive behavior, that they were mostly freezing in the chamber and did not explore as much as the control mice did. Here shows you their movement tracks. As in 10 minutes, you can see the control mice moves aloud and the food shock mice just stay there. And we quantified their exploration as their horizontal exploration, which is the distance traveled in the chamber, and their vertical exploration, which is their rearing counts in the chamber, the climb on the wall. And both of them decreased for the food shocked group. At the same time, the shocked mice showed a dramatic increasing of freezing time in the chamber. So this tells us that food shock stress reduces active and promotes passive behavior. We then search for transmitter switching and look at the dorsal raphe nucleus, because it is the largest serotonergic nucleus, and serotonin is the most common target to treat fear disorders. Interestingly, dorsal raphe nucleus has subregions, and its dorsal half, including lateral wings dorsal raphe and dorsal raphe, project to fear centers, including lateral habenula, central amygdala, and paraventricular zones of hypothalamus. And the ventral part project to the cortex. We then looked at the dorsal part of the dorsal raphe, and I counted the serotonergic neurons there, but found no difference between the control and the food shot group in both regions. Fortunately, the game was not over, that recent studies have found glutamatergic neurons and GABAergic neurons also play a role in coping strategies. So I sent 3 of them together and found consistently as what reported before, that the serotonergic neurons here co-express glutamatergic markers and GABAergic markers. But what is new is that I found in the shocked group, there were more neurons that express serotonin as well as GATK7, which is the enzyme co-expressing enzyme to synthesize GABA, and there were less neurons that co-express serotonin and VGlu3, which is the transporter for glutamate. This is the summary of my counting, that there were more serotonergic neurons that contain GATK7 and less of them contain VGlu3. And there is also a very small population of neurons have 3 of them. And this population gets even smaller in the shocked group. And the total number of the neurons combining these 3 groups did not change, which indicates that the new serotonergic neurons co-expressing GATK7 may come from the neurons were previously expressing VGlu3. So as a summary, I found that food shock induces passive behavior, but there was no change in the serotonergic neurons in the dorsal raphe, but there was a difference in the neurons co-expressing GATK7 and co-expressing VGlut3. So now we're testing whether the switch occur at the same cells or not, and we're inventing the transmitter switch to see how it affects behavior, and we're also looking at their circuit and molecular mechanisms. Last but not least, I would like to thank my PI and exhibitor, my brilliant students, Uji, Lily, Rahul, Kay, and Cal, and all of my collaborators and my funding resource. Thank you so much for your attention.
Thank you so much for that excellent talk, Huichuan.
Thank you.
All right, so any questions, feel free to submit them to the Q&A box. And again, as usual, I'll also be monitoring the chat. And here we are. We'll start with Priscilla Ambrosi, who asks, are the changes in chat and GAD expression long-lasting, specifically in the running wheel experiment?
That's a terrific question. So we have looked at it. It's reversed, both the change of chat and the GAD reversed 1 week after we removed the running wheel. So it's easily reversed. Probably like you may be curious about the second story as well, but for the second one, when we use food shock stress, it's the switch is very stable, that at least after 1 month, the switch is still there. So we think that probably it's the intensity of the stress, like for running, it's not that intense. So the transmitter switch goes back very soon. But for food shock stress, it causes a long-term, long-lasting changes in the expression. Thank you very much.
That makes sense. Thank you. All right, and Priscilla follows up with saying that that seems really cool. And she says, wow. What do you think is the role of switching then?
What do you think is the role of the switching then? So are you asking about the second story? For the first story about the role of switching, we have found it's essential for motor skill learning that is enhanced by running. And for the second question, we have found a nice correlation between the switch and the behavior change. So when the mice have more GABAergic neurons in their lateral wings of dorsal raphe, their behavior are more passive compared to the neurons that have more glutamatergic neurons and less GABAergic neurons. So we think they play a role in regulating the passive and active coping, and we're now testing it. Thank you.
Thank you for that. And we're going to move on to the next point, but there's another question for you by Joseph D'Souza. You can respond to that in the chat. And I just want to follow up that he says that this is amazing. So just for you to know that.
Thank you so much.
So, of course. And now we're going to switch to Marta Portelli. She's coming to us from the UCSD and going to talk about glutamate to GABA neurotransmitter switching induced by drugs of abuse and its impact on behavior. Go ahead, Marta.
Yeah, I need, it don't allow me to share the screen because
sorry.
No problem.
Sorry, I was jumping to answer the questions.
No worries.
Okay, so good afternoon, everybody, and thanks for being here. And I would like to talk to you today about a form of neuroplasticity that is induced by exposure to some drugs of abuse, and that seems to play a very important role in the modulation of the behavioral alteration that exposure to drug cause. So I want you to keep in mind that when I'm talking about drug of abuse, I'm not referring to any particular singular molecules, but instead to a class of different compounds that have different molecular structure, different targets in the brain, different effects, but all of them share the ability of inducing plastic changes in the brain that turn out to alter the functionality of brain circuits and of neurons, leading to the appearance of changes in behavior, and sometimes this led to the development of addiction. So it is important to know what, despite the single and molecule specific effect of a drug, what are these shared common plastic changes that exposure to drug cause. And specifically, I will focus on one form of neuroplasticity, that is neurotransmitter switching. You already heard about it. This is an environmentally induced form of neuroplasticity that occurs when there is a sustained environmental stimulus that causes changes in the activity of a brain region. And this induces some neurons to lose the transmitter they were expressing and gain the expression of a different one. And this switch generally goes from excitatory toward an inhibitory transmitters or vice versa, and goes along with concomitant and matching changes in the types of receptors that are expressed at the postsynaptic terminal. And this means that we do not only have a change in the transmitter, but a cell that was previously sending an excitatory signal is now sending the opposite signal to the postsynaptic terminal. And this shows clearly how this type of switch can impact the functionality of a circuit and may lead to consequences in behavior. So here we want to know whether different drugs can induce a similar neurotransmitter switching, and if yes, whether this switch have or not something to do with the appearance of drug-induced behavioral alterations. And to answer this question, I've been using two different drugs, two different compounds, penciclidine, PCP, or methamphetamine. So these are two completely different drugs, two completely different molecules. But for example, it has been reported that after repeated exposure, they both cause similar types of cognitive deficit. Not only, it is reported many times in literature that both can cause alteration in the activity of the prelimbic cortex, which is a region that is important and crucial for cognition. And it seems that these affect mainly glutamatergic neurons. So I start to wonder, is a switch going on here in the prelimbic cortex, maybe glutamatergic neurons? So I use this mouse over here, vglut1 cream cherry, to permanently label glutamatergic neurons with mcherry. So even if the neurons change phenotype and expression pattern, they will still be marked by mcherry. So I can treat the mice with the drugs or with PCP as a control, and then see whether in these mcherry neurons an inhibitory transmitter appears. For example, GABA. But let's see the results. So interestingly enough, we immediately notice that there are some neurons in the prelimbic cortex that are already co-expressed, even in saline treated mice, mcherry and GABA. However, in mice that have been treated with PCP for 10 days, the number of those neurons appear to be drastically increased, here with the green arrows. And indeed, when I counted the number of mcherry positive and GABA positive neurons and compare it in saline versus PCP treated mice, as you can see here, I noticed that there is a doubling of the number of these type of neurons after treatment with PCP. And how it is that now, after a drug, there are neurons that start to synthesize GABA. But we know that GABA is synthesized starting from glutamate thanks to the activity of GAT67, the GABA synthetic enzyme. So we stained for GAT67, and we saw exactly that the same, so that PCP treatment caused approximately 600 cells, all these neurons here, to gain the expression of GAT67. So there is an acquisition of a GABAergic phenotype here, right? What happened to the glutamatergic phenotype? So to answer this question, I'm now going to focus on PCP treated mice specifically, and I will measure in different cells the expression level of the vesicular glutamate transporter 1, vglut1, that is necessary for the loading of glutamate into synaptic vesicles. So how do we do this measurement? But we use mcherry to draw the perimeters of different types of cells, for example, glutamatergic, and then we measure the percentage of the area of the cell that is occupied by white panta. So I did that for cells that, for example, are co-expressing the GABA synthetic enzyme and mcherry. So these are allegedly our switching neurons. And then we can compare it with cells, for example, that instead, oh, sorry, slide too quick, that instead express only mcherry, and so they are purely glutamatergic. And what you can see is that there is a drastic drop in the expression level of the vesicular glutamate transporters in neurons that have acquired a GABAergic phenotype. So there is an impairment in the ability of these neurons over here to release glutamate. Now, we are also curious to see whether at the same time they start to express some new transmitter, so some new transporters. So we decided to look for the vesicular GABA transporter, VGAT, here in green. And what we saw is that this time in VGAT, that in our switching neurons, the expression level of VGAT is exactly the same as in only GABAergic cells. So altogether, this shows that we have a switch, right, from glutamate to GABA. But is that something that can be caused only by PCP? Or is this a more broad effect? Can it be caused by other drugs? So we look at methamphetamine. And interestingly enough, we again saw that methamphetamine, despite through different mechanisms, was able to induce an increase in the number of glutamatergic neurons in the prelimbic cortex that are now expressing GABA and the GABA synthetic enzyme. Not only, but when we look at those neurons that have acquired the GABAergic phenotype, we can see that they show a decrease in the expression level of the vesicular glutamate transporter 1, while at the same time, they start to express as purely GABAergic neurons a normal amount of the vesicular GABA transporter. So this brings us to the first set of conclusions, right? We have found two different drugs that induce a subset of neurons in the prelimbic cortex to switch from glutamate to GABA. But now the big question, does it have anything to do with the appearance of drug-induced behavioral alterations, for example, cognitive deficit? So this is what we want to answer now. And how do we do that? But the idea now is to prevent glutamatergic neurons in this area for being able to start to produce GABA, even if they want to. And we do that by injecting a small interference RNA for GAD1, SH-GAD1, in order to prevent the expression of the GABA synthetic enzyme. So then, or we can use, of course, a control scramble virus. And then after waiting for the virus to be expressed, we treat the mice with PCP or saline, and then we subject them to a battery of cognitive tests, such as the novel object recognition test and the spontaneous alternation task. So let's have a look at the results. Unfortunately, for now, I only have the results for PCP. We will have to wait for math. The experiments are ongoing. So if we look at the novel object recognition test, that is a test that measures memory retention through the recognition index, we can see here that in mice that have been treated with the control virus, so in which the switch can occur eventually, and receive either saline or PCP, treatment with PCP induces a significant reduction in memory retention. However, what we see when mice have been treated with PCP but cannot undergo the switch because they've also received the small interference GAD1 RNA virus, this time the memory retention is completely rescued. Similarly, when we perform the spontaneous alternation task, the measure the performance in working memory by alternation percentage, we can see that PCP, when the switch can occur, induce a decrease in working memory. However, this is almost completely rescued when PCP is accompanied by the impossibility of cell to switch because of this virus. So this was a very exciting result for us because it tells us that this specific glutamate to GABA switch caused by PCP, and I hope to be able soon to bring you the data also for math, is actually related to the appearance of the behavioral alteration in a cause-effect way. Indeed, when we treat with PCP, but we don't have the switch, we have an almost complete rescue of all the deficit, at least in the test that we have done. So I want to conclude by thanking you all for the attention, thanking all the amazing members of my lab, my PI, Nick Spitzer. I hope you have questions, and if you want, you can also write to me later at this address over here.
All right, thank you so much, Martha. Okay, we're going to start with a question from Joseph D'Souza. Could this receptor switching happen in a specific brain location, like in one nucleus, but not in an adjacent nucleus? As in, is it spatially localized? He adds that he studies people with Parkinson's dancing. Something about seeing a synapse dance document. Could this be happening in Parkinson's?
So I don't know specifically about Parkinson, but and I'm not sure what you mean here, and eventually you can clarify that to me. If you talk about nucleus, meaning different parts of the brain, different subregions, or because I think that it can definitely occur in specific part, but not in other. Also, the cells that are involved are different. Something that we still don't know, for example, is whether if you have a switch, this is involved. So it happened, for example, in all D terminals. Yeah, so I see that the question was about the nuclei. And yeah, for example, in my case, I don't know for Parkinson, but I can see that this happens in the prelimbic cortex. If I look at the infralimbic or at the cingulate cortex, I don't see it. So it seems to be particular specific.
Thank you so much for clarifying that. Yes, so it could be very specific to certain areas.
Thanks for the question and the clarification.
Yes, thank you. And thank you, Joseph, for all your great questions so far. All right, so I'll be monitoring the Q&A and the chat for anything else that might come in. We're approaching the end of the session, but we'd love to still stick around and answer any question that comes in. All right, and we have one from Carla Sorry, I keep butchering the names. Acknowledgements. Awesome talk. Do you know if the switching neurons in the prelimbic cortex were long projection neurons or local circuit neurons? If it's long range, do you know if the targets of those new, do you know the targets of the new GABAergic neurons?
So thank you so much for the question. I'm glad you asked because I'm now doing some experiment to figure it out. So it seems that to be long projecting neurons, and it seems, I just have preliminary data, so everything can still happen. But for example, it seems that some of them project to the nucleus accumbens. And for the rest, I have different targets to validate that seems to be reached, but yeah, I need to do more analysis.
Excellent, excellent point. And thank you for those questions, Carla. All right, I know that we're going to be kicked out in just a few minutes, so I want to thank all the panelists. You guys gave excellent talks today. I see Joseph D'Souza just chimed in saying he learned a lot and this was a great session, and absolutely, I completely agree. Thank you all very much for making this such an engaging and wonderful session.
Thank you for this possibility and for everybody who joined and asked amazing questions.
Absolutely. I also see a thank you from Sylvia. All right, I think we'll head out then, unless there are any final little questions that trail in before we get kicked out. Thank you, Sam. We appreciated having you here. All right, goodbye.
Thank you, Patrick and Stephanie. Thank you.
Of course. Thanks for tuning in. And thanks for participating.
Everybody.
Now broadcasting, so I see Pablo in the audience. Great.
Let's go ahead and.
Okay, so if you would promote him.
I
All right.
believe I did.
Great. And there's Pablo. Hello, Pablo. Okay, so hello, everyone. Welcome to the second day of NeuroMatch 3.0. I'm Medina Sarvastani. I'm going to be the on-camera host for this session. And we have Ami Rahana. He's the off-camera host, making sure that all of the technical things go smoothly in the background. Thank you, Ami. Just a few reminders before we get started, and we wait for a few more people to roll in. This is an interactive session. And with these interactive sessions, we're really running an experiment at NeuroMatch Academy in how to translate the in-person poster sessions to the virtual format. It's a little more of a challenge than just translating the traditional talks. So each speaker will have a 15-minute total chunk, and we have to stick pretty close to that because at 45 minutes after the hour, we have to give the room up to the next session. But within that 15 minutes, you can sort of distribute the time however you want. Typically, you give about a 5 to 10 minute, it's up to you, broad overview of your work. So this is like the overview when somebody just walks up to your poster session. And then we'll open up to the audience and importantly to the other panelists, so you can unmute your mics, and we'll have a casual Q&A between all of us here, and we'll just have to keep tight at 15 minutes. So for attendees, please post your questions in the Q&A and upvote each other's questions. Feel super free to use the chat to make comments, or clap or thumbs up emoji to ask for references and papers, methods, whatever you like. The Q&A and the chat is for everyone, and there's no threshold. You can ask clarifying questions or overview questions. Let's see what else. Great, so we have all of our panelists. So let's get started. I think all of the technical stuff is in place. Great. This session is focused on understanding cognitive control and decision-making networks in drug and alcohol addiction, and in our last talk in complex problem solving. Our first speaker is Priya Modak, who's a second-year grad student at Department of Psychological and Brain Sciences at Indiana University Bloomington. And she's going to tell us about neural bases of risky decisions involving nicotine vapor versus monetary award. And from the abstract, Priya, your task seemed really interesting. So why don't you take it away?
Thank you. So, hi everyone, I'm Priyamvada Modak, but you can call me Priya. I'm a second year graduate student. I'm working with Professor Joshua Brown in Cognitive Control Lab at Indiana University, Bloomington. I'm going to present this ongoing project in our lab. The overarching goal of this work is to understand how substance-dependent individuals make decisions about drugs. In this project, we have specifically looked at nicotine-dependent subjects. The motivation was that risky decision-making tasks are frequently employed to study the impairment in substance use disorders. Majority of the previous neuroimaging studies that follow this approach use money as the reward in their decision-making tasks. Even in the studies that provide the drug of interest as the reward, subjects receive the accumulated reward outside the scanner and at the end of the entire session. However, previous research has shown that sensitivity to monetary rewards is altered in substance use disorders. So in case of this study, we wanted to find the neural correlates of risk and reward associated with nicotine, when the nicotine worn by the subjects could be realized immediately inside the scanner with the help of an e-cigarette. Moreover, to evaluate whether the results from decisions about non-drug rewards in substance-dependent individuals can be extrapolated to infer decisions about drugs, we also wanted to compare the risk and reward representations of nicotine and money in nicotine-dependent subjects. We employed nicotine-dependent subjects who were asked to remain abstinent for 6 hours before the task. The subjects performed 2 trials on 2 separate days. One was money trial and one was nicotine trial. An MR-compatible electronic cigarette was used to deliver nicotine vapors inside the fMRI scanner. You can see the image of the e-cigarette on left of the slide. The task was to choose between 2 options. One was a gamble option and one was a sure thing option. We had 5 different gambles such that the probability of loss, expected value, and variance of the gamble outcomes were orthogonalized. This enabled us to look at the effects of these 3 things separately. Subjects performed 2 trials. In case of money trial, the reward was in terms of dollars, while in case of nicotine trial, the reward was in terms of seconds for which they could inhale nicotine vapors from the e-cigarette. We analyzed this fMRI data using the general linear modeling. The time at which subjects indicated their choice was modeled as a choice event, and the gamble characteristics were used to parametrically modulate this choice event regressor. At the time of the feedback, we had 2 separate regressors to account for instances of winning and the instances of losing the gamble. The analysis of the choice behavior revealed that when a gamble had greater expected value, then there was a greater tendency to choose the gamble. This ascertains that subjects wanted the rewards and were engaged in the task. Now moving to the results from fMRI analysis using the GLM approach. We saw the effect of probability of winning nicotine in caudate. This is not surprising because caudate has been shown to be involved in reward anticipation in previous studies. The regions around cingulate and middle inferior frontal gyrus showed effects of loss and variance of gamble outcomes in case of monetary rewards. We saw the most interesting results in the feedback phase, where there was a reversal in representation of choice feedback between money and nicotine trials. In the figure that is shown, whatever is blue corresponds to nicotine and whatever is red corresponds to money. We observed that there are distinct but overlapping regions that are representing nicotine win minus loss, but money loss minus win. So in this figure, the regions in blue show greater activation to win than loss in case of nicotine, while the regions in red show greater activation to loss than win in case of money. The same thing is shown in the plots on the right. Moreover, in the nicotine trial, winning a gamble was followed by an inhalation phase when the subjects inhaled nicotine vapors. The blue regions shown in the top left of the figure are the motor regions that activated during inhalation. We performed psychophysiological interaction analysis with these motor regions as seeds and found that the right middle temporal gyrus has increased functional connectivity with these regions during inhalation. This suggested that middle temporal gyrus might have some role to play in actual inhalation or consumption of the nicotine. So with this, we are at the end of the results. To summarize, our first objective was to find the neural correlates of risk and reward associated with nicotine in nicotine-dependent subjects. In that respect, we found that the effect of probability of winning nicotine is in caudate. The effect of loss minus win was seen in superior temporal gyrus and supramarginal gyrus. The effect of win minus loss was seen in cingulate gyrus and insula. Or another objective was to compare the risk and reward representations of nicotine and money in nicotine-dependent subjects. In that respect, we found the effect of probability of winning was not seen in reward processing areas like caudate in case of monetary rewards. We see the effect of variance of Campbell outcomes and probability of loss in cingulate in case of monetary rewards. Most importantly, we see a reversal of feedback representation in cingulate and insula representing monetary loss minus win, while nicotine win minus loss. So in conclusion, the results from our study show that expected value of the options are the strongest predictor of choice behavior in decisions about both money and nicotine in nicotine-dependent subjects. The results also suggest that there might be reduced sensitivity of reward processing areas like caudate to monetary rewards in nicotine-dependent subjects. With respect to the error reversal that we see between money and nicotine, it appears that the monetary decision-making tasks might not be the best for studying impairment in substance use disorders. Also, there are parts of cingulate and insula showing effect of nicotine win minus loss. And there is middle temporal gyrus showing increased functional connectivity with motor regions involved in nicotine inhalation. They seem important candidates for any future work investigating causal role of these regions in substance use disorders. So thanks for listening to my talk, and please feel free to ask any questions.
Great, thank you, Priya. Yeah, I thought it was really interesting with the error reversal for monetary and drug rewards. And if I got it correctly, losing money was, can you repeat that? Losing money is worse than, it's asymmetric than, yeah, can you repeat that?
Yes, sure. So what this figure is showing is there are these regions in cingulate which show greater activation to monetary loss than monetary win, while these regions show greater activation to nicotine win than nicotine loss. So it would probably be more clear if I explain these plots to you. So in this plot, we are looking at the regions which are shown in red, and the regions which are shown in red show greater activation to nicotine win and show greater activation to monetary loss. And same is the case for regions in blue.
Okay,
so if we were to talk about these regions in red and blue as one big region, then we can say that this region represents monetary loss than it represents monetary win, and this region represents like nicotine win as compared to its activation in nicotine loss.
I see. So do you think this has to do with the fact that it's money particularly, or that it's not nicotine? Why this reversal?
That's a very good question. So in this study, we have compared money and nicotine, but it's just my guess that it would probably be the case even with any other non-drug reward, like even if we provide points or say something else, say a gift card.
I see. So just not the nicotine reward. And in general, I see. And in general, is it kind of looks like the magnitude of the the money curves are similar, but I can't tell. So in general, is the negative value of losing money similar as far as activation to the value of winning the same amount of money? So in gambling behavior, are those two things equal?
Oh, so like if I understand it correctly, your question is whether the difference between loss and win in case of money greater than the difference between loss and win in case of nicotine.
Yeah, that's one version of the question. Yeah.
Okay. So from these plots, it seems like the gradient, so the difference between activation to loss and win in case of nicotine is greater, at least for this region.
Yeah.
And in this region, the gradient for money seems greater.
Let's see, that's interesting. Okay, and as far as, so you conclude from this data, and I would say convincingly, that maybe monetary rewards, maybe we shouldn't be using monetary rewards in these studies because they could give us essentially effect reversals. And something with nicotine, as you guys have done, you can, give to someone under an fMRI with some sort of clever task design. But for other drugs and other addiction studies, that's not really possible. So how do you suggest we go about it?
So in case of, say, cocaine addiction, there have been studies where cocaine has been injected inside the scanner.
Okay.
So yeah, that is another study that I know where such an effort was made to provide drug directly inside the scanner.
I see. And what's next for this work?
So we would want to, so as you said, we would want to test other drugs and other non-drug rewards, whether we see same kind of error reversal in that case.
Great. Okay, and that puts us right out of time. Thank you, Priya.
Thank you.
All right, our next speaker, and I think everything is functioning properly now, is Amanda Ankara. She's a senior, hi Amanda, in the Behavioral Genetics Laboratory at the University of Puerto Rico, and she's going to tell us about the molecular mechanisms of ethanol cognitive neuroadaptations. Amanda, the stage is yours.
Okay, first, can you all hear me well?
You're a little quiet on my end, but it's clear.
Okay, let me know if anything changes. I am speaking a little bit louder now.
Okay, yeah, that's good.
I'm going to just share my screen. Can you see?
Not yet. Yes, now I can see, looks good.
Okay. Let me just click this here really quick. It allows me. Oh no. I apologize, some technical difficulties that we're undergoing here. Okay, so I'm not sure who's watching us today, but for me it's night, so good night to all. My name is Amanda Ankara, as I cannot kind of pronounce her name, but thank you. My name is Amanda Ankara, and I'm from the University of Puerto Rico, and I will be talking to you about my project called the Molecular Mechanisms of Ethanol Neuroadaptations. And essentially, this project kind of roots from this study done by Robinson, where the purpose of the study essentially is to create, for these flies, Drosophila melanogaster fruit flies, to create an adaptation with alcohol. But essentially what they do in the study is that they subject flies to a conditioning training, where they're supposed to make an association between an odor and a heat shock. And essentially we're going to measure learning with this assay. What I want you to focus in, trying to keep track of time, is that they tested different groups, different larvae, Drosophila melanogaster. And the most important thing that they got from this was that flies that had been chronically exposed to ethanol versus flies that hadn't been exposed to ethanol at all both learned the same way. They both made the association that is here. If you want to hear more about it at the end, I'll gladly explain it. But for now, it's just important that you know that they make an association. And if they do, they have a positive learning index, which means that they learned correctly. The other groups, without going into much detail, were withdrawal group, which were flies that had been grown in ethanol containing food for their larval stage. And once they were taken out of the ethanol for 6 hours, they did not learn. So this was the withdrawal group. This was able to evidence what happens essentially when someone, or essentially in this case, well, a fly, is adapted or addicted to ethanol, and we take the flies out of the ethanol, or for a person that stops drinking, that's what essentially what happens, the withdrawal symptoms and all these other associated symptoms. Another group that was important was the reinstatement group. Okay. Yeah, it was a group of flies that had been grown in non-containing ethanol food and then placed in ethanol for 6 additional hours. That group additionally did not learn well. So what they saw is that it was curious how the group that had grown in ethanol did not have any cognitive disability, or essentially no learning impairment. So what we got from this study is 3 main questions arose. The first one being, how does gene expression change when we compare the two groups that learned, the flies that had been grown in ethanol and the flies that hadn't been grown in ethanol for their whole larval period? How does gene expression change? What genes may be contributing to that adaptation? What other behaviors change? Could be sleep, could be learning and memory as they tested here, but in the adult, or preference for ethanol or other odors, which you will see here. And last but not least, how do neurophysiological morphology changes? How do electrophysiology properties change? And so the first thing we did is an RNA-seq on the larval brain, comparing the two groups that had been grown in ethanol and the group that had not been grown in ethanol. When we compared the two groups via RNA-seq, we got 193 downregulated transcripts and 258 upregulated transcripts. And again, without going into too much detail, these are some of the genes that were most impactful to us. It's very biased, this table right here, because obviously there's a lot of genes. There's about 400 genes, but these are some that we recognized at the instant, like, for example, calcium calmodulin dependent protein kinase or synaptotacmin. These are all probably, you recognize them, being in from neuro. So they definitely raised some flags, and they are candidate genes that we're going to look into further in the study.
Amanda, if you can hear me, your connection seems to have dropped out because I can't hear you. Your video is frozen.
Adults. Oh, okay. Is everything okay now? No? Okay.
I'm sorry, everything is okay now, but if you could start back at the top of the slide.
Okay.
We lost you for a minute. Great.
Okay. So we're still in the preference to ethanol and other odors, like, correct?
Correct.
Okay. So basically what I was saying is that Robinson, the study where we saw the preference and the learning assay with the larvae, that was in larvae. And we decided to test preference to ethanol and other odors. Yeah.
I'm sorry, your connection dropped out again. Why don't you, yeah, why don't you try turning off your camera? Maybe that might help bandwidth issues.
Okay.
And then we'll start at the top of this slide again because we missed it both times.
Oh no, it says Zoom meetings is not responding. Okay, I'm gonna try something.
Okay. We have a little wiggle room still, so no worries. In the meanwhile, since this is an international conference, I like to play this game. And oh, great.
Yeah, I'm back.
Yeah. Okay, and
Yes.
can your screen still seems to be frozen?
Yeah, I know.
Okay, great.
Let me try this here. I'm so sorry this is happening.
It's you know, we're all used to, this is a part of life these days, so it's okay.
Okay, so I'm going to leave my off so that the internet does not bother us again.
Great.
But I'm gonna go back to sharing really quickly.
Okay.
Let me know when you can see it.
I can see it. I see the wicked background and now your slides.
Awesome for recognizing that was wicked. Okay. So we left off in this slide, correct?
Yeah. Yes, at the top of the slide again, please.
Okay. So really quickly, once again, we essentially just wanted to test preference to ethanol and other odors in the adult rather than the larvae. And so we prepared this little assay that you see here where one of the odors goes from one side, the blue side specifically, for 7 minutes. And then we just see what happens. Are they more attracted to the odor or not? We measure, we take a picture of the flies in the assay and we kind of count which flies are closer to the assay and the part where the odor is coming out of. And we just calculate a preference index and we put together some graphs for you here. So the first one that I'm going to show you is preference to ethanol, which is the one that's on the top in red and black. And essentially what we saw is that there was a reduction in the attraction to ethanol in the female flies, not in the male flies specifically, only in the female flies, not the male flies, which is very peculiar. Because usually the females will go towards the ethanol to lay eggs, female Drosophilas. So it's very weird that we see that after the ethanol exposure and the ethanol, growth, they are less attracted to the ethanol. So the next preference that we measured was vinegar, and vinegar is also peculiar because vinegar is a naturally attractive odor to Drosophila. And we saw that there was actually no reduction in the female, but we had a reduction in the attraction in ethanol males and experimental males, but not in the females. Again, this is really fascinating because vinegar is naturally attractive. So it's kind of weird that suddenly they're not as attracted when they've been grown in ethanol. Last but not least, preference to benzaldehyde. Benzaldehyde is naturally aversive to Drosophila. And once again, we have a difference in preferences in the males rather than the females. And in this case, they are more attracted to the benzaldehyde rather than less attracted. And as I said, benzaldehyde is naturally aversive. So this is also kind of strange, but cool. And so that's basically it. If you have any questions or any comments, suggestions, I'll gladly take them and I'll go back to the Zoom call.
Okay, great.
Sorry that there was so many mistakes.
Oh, good job for getting through it. That's the best we can do in these situations. And this is about the 10th time today, so no worries. Yeah, I have a question. So that's a very unusual effect that larvas that were grown under the ethanol condition had really no deficits in learning. I'd just love to hear more about that. If that's an effect that's been seen in various fly strains, any other species. Yeah, if you could just tell me a little bit more about that and what it means.
At least I don't know about many species, but the first species that I think will raise a little light bulb in everybody's head is humans, actually, because we're comparing how learning deficits actually happen in humans as well. And if you compare it to a human that's addicted to ethanol, compare this, everybody who's watching, to functional alcoholism or functional addiction. When an addict is, well, addicted to a substance, they will essentially function very well under the influence of that substance. However, when you take the substance away from them, just as we saw in Robinson's experiment, when we took away the alcohol from the flies, they seem to have not necessarily learning deficits, but other kinds of deficits, sleep deficits, learning deficits as well. So there's a lot involved in the different deficits that we can see. So yeah, I would have to check out, for example, in mice or some stuff like that. I am pretty sure that I would see essentially the same thing, given that this is a naturally occurring thing when it comes to ethanol and substance abuse. When they are taken in large amounts, you develop the addiction, and more than that, you develop the dependence, and this physiological dependence, that's just the brain's way to compensate for the amount of substance that you're receiving.
Okay, great. We have time for, I think, one more question. Priya is asking a question in the chat. What would you make of reduced preference of female flies to ethanol and vinegar odors? I had the same question. Does this suggest more susceptibility of the females to maladaptive neuroadaptation?
Perhaps. It's a very interesting thing that we really haven't looked at further, but it was surprising. We are redoing the experiments once again with a different assay, just to ensure this is not a thing with the assay, even though the results are clear. But I would say perhaps, because some of the genes that we kind of looked at were involved in oviposition, for example, in odor response. So I'm thinking that ethanol is having maybe some effect in those circuits that are specific to each sex. But that's something that's very early on to say. It's way too soon to tell. But it definitely could be a possibility.
Okay, great. Thanks, Amanda. That's our time for you. Let's move on to our last speaker, Pablo Franco. Pablo is a PhD candidate in the Brain, Mind, and Markets Lab at the University of Melbourne, and he's going to tell us about neural correlates of computational complexity in a problem-solving task. Pablo, take it away, and I'll mute my mic.
Oops. Okay. Thank you, Marina. Okay, I'll not start with a summary discussion. So thanks everyone for coming. I'm gonna try to be as fast as possible and maybe leave some time for questions and discussion. So I'm gonna present to you a study that we've been working on, looking at the neural correlates of computational complexity in a problem-solving task. So in terms of a bit of motivation for this, we are presented on a daily basis with a huge range of difficult problems, and many of these problems are what computer scientists called NP-hard. So they belong to a class of problems that are very hard for computers to solve. And there is, for those that are interested, there's a whole book describing this kind of problems and their difficulty by Van Roy et al. Now, a question arises, and it is how to study cognitive load on complex problem solving. So we're particularly interested in the cognitive control network, but this cognitive control network has been mainly studied by studies looking at perceptual tasks, probabilistic tasks, and not as much as in tasks that required a lot of logical steps and complex problems in complex problems. So we ask ourselves, can we actually quantify cognitive load in these problems and study the neural correlates of this cognitive load metrics. And to do this, we're going to use computational complexity theory, which is a generic framework to study cognitive load or computational effort, if you may, in computers. So previous study has shown that this theory has applications in human decision making, and then they have shown that human performance and the time people spend solving problems can be predicted by metrics from computational complexity theory. But everything that has done so far has been done behavioral studies in humans. Now we're going to study the neural correlates of this metrics of cognitive load in complex problem solving. To do that, we're going to use a problem that is quite ubiquitous and has many applications and in decision theory, and that's called the knapsack decision problem. And I'm just going to quickly introduce it to you. It's imagine you have a bag, and that bag has a weight capacity. In front of you, you have a set of items. Each item has a value and a weight. Your task is, can you put in your bag $22 worth of items without exceeding the weight capacity of your bag? So the answer is either yes or no. In this case, the answer will be clearly yes. You can put $15 and $12 in your bag, and the answer would be yes, you can. However, difficulty can be modulated when we change one parameter that we call constraintness. So imagine that you were asked instead, can you reach $99? So if you reach $99, the difficulty is going to be quite easy for you to say no, because that's just way too much. And on the other side, if you say, can you reach $1? The answer is going to be clearly yes. So it's in this mid-range that problems are difficult for humans and are difficult for computers. So we're going to use this particular metric that has been applied before, and we're going to call it, and it's called typical case complexity. So we're going to call instances in the mid-range that we're going to define them as having high typical case complexity, and out of that mid-range, we define them as having low typical case complexity. Now, we replicated behavioral results that have been previously shown, and basically what you can see here in panel C is the accuracy of people when solving this knapsack problem when we modify this constraintness parameter. So we basically say when you are trying to fit $1, people perform quite well. When you're trying to fit $99, people perform quite well. When you ask them if they can fit $50, they start performing worse. So we're going to use this typical case complexity, which has also been applied to computers. And I'm not going to go into details, but this is basically the amount of time that computers spend or algorithms spend solving the problem. And it also peaks around the same region that people perform worse. So we're going to use this TCC, which I'm going to be mentioning the rest of the talk as TCC. And we're going to scan people while they perform this knapsack problem.
Pablo, quick question.
Yeah, go ahead.
I missed if people have a time cap.
Yes, yes. Yes, so that's exactly this slide. So this is the task itself, what people are seeing in the screen. They have 3 seconds in which they're shown only the items, and then they have 22 seconds to solve the problem. And this 22 seconds, it's a maximum and it's completely set, so you cannot skip time if you want. And at the end, afterwards, you have 2 seconds to answer whether the solution is yes or no. So we collected data from 20 participants, and each participant solved 56 trials of this problem. And we varied the degree of computational complexity according to the definition TCC. They did this while we were scanning them with a 7T fMRI, and this is basically the experimental design, but I'm just going to show you a bit of information on the scanning while they were solving the task, not resting state or connectivity. Just to note, 7T allowed us to have a TR of 0.8 seconds, which means that we have finely grained data points, so we can see actually how people progress through the task, through the whole trial, and how they perform it. Okay, so in terms of results, I have I'm gonna break the task into 5 bits. I have the items part in which they only look at the items for 3 seconds, and then I divide the rest 22 seconds into 4 boxcar or 4 blocks. And I'm gonna only show you now the part for the block 4 because of sake of time. But if anyone's interested, I have also the results for the other boxcars. So this is what you see. This is basically a whole brain corrected activation of the contrast between high typical case complexity compared to low typical case complexity. And what we see is quite a distinctly different activation pattern between the hemispheres. So in the right hemisphere, we can see the regions that we originally were looking for, and we hypothesized in terms of the cognitive control network, which is dorsal anterior cingulate cortex and the right anterior insula. However, we also found quite a bit of activity in the interparietal sulcus and the angular gyrus here on the right hand side, and this pattern shows as well in the task and the other 3 parts of the task, which I haven't shown you. As opposed to that, which is the right hemisphere here, on the left hemisphere we find a bit of activation in the DACC, just as the same cluster as you can see here, and we also find this DLPFC set of activity in the, more specifically, this is a left superior frontal gyrus, and a bit of differential activity in visual cortex as well. But just to show you what we can do actually in terms of looking at the time series of this, and we're going to focus on the right hemisphere and on the cognitive control network that we were interested in, which is the DACC and the anterior insula. So I generated some region of interest, and what I'm showing you here on the left panel is the contrast, but along the whole time of the trial. And this was modeled differently. I'm not going to go into detail at this stage, but basically it means you can just see that the activity starts rising from early on in the trial, and for both dorsal anterior cingulate cortex and the anterior insula, roughly through around midway through the trial. In contrast, you can see underneath here, this is the angular gyrus or anterior IPS cluster, and it does show similar pattern to both. So in order to distinguish and see what the difference between the 3 different clusters and how they go and how they show activity across the whole trial, I separated and plotted only the betas for the high condition and as opposed to the low complexity condition. And this is what I'm showing you here. So here we can see now that there is a differential, there's a difference between activity in the DACC and AI as opposed to the angular gyrus, whereas in the DACC and anterior insula, there's a rise, continuous rise and a peak for a high typical case complexity through the task. For angular gyrus, the peak is at the beginning, and then the difference arises from low typical case complexity activity decreasing. So that's just a quick overview of the results we have so far. This is still a work in progress. But as a quick summary, we find that activity at the end of the trial for anterior insular and dorsal anterior DACC that correlate with typical case complexity. And we see that this pattern starts rising from midway through the task. And from early on in the task, and these are the results I didn't show you, we see activity in DLPFC and as well as angular gyrus and interparietal sulcus. As just an opening here for more discussion is this metric that I showed you is quite generic, so it can be applied to any other problem that is whose solution is yes or no, and has been shown to apply in terms of human behavior. However, we don't know if this is the case for neural correlates, and we don't know if the neural correlates we found are specific to this task and what are actually generic correlates. So this is an open question, and hopefully one will be able to address eventually. And with that, I will just say thank you and acknowledge my supervisors, Carson Maskey and Peter Bosides.
Okay, thank you, Pablo. That was a super interesting task and talk. We have time for only one question, unfortunately, so I'm gonna give it up to the audience member who's asking. Were participant selections recorded before they made up their mind? For example, some people might select items with higher value, and some people might select items with higher value to weight ratio. If so, what do you think is the relationship? What do you think the relationship would be between their selection patterns and the brain activities? That's a good question.
That's a really great question. Unfortunately, it's one we cannot address directly with this study because they did not have any, we did not have any way of extracting data on what items they were selecting, because the only thing that they were able to click while they were in the scanner is whether they clicked yes or no as the answer of the problem. However, we did collect for each of these participants, we did collect outside of the scanner an optimization version of this next problem, and they were able to click around. And we're still trying to understand what strategies people use behind this. However, the beauty of this typical case complexity metric is that it's independent of the strategies that people use. So basically, it has been shown to apply to many algorithms in for computers. So we can extrapolate that it would apply to many algorithms that people use.
Okay. Thank you. Unfortunately, we have to cut it off there. We're super out of time, but thank you, Pablo. Thank you, Priya, and thank you, Amanda, and to all of the attendees and to Amit for running a smooth technical session. Have a good day, everyone, and please join us for the rest of the conference. Bye.